And welcome to Miss Estrick Biology and the big one, the entire paper 3 theory video, which in other words means all of the theory from AQA A-level biology, topics 1 through to 8. It is an enormously long video, so if you're pushed for time, watch it at ×2 speed to get through it that little bit quicker. And don't forget, you can download for free my workbooks that go along with this video so that you can fill in information as you go to create your own set of notes completely for free. If you're more interested though in ready-made resources, I've got you covered. Don't forget, I've got my full theory A-level notes and my A-level theory flashcards, so check this out in the description below if you are serious about boosting your grade in this limited time you have left. But for now, best of luck everyone, and I hope your exams go well. AQA Biology Topic 1. And we start with 3.1.1 monomers and polymers with two very simple definitions that are here for you to learn. Monomer, which is the smaller units from which larger ones are made. The way to remember this is mono means one, so you've got these single smaller units. Poly means many, but the literal definition that they'd want for AQA is polymers are molecules made from a large number of monomers joined together. These are the definitions as they appear in the specification, which means that is what they'd want on the mark scheme. So the monomers and polymers that you need to know for AQA A-level biology are the monosaccharides, for example glucose, and we'll look at all of the other monosaccharides that you need to know, amino acids and nucleotides. The polymers are starch, cellulose and glycogen, which I've put within this row because those are carbohydrates. Then the polymer protein made up of amino acid monomers, and then DNA and RNA polymers, which are made up of the monomer nucleotides. Now there are two key chemical reactions that you'll see come up throughout this topic because condensation reactions are the ones that join molecules together. Hydrolysis reactions are the ones that split molecules apart. But in terms of an exam style answer, here are your bullet points of exactly what you would need to say to get full marks on a question that asks you to define a condensation reaction. It joins two molecules together, it forms a chemical bond, and it involves the elimination of a water molecule. Hydrolysis is the opposite. It breaks a chemical bond between two molecules, meaning it's going to split them apart, and involves the use of a water molecule. Now these are your generic definitions. If you are asked in an exam question to describe how a protein forms or how a disaccharide forms, they would want you to give the definition of a condensation reaction, but in terms of proteins, you'd be saying two amino acids together forming a peptide bond. So basically, if it's an applied example, you have to name the two molecules and name the bond. Same with a hydrolysis reaction. If it was applied example, you should be naming the chemical bond that's broken and naming the molecule that's been split apart and what are the two molecules that's been split into. More on that later throughout this video when we see these applied examples. So that takes us on to 3.1.2 carbohydrates. And here's just a general overview of the different carbohydrates that you need to know for AQA biology. The monosaccharides are the monomers, and it's these three here: glucose, fructose and galactose. Disaccharides are made up of two monosaccharides, and you need to know about sucrose, maltose and lactose. And then the polysaccharides, these are made up of multiple monosaccharides bonded together, and you need to know about starch, cellulose and glycogen. Looking at the prefixes here, we've got mono, that means one, di means two, poly means many. So bear that in mind for all of these. Monosaccharides are one sugar unit, disaccharides are two, poly means multiple joined together. So let's have a look at each of these in the level of detail that you need to know. Starting with the monosaccharides, we have glucose, fructose and galactose like we just said. These are the monosaccharides. And a monosaccharide is the generic term for the monomers from which larger carbohydrates are made. And to turn those monosaccharides or monomers into the polymers, which are polysaccharides, or even into disaccharides, you would have a condensation reaction between two monosaccharides, and the name of the bond is a glycosidic bond. So in carbohydrates, the bonds that you need to learn for how you join multiple monosaccharides together is a glycosidic bond. Now you need to know the structure and the chemical formula for glucose. You don't need to know it for fructose and galactose, but you do need to know it for glucose. So chemical formula, as you learnt at GCSE, 6 carbons, 12 hydrogens, and 6 oxygen atoms. This is the level of detail that you need to know the diagram to show how those atoms are structurally arranged. So you don't literally have to show where all of the carbons are, all of the hydrogens and all of the oxygens. They just want you to see the ones that are going to be of relevant information when we come to the bonding. But just as a bit of information, inside of this ring, this hexagonal ring, we have got these corners, let's call them. And wherever you've got these corners, that is representing where a carbon would be. So this would be carbon, and we actually call it carbon 1. This is carbon 2, carbon 3, carbon 4, carbon 5, and the 6th carbon branches off the top there. But you wouldn't be asked to do that. You just need to know you've got a hexagon, you've got a branch coming off here. Oxygen is at this position in your hexagon. And then on carbon 1, in carbon 4, it's symmetrical. A hydrogen atom on top, a hydroxyl group on the bottom. So you do need to know that that is called a hydroxyl group. Now this is actually called alpha glucose, and that's because there are 2 isomers of glucose that you learn. And for those of you that don't take chemistry, you might be thinking, what is an isomer? Isomers are when you have the same molecular formula, so C6H12O6, but those atoms are arranged in a different structure. So structurally it's different, but it contains the same atoms. So we've got alpha glucose here on the left that we just talked about. Beta glucose is here on the right. And the only difference between them is carbon 1, we have flipped the hydrogen to be at the bottom, hydroxyl to be at the top now. So for alpha glucose, it's pretty much symmetrical, with the exception of the branch there and the oxygen. But in terms of what's on carbon 1 and 4, it's symmetrical, whereas on beta glucose, they've flipped around. And that has relevance to the final shape that polysaccharides have when lots of these bond together. So I'll come back to that when we get to the polysaccharides. But first, it's the disaccharides. We've covered now everything that you need to know for the monosaccharides. Disaccharides are formed by a condensation reaction, or the condensation of 2 monosaccharides. We have a chemical bond, which is a glycosidic bond that occurs. And these are the disaccharides that you need to know. We said maltose, lactose, and sucrose, but here are the word equations. Glucose and glucose is maltose plus water. Glucose plus galactose is lactose plus water. Glucose plus fructose will make sucrose and water. So ways to try and make this a bit easier to remember is, first of all of them contain at least 1 glucose molecule, which is why I put those glucose in bold. Maltose has got 2 glucose, so that's what you need to remember for maltose. Lactose is made up of glucose and galactose. That one's probably an easy one to remember because galactose has lactose within the name. And then sucrose is glucose plus fructose. Other thing I've put in bold here is something else that's the same for all of them. One of the products for all of these is water. And that's because they are joined by a condensation reaction, which we said is the joining together of 2 molecules, forming a chemical bond with the elimination of water. So that is why water is one of the products each time. So those could be a good thing to put on a flashcard just to test yourself until you can remember those word equations. So just to show you these condensation reactions, we can see here we've got 2 monosaccharides. We've shown some of the atoms within them, but the ones of interest are on carbon 1 and carbon 4, the hydroxyl group at the bottom here. And this is where the water is going to be removed from. So I've highlighted in blue the 2 hydrogens and the oxygen to make up the H2O that gets removed. Now it doesn't have to be the OH of carbon 4 and the H of carbon 1. It could be the OH of carbon 1 and the H of carbon 4. I just happen to have highlighted these. But the main thing is it's from the 2 hydroxyl groups that the water is removed. That leaves you 1 oxygen atom behind, and that's where we get this glycosidic bond, the carbon to the oxygen to the carbon. And this is actually called a 1-to-4 glycosidic bond because it's forming between carbon 1 and carbon 4. A hydrolysis reaction, so the opposite of this, it's through the addition of water, a chemical bond is broken, so that'd be breaking this glycosidic bond, and that then splits those two molecules apart back to their original monosaccharides. And we can see here the water has been reinserted as our hydroxyl and hydrogen to make up that other hydroxyl group. So last in the carbohydrates then is the polysaccharides, which are created by condensation reactions between many glucose monomers. And you learn about starch, cellulose and glycogen. Starch is found in plants, as is cellulose. Glycogen is found in animals. And the overall function of these are starch is a store of glucose, cellulose provides structural strength to the cell walls, and glycogen is also a store of glucose. But we can have a look at all of those in more detail. This is your summary table of everything you need to know for the polysaccharides. So take a screenshot or pause, or if you have downloaded the booklets that you can get for free in the link in the description, then pause at this stage and fill in this table in your booklets. So the monomers is one thing that you need to know. Starch has alpha glucose, as does glycogen. Cellulose is beta glucose. Now, as we go through this table, you will notice that everything we say for starch, it's very, very similar to what we say for glycogen. And that's because these two molecules are structurally very similar. And that's because they both perform the same function. It's just starch performs this function of being a glucose store in plants, whereas glycogen, it's a glucose store in animals. So there are a lot of similarities between these two. Cellulose has got a completely different structure because it's got a completely different function. So bear that in mind as we go through. So starch, alpha glucose is the monomer. It's glycosidic bonds, but we talked about a 1 to 4 glycosidic bond when we saw that very basic animation showing a condensation reaction. But you can actually also get 1 to 6 glycosidic bonds. And carbon 6 is the one that branches off the top of the hexagon. So if you get a bond between carbon 1 of one monosaccharide and carbon 6 of another, that creates a branching point in the structure. So we get 1 to 4 and 1 to 6 glycosidic bonds here. Function, we've talked about store of glucose, found in plants, in particular get a lot in the chloroplasts. And then the structure. Although I've talked about amylose and amylopectin here, I haven't actually seen those two terms come up on the new spec yet in a mark scheme. But I'm keeping it in just in case and so that you've got the full understanding. So amylose, this is one of the polymers in starch, which is made up of only 1 to 4 glycosidic bonds between the alpha glucose. And that results in this long unbranched chain, which then coils up to make a helix. Amylopectin is another of the polymers in starch, and this one has got 1 to 4 and 1 to 6 glycosidic bonds. So that means you end up with these straight chains with branches coming off them. Now, the advantage of these two structures linking to the function of being an energy storage is if it can coil up into a helix, because it's a coil, that means you can then compact it down to fit a lot of glucose in a small space. The branch structure on the amylopectin, that provides a greater surface area of exposed ends of the chains. So if you did need to hydrolyze the starch back into glucose for respiration, then having more branches and therefore more exposed ends means that hydrolysis of starch back into glucose happens more rapidly, which is needed for glucose for respiration. And I actually paused and typed that in here because I've remembered they've got really picky on you saying that in the mark scheme in the last couple of years. So you would have to say rapid hydrolysis into glucose for respiration to get that mark. So highlight that in this table as you write it out. Now, it's also a really large molecule, which means it's insoluble and therefore it's not going to dissolve and affect the water potential and therefore have any impact on osmosis. I'm going to jump to glycogen next because that's so similar. Alpha glucose, again, 1 to 4 and 1 to 6 glycosidic bonds. Store of glucose, but this time it's found in animals, mainly the muscle and liver cells. It's a highly branched molecule. So this actually has a higher proportion of the 1 to 6 glycosidic bonds compared to starch. And this means we have even more branches and you get an even more rapid hydrolysis of glycogen back into glucose for respiration. The advantage here is glycogen is the store of glucose in animals, and animals move. They might have to run from a predator, and for that you need muscle contraction, which requires ATP from respiration. So animals need to be able to hydrolyze their glucose store more rapidly for respiration because they might need to run or fight to protect themselves. Plants don't have that, so that's why they don't have as many 1-to-6 glycosidic bonds. But in terms of the structure linking to the function, exactly the same as it was for starch. Branch structure increases the surface area for more rapid hydrolysis back to glucose for respiration. Insoluble, so it's not going to affect the water potential. You don't actually get this coiled structure, so I'd go for these two if I was asked to give marking points for that one. Then cellulose, this one's completely different. It's beta glucose. We have 1-to-4 glycosidic bonds. And this time we have the function of it provides structural strength to the plant cell walls. Now for the structure here, you have long straight chains of beta glucose. And that's because the 1-to-4 glycosidic bonds form directly next to each other, which we saw in that animation when we looked at where the bond forms. So you end up with this long straight chain. The chains are held in parallel, so you have multiple long straight chains held in parallel by hydrogen bonds to make a structure known as a fibril. And one hydrogen bond is not strong, but because there are so many hydrogen bonds, collectively, they provide the structural strength to the plant cell wall. It is also insoluble, so it's not going to affect the water potential. One extra point just to add here about the structure that you'll see is every other beta glucose in these long straight chains is inverted 180 degrees C, which basically means it's flipped upside down. So we can see here the CH2OH is at the bottom, then it's at the top, then it's at the bottom, then it's at the top. So that has to happen in order for you to get this continual chain of 1-to-4 glycosidic bonds and these long straight chains. So that's often worth a marking point as well. So how do we test for these biological molecules then? So the test for starch is you add iodine solution. A positive test observation would be that it goes from this orangey brown colour to bluey black. The test for reducing sugars, you add Benedict's reagent and heat. You need to name the reagent and you have to say heat to get this mark. And we can see here, Benedict's reagent is this blue colour. A positive observation would be that that blue colour turns, and there's actually a range of different colours it can turn depending on the concentration of sugar present. So you can go from green to yellow to orange or brick red, and the more red it is, the higher the concentration of sugar present. So this one here is orange, that's quite a high concentration of reducing sugar present. The non-reducing sugars, which is only sucrose. The reducing sugars is all of the monosaccharides, all of the disaccharides except sucrose. You don't need to learn about why that is the case. You just need to know that sucrose is the only sugar that you learn that is non-reducing, which basically means it can't reduce the copper sulfate in Benedict's reagent. So for the non-reducing sugars test, this would be a longer question because there's more stages. First of all, you have to do a reducing sugars test. And if you get a negative test result for that, meaning it remains blue, that tells you you don't have a reducing sugar. So the next step is then to see, well, maybe it is still a sugar, but it's non-reducing. And the way to test for this is by splitting sucrose, or non-reducing sugar, back into its monosaccharides to confirm that it is sucrose. Now to do this, we have to hydrolyze sucrose back into the monosaccharides. And this is done by something called acid hydrolysis. But the marking point would be add acid and boil, or boil in acid. You don't have to name which type of acid. You would then cool the solution and add an alkali to neutralize it, or in fact it goes beyond neutralizing, it makes it slightly alkaline because that is the pH that Benedict's reagent works best in. Then you would have to say add Benedict's reagent and heat. And yes, you do have to say step 4. Sometimes students miss that one out because I think, well, I've already talked about that in step 1, but this is actually now starting again after you've done the acid hydrolysis. And because we cooled it down, you have to heat it up again. So don't miss this mark. Add Benedict's reagent and heat. And this time, the positive test observation, it will go from that blue to probably orange or brick red. You don't usually get the yellows or greens for this. And this is often an application question. And it's because you should have quite a high concentration of sugar now. Because if you started with sucrose, lots of sucrose molecules, which are disaccharides, and then you hydrolyze that into monosaccharides, you've now doubled your sugar concentration, because you might have gone from, let's just make this simple and say 10 sucrose molecules. After we hydrolyzed that now made 20 monosaccharides. So you have a higher sugar concentration. That's why you should get orange or brick red as your positive test result. 3.1.3 is lipids. So the 2 lipids that you need to learn about are triglycerides and phospholipids. Triglycerides are formed by a condensation reaction, and the molecules that you're joining together are 1 molecule of glycerol gets 3 fatty acid molecules attached to it. So you actually have 3 condensation reactions happening here, 1 for each of those fatty acid chains. In a phospholipid, you still have a glycerol molecule, you still have fatty acid chains, but you don't have 3, you have 2, because one of the fatty acid chains is swapped for a phosphate group instead. And we'll have a look at some images of this as well. So the condensation reaction is occurring between the glycerol molecule and the fatty acid for both of these, and the bond that is created is called an ester bond. We talk about R groups with the lipids, and the R group is linked to the fatty acid. And with your fatty acid, the R group can either be saturated or unsaturated, which we'll come back to. But this is just visualizing what we were saying in terms of the structure. So this one here is a triglyceride, tri meaning 3, and we have 3 fatty acid chains. So you have glycerol at the top here. You might be showing it from a different angle, but basically you have a glycerol backbone with 3 fatty acid chains attached to it, which would have been 3 condensation reactions and therefore 3 ester bonds. For the phospholipid, one of those fatty acid chains is not present, and instead we have a phosphate group. So it would have been 2 condensation reactions, 2 ester bonds. And this is just showing you a bit more detail about the condensation reaction between the 1 molecule of glycerol and the 3 fatty acid chains. So here is glycerol. You don't need to learn the chemical structure of it, but we do have glycerol with the hydrogens on this side, fatty acid chains which end in a carboxyl group, C double bond O, OH. And the water is removed from that hydroxyl group and that hydrogen, and we can see that ester bond then is a carbon to the oxygen to the carbon, but with that double bond O and the H and H there. So that forms an ester bond, and they've got that written here, our ester bond. So this is just showing you once more, just to make sure we're all clear on this, because sometimes they do ask you to circle where the water's removed from, where the ester bond would form. So here's glycerol. Here's one of the 3 fatty acids. Here's where we're removing the water from. And that is where the water is going to be removed. There is our ester bond that is made, or the 3 ester bonds. And that means 3 molecules of water have been made because of those 3 condensation reactions. Now we talked about the fact that there are fatty acid chains, and the R group can be either saturated or unsaturated. So R group generally means a variable group, the bit that's different every time. So for all fatty acids, they all start with a carboxyl group, which is the name for this C double bond O, OH. And then you have this hydrocarbon R group, meaning it's just made up of hydrogens and carbons. And it can either be saturated or unsaturated. Saturated is when you only have single bonds between all of the carbons in the chain, and therefore you have the maximum number of hydrogens bonding to it. So it's saturated. But to get the mark, you would have to say only single bonds between those carbon atoms. When you have an unsaturated fatty acid, you have at least one, we've only got one in this picture, but you could have multiple, so at least one double bond between the carbon atoms. And because of that double bond there, that now means we've got bonds used up here, which would have been used to bond a hydrogen. So it is now unsaturated because it's not bonded to the maximum number of hydrogens possible because the bond is used up in that carbon-carbon double bond instead. But for biology, the definition is the parts highlighted in yellow. So you need to know the properties of these different lipids. So triglycerides, they are an energy storage molecule. And the way that the structure links to the function is the energy storage part is the idea that there's a large ratio of these energy storing carbon to hydrogen bonds compared to the number of carbon atoms present. And that, in terms of the chemistry behind this, provides a lot of energy storage. You don't need to know the ins and outs of why, you just need to know that's the fact. There's also a high ratio of hydrogen to oxygen atoms, and they can act as a metabolic water source, basically meaning that triglycerides can release water if they are oxidized. And that's essential for animals in deserts. So for example, camels, their hump is made up of triglycerides, not of water, but they can then break down those triglycerides to get this metabolic water source. Triglycerides do not affect water potentials because they are insoluble in water, therefore has no impact on osmosis. And that's good because we don't want lots of water moving in or out, causing cells to burst or shrivel. Now that is because they're very, very large, but they're also hydrophobic. Now this isn't a term we've come across yet, but hydro means water. Phobic literally means fearing. But in terms of biology, it means to repel. So water repelling, and that's what makes them insoluble in water. Lipids also have a relatively low mass, and that means you can store a lot of these lipids without it increasing the mass as much as, let's say, muscle tissue would, and therefore it's not going to impact mobility as much. So phospholipids, in contrast, these have a very different function. This is just showing us the structure again. We've got the glycerol molecule, two fatty acid chains, and the phosphate group. Now, although this picture is showing one saturated and one unsaturated fatty acid chain, that is not always the case. It's just what this picture is happening to show. So we've got two condensation reactions would have occurred to make two ester bonds. And the key difference here is the properties that this phosphate group provides on what we call the head of this structure. And the head is basically the red circle that we've drawn around the phosphate group and the glycerol molecule. That part is known as the head, and it's described as being hydrophilic. Hydro meaning water. Philic literally means loving, but that's not what we mean in biology. Hydrophilic means that it can interact with water. It's not going to be repelled, instead it can interact. And that is because of this charge on that oxygen in the phosphate group that makes it polar, meaning it's got a charge, and therefore it's going to be able to interact with water, but polar molecules repel fats or lipids. So we've got this hydrophilic head that can interact with water but repel lipids. The tails are known as hydrophobic tails. They don't have any charges on them, so they are non-polar. And hydrophobic tail means it repels water, but it interacts with fats. Now, it's that hydrophilic head and hydrophobic tail that link to the main function, which is the fact that phospholipids form cell membranes. They create this phospholipid bilayer, two layers of phospholipids, and that makes all cell membranes, whether it's cell surface membrane or the membranes on organelles. So in aqueous environments, because of that liquid, that water, the phospholipids will arrange like this. The hydrophilic heads face outwards towards the water because they can interact with it, but they're repelled by the lipids. Whereas the hydrophobic tails are repelled by water on the outside, so they spin inwards and they can interact with the fatty acids on the other side. So that's why it forms this bilayer in this structure. Now, the test for lipids in terms of the biochemical test is you would need to take your sample that you're testing, mix it or shake it with ethanol, then you add distilled water, And a positive test result is white in colour, and we describe this consistency as an emulsion. For this one, it's really important that you don't say add ethanol and water and mix. You have to add the ethanol first, and then you add the distilled water. And in terms of in the mark scheme, they often accept you saying mix and shake at point 1 and at point 2. Whenever I've done this experiment in the lab, I always do it in this order, and you can see you do get that test result there. 3.14 is proteins. So amino acids are the monomers from which proteins are made. And proteins have loads of different functions because there's so many different proteins that can be made. And what's different about all of them is their final 3D shape. And that is what determines the function of a protein. You need to know the general structure of the monomers, the amino acids. So here we have what's known as the central carbon, and coming off that, you have the amine group, NH2, a carboxyl group, COOH. You have an R group, which is this variable group, meaning it's different every time. And because there's 20 different amino acids, there's 20 different options of what that R group could be. And then there's always a hydrogen atom as well. You do need to draw the amine group on the left, the carboxyl group on the right, but it doesn't matter if you have the H or the R on top or on bottom. So the best way that I find to remember this is always draw your central carbon first, which means the carbon in the middle, and then 4 branches coming off it. And then it's just trying to remember amine group NH2 on the left, carboxyl group COOH on the right, R and H top and bottom. So as I said, there's 20 different amino acids, and what is different each time is which side chain they have, which is that variable R group. To form a dipeptide, which means di means two amino acids bonded together, it is another condensation reaction. So we can see we've got the hydroxyl group removed from the carboxyl group of one amino acid, and the hydrogen removed from one amine group of the other amino acid. That is where the water is being removed from. The two molecules being joined together are 2 amino acids, and the bond that is formed is a peptide bond. So that is how we make a dipeptide. A polypeptide means many amino acids joined together via many condensation reactions, and many peptide bonds would form. Now, when you do have your polypeptide chain, that is then arranged into 4 levels of structural arrangement. The final finished protein is either tertiary or quaternary in structure. Primary is the first structure that's made, but it has to be processed, folded and packaged. So the first stage is making it a secondary, then a tertiary, sometimes it's a quaternary. So let's go through what each of these terms mean. The primary structure is the order or sequence of the amino acids in a polypeptide chain, and those amino acids are held by peptide bonds. This used to always come up as a 1-mark question. It'd just be that first bullet point. Now, since about 2023, I've seen it come up as 2 marks, and they wanted you to say as your second mark, held by peptide bonds. So let's add that in. And as a key point, they will underline the word order or sequence. You have to say that to get the mark, because the order that the amino acids form is going to determine the 3D shape that we see in the tertiary structure. So that's the first level of arrangement. That then will go to the Golgi body or Golgi apparatus and gets processed further into our secondary structure, which will either be an alpha helix or a beta pleated sheet. That's the name for these 2 shapes. And what happens is that sequence of amino acids that we saw in the primary structure, that goes and gets coiled or folded into these 2 shapes. And the key thing is knowing those are held in those shapes by many hydrogen bonds. So your key marking points of this one would be stating alpha helix and beta pleated sheet held by hydrogen bonds for your 2 marks. And you could be asked, where do the hydrogen bonds form? So it is between different amino acids, and it is between the oxygen and hydrogen, the oxygen in the C double bond O of a carboxyl group, and the hydrogen in an amine group. So that's what I'm showing you here. These pink dashed lines are representing hydrogen bonds forming between different amino acids, between the oxygen of the carboxyl group and the hydrogen of an amine group. Then we get the tertiary structure. So this is often a 3-mark definition. It's the further folding of that secondary structure. So again, this is happening in the Golgi body where you process and package proteins. That forms this unique 3D shape. It doesn't look a lot there because it's just a load of squiggly lines, but that is folding on top of folding to create a 3D shape. And then the 3rd mark, you have to name the different bonds that hold that 3D shape in place. So you have ionic bonds, more hydrogen bonds, and sometimes you get a disulfide bond. Disulfide bonds means 2, di meaning 2, sulfide, so it's the element sulfur. So if you happen to have an R group that contains sulfur and you have 2 of those in your chain, then you can get a disulfide bond as well. But you don't always get them because you only get those disulfide bonds if you do have 2 R groups that have sulfur. Just to point out here, I've noticed I've used different spelling. It used to be disulfide was the British spelling with a PH, disulfide with an F was US spelling. They have now just used the F spelling universally. So PH isn't actually used, F is. You wouldn't get penalised in biology, but that's just why I've got that disparity there. Okay, lastly then, quaternary structure. This is when you have a protein made up of more than one polypeptide chain. And in this example, I've picked 4 because that links to an example that you learn later on in the course for hemoglobin. Each of those polypeptide chains is still folded up into a tertiary structure though. So this is still going to be a unique 3D shape. It's just you've got more than one polypeptide chain doing that. So the importance of the primary structure here, which was our first one, that unique sequence of amino acids held by peptide bonds, is even one amino acid being in a different position or different sequence will mean that the bonds that would form in the tertiary structure, so those ionic, hydrogen disulfide bonds, would be in a different location. And that's because these bonds, ionic bonds, disulfide bonds, they occur between the R groups. So if you're moving the amino acids in the chain, you're moving where the bonds are going to form between the R groups. And if you have bonds in a different place, that causes a different folding shape, and that's why you end up with a different 3D shape. So this comes up a bit later, more synoptically, when you learn about mutations in topic 4 and topic 8, because the sequence of amino acids in the primary structure is coded for by the sequence of bases in DNA. So if your DNA mutates and you get a different base sequence, you might code for a different amino acid sequence. Therefore, these bonds occur in different locations. You get a different 3D shape. And if that happens to an enzyme, for example, and you get a different 3D shape, that now means you've got an enzyme with a different shaped active site and it's not going to function. And that's what we're just showing here. If it was carrier proteins, though, or channel proteins that you have embedded in the cell surface membrane, if they change shape, then the molecule that they transport might no longer be complementary and it can't be transported across the membrane anymore. So that bit I've actually already said. Mutations could cause this. So test for proteins then is this bright blue solution biuret and it turns purple. No heat is needed, but the key thing to remember here is the spelling. Everyone that takes chemistry, you are familiar with a burette that you use in titrations. One common mistake here is writing burette instead of biuret. They will accept if you've spelt it incorrectly, but phonetically it sounds the same. But I just split it up here just to try and help you remember. Biuret. Don't say burette, you're going to lose the mark. Okay, enzymes. This is still within the proteins topic. This is one of the key examples that you need to learn about. Enzymes are proteins that have a tertiary structure, and their role is to catalyze reactions, and they do this by lowering the activation energy, meaning the amount of energy needed to start the reaction. And whilst enzymes are pretty big proteins, it's only the active site, that small part here, that is of importance, because that's where the substrate binds. So the active site is specific and unique in shape, And that is because of everything we just talked about there in protein structure. That unique sequence of amino acids, determining the exact location of the tertiary structure bonds, like the ionic and disulfide and hydrogen bonds, causes a particular 3D folding in the tertiary structure to create that unique shape active site. And due to this specific active site, enzymes can only bind to a particular substrate, and therefore they only catalyze one particular reaction, and they will create this enzyme substrate complex. Key marking term, that enzyme substrate complex. And once you get that complex, that is what lowers the activation energy. Now there's two models of enzyme action to explain how the enzymes lower this activation energy. You learn the lock and key model at GCSE. You now learn the induced fit model at A level as well. And this is the accepted model now, the induced fit one. But you need to have an awareness that the lock and key used to be the accepted one. So lock and key, just briefly, this is the idea that the enzyme is like the lock, substrate is like the key, and they fit due to being perfectly complementary in shape. The enzyme never changes shape. So the enzyme active site is a fixed shape. Due to random collisions, we would get the substrate binding, you get an enzyme substrate complex, and once the enzyme substrate complex is formed, the charge groups within the active site are thought to distort the substrate and therefore lower the activation energy. And then the products are released, the enzyme active site is empty, and the enzyme can be reused. It's not used up in the reaction. The induced fit model is the current accepted model. And in this model, the enzyme is more like a glove and the substrate is like your hand. So when you've got an empty glove, it's not exactly complementary in shape to your hand. But when you put your hand inside of it, it enables the glove to mold around your fingers and then it becomes completely complementary in shape to your hand. That is what is happening in this model of how we think enzymes work. The active site is not actually completely complementary in shape to the substrate until it collides and binds, because when it does that, the active site molds around the substrate and then it does become completely complementary. But because it's molding around it, when it's in this enzyme substrate complex, that is going to be putting strain and tension on the bonds, and therefore less energy is needed to break them. So that is the accepted model. Now we have more of an awareness that proteins aren't completely rigid. There is some flexibility to their structure, and it better explains how the activation energy is lowered. So factors that affect enzymes then are temperature, pH, substrate concentration, enzyme concentration, and also inhibitors. So we're going to go through each of these. Temperature then, if you have too low a temperature, then the enzyme and the substrate don't have enough kinetic energy to be able to move fast enough and have successful collisions, meaning they bind and you get those enzyme substrate complexes. And that's why we can see at the lower temperatures, there's a lower rate of reaction. But as you increase the temperature, the rate increases because more kinetic energy is gained and therefore more successful collisions, more enzyme substrate complexes. So at that point, temperature is the limiting factor, meaning it's limiting the rate, and as you increase the temperature, the rate increases. We then have our optimum temperature, which means the temperature at which the reaction happens the fastest. Beyond the optimum, there is now so much kinetic energy that the tertiary structure bonds will break. So the ionic bonds, hydrogen bonds will start to break. And if you break those bonds in the tertiary structure, the tertiary structure protein is then going to unfold and you lose that unique 3D shape. And therefore the active site's a different shape. The substrate can't bind, you don't get enzyme substrate complexes, and the rate of reaction decreases. With pH, too high, meaning too alkali, or too low, meaning too acidic, a pH, interferes with the charges in the amino acid, and in particular, of relevance is the amino acids in the active site. And this again causes a break in the bonds. So if you've got too high a pH, that means you've got too many OH minus ions. Too low a pH, which is more acidic, you've got too many H plus ions. And because you've got too lots of ions there, whichever it is, those ions interfere with those charges, causing the bonds to break, the ionic bonds to break there, that are holding the tertiary structure in place. So same idea again, the bonds break, that means the tertiary structure unfolds and you lose that unique 3D shape. We describe that as the enzymes denatured, fewer enzyme substrate complexes form, and that's why the rate of reaction decreases either side of the optimum pH. Optimum pH meaning the pH which the rate of reaction is fastest. And the optimum pH is different for different enzymes because some enzymes are going to be working in locations that are very acidic. So for example, the stomach has stomach acid, which is very acidic. So enzymes that work there, such as proteases, are going to have an optimum closer to 1 or 2. But the enzyme amylase has an optimum closer to 8 because the pH of the small intestines is slightly alkaline. Substrate and enzyme concentration then. If there is insufficient substrate, then the reaction will be lower. So we can see that here at lower substrate concentrations, rate of reaction is lower. And that's because if you have less substrate present, you're just less likely to have substrate present to collide with the enzyme active site, and therefore fewer enzyme substrate complexes, lower rate of reaction. So as you add more and more substrate, rate of reaction increases because you're going to have more successful collisions. So at this point, with the low substrate concentration, substrate's a limiting factor. But then we get this plateau in the rate of reaction, and therefore substrate concentration is no longer the limiting factor, and it must be something else. And in this case, it's going to be the enzyme concentration is now limiting, because if you add more and more substrate, eventually all of the enzymes are going to be saturated or in use. Every active site will have a substrate already bound to it. So that's why it levels off. For enzyme concentration, some similar concepts here, because at the low enzyme concentrations, you just don't have enough enzyme there to collide and bind with the substrate, so you have fewer enzyme substrate complexes. So at the lower enzyme concentrations, enzyme concentration is limiting the rate of reaction. But as you add more and more enzymes, then eventually you end up with enzymes with empty active sites, because unless you also add more substrate, then you're going to end up running out substrate because that has been broken down or been turned into the product, and you just have more and more enzymes that are not in use. So it doesn't have any impact on the rate of reaction. Enzyme inhibitors then are molecules that can inhibit or prevent an enzyme from working. And you have competitive and non-competitive inhibitors. A competitive inhibitor has a very similar shape to the substrate. And because it's similar in shape to the substrate, that means it's almost complementary to the active site, and it can bind to the active site to form an enzyme inhibitor complex and prevent enzyme substrate complexes. And in this way, it reduces the rate of reaction. But if you add enough substrate to the reaction, that can actually knock out the inhibitor, and then the substrate can bind again. So that's why this one's known as competitive inhibitors, because they're both competing for the active site. Non-competitive inhibitors bind to the enzyme at a point that is not the active site, which is technically called an allosteric site, but you don't have to know that term to get the mark. You just need to know that it's a point that is not the active site. But when the inhibitor binds to that point, the allosteric site or not the active site section, it causes a change in the shape of the protein, so a slight change to the tertiary structure. And because we've slightly changed the tertiary structure, meaning that unique 3D shape, the active site changes shape, and therefore the substrate is no longer complementary and it cannot bind. So you still have this enzyme inhibitor complex, and you cannot form enzyme substrate complexes. And no matter how much extra substrate you add, that isn't going to reverse this. So this can be demonstrated with this graph here. We've got rate of reaction against substrate concentration. The blue line is showing us if there was no inhibitor, and that links to what we talked about previously with low substrate concentration, fewer collisions, but then it plateaus because you don't have enough enzymes. But in this one, we've now can see with both a competitive and non-competitive inhibitor, the rate of reaction is lower at almost all points until this bit here. And we can see that because the curve has shifted to the right. Now, the competitive inhibitor, when you get to the high substrate concentrations, it does actually have the same maximum rate of reaction when you have a high enough substrate concentration. And that's because the high concentration of substrate will knock the competitive inhibitors out of the active site. Enzymes can then all start working again, and therefore you get the same maximum rate of reaction. Whereas a non-competitive inhibitor, it doesn't matter how much extra substrate you add, it's going to be plateauing at a much lower rate of reaction because as that non-competitive inhibitor binds to the allosteric site, changes the shape of the active site, and therefore the enzymes can't work any faster because the active site isn't going to be in use. So this might be some of the enzymes haven't actually got a non-competitive inhibitor bound to them. That's why you get some rate of reaction still rather than 0. Next then, 3.1.5 nucleic acids. So deoxyribonucleic acid or DNA and ribonucleic acid RNA are important information carrying molecules. These two molecules will have the same function in any living cell that they're found in. DNA holds the genetic information, whereas RNA transfers the genetic information. Ribosomes are formed from RNA and proteins. That's such a small spec point that sometimes gets forgotten, but it's come up a couple of times now as a 1-mark question, so just remember that fact. And both DNA and RNA are polymers made up of the monomer nucleotides. So here we see a generic nucleotide. It has a phosphate group, a pentose sugar, and a nitrogen-containing organic base. But for DNA, you need to know that that pentose sugar is deoxyribose and that the options for the nitrogen-containing organic base are adenine, guanine, cytosine, and thymine. For RNA, the pentose sugar is ribose, and you still have adenine, guanine, and cytosine, but instead of thymine, you have the base uracil. So those are the two differences between the nucleotides, which pentose sugar it is, and thymine in DNA versus uracil in RNA. The polynucleotides then, both DNA and RNA form polynucleotides, which means a polymer made up of those nucleotides. And the way that they form is exactly the same as well. It's a series of condensation reactions, and you get a phosphodiester bond. That is the name of the bond that forms, and it's between the pentose sugar and the phosphate group. So for DNA, it's between the deoxyribose and the phosphate group. For RNA, it's between the ribose and the phosphate group. And this is a very strong covalent bond, which is important because we do not want this polynucleotide to break down, because the sequence of these bases is what is going to code for the sequence of amino acids when we come to creating proteins. So a few more things about this molecule then. A DNA molecule, so we're just focusing on DNA now, this is actually made up of 2 polynucleotide chains, and they join together by hydrogen bonds to form this shape here, which is a double helix. And you have complementary base pairs opposite each other, which is where the hydrogen bonds form. In contrast, an RNA polynucleotide molecule, that is just single-stranded, and they are much shorter compared to the length of a DNA polynucleotide molecule. So let's have a look then at this complementary base pairing. Paying close attention as well to the complementary spelled with an E, not an I. This type of complementary means like opposites attracting. Complementary with an I means saying something nice to someone. So we have our complementary bases here. Cytosine and guanine are complementary base pairs, meaning if they align opposite each other, hydrogen bonds can form. Adenine and thymine are the complementary bases as well in DNA. Adenine and thymine can form 2 hydrogen bonds, whereas guanine and cytosine can form 3. So this complementary base pairing is really important because it helps to maintain the order of the sequences of bases when DNA replicates, and also when mRNA is being created, to help ensure that the correct base sequence is maintained so that it codes for the correct amino acid sequence. So how the structure relates to the function then. So it's a very stable structure due to the phosphodiester bonds in the double helix. This is talking about DNA. The double strand means that when DNA replication occurs, both strands can act as a template. The hydrogen bonds between the two strands are quite weak, which is actually beneficial because it means they can be easily broken. So again, the two strands can separate and both act as a template during DNA replication. It's a large molecule, so it carries lots of genetic information, and those complementary base pairs enable identical copies of DNA to be made, which takes us on to DNA replication. So before any cell divides or replicates, whether that's by mitosis or meiosis, all the DNA in the cell must double or replicate first of all, so that when the cell divides, each new cell has its own copy of DNA. This process of DNA replication is described as being semi-conservative replication. What that means is, in the daughter DNA, which means the new molecule that's made, one strand is from the parental DNA, which means the original DNA molecule, and one strand is newly synthesized. So if we have a look here, this top bit is showing us the parental DNA, and we can see that we're going to make two new copies, and each of those has one strand of the original parental DNA, and one strand is going to be newly synthesized. That's what we mean by semi-conservative replication. So some key things then, before we go through the exact process. This is dependent on complementary base pairing. So again, just a reminder, it's adenine and thymine, cytosine and guanine, and there's two enzymes involved, DNA helicase, DNA polymerase, both of which you're commonly assessed on knowing what those enzymes do. So that's a key thing to make a note of here. Starting with DNA helicase. This is the enzyme that breaks the hydrogen bonds between complementary bases in the between those two polynucleotide strands. And that then causes the two strands to separate and also the double helix to unwind. Then within the nucleus, because this whole process happens in the nucleus, there are free floating DNA nucleotides that are attracted and align opposite to their complementary base pairs. Then DNA polymerase can catalyze the joining together of these adjacent nucleotides. So DNA polymerase is catalyzing the condensation reaction and the forming of the phosphodiester bond to create that new DNA polynucleotide chain. Now, the evidence for semi-conservative replication is linked to, first of all, Watson and Crick, who discovered the structure of DNA in 1953, but they were helped significantly by Rosalind Franklin's research on X-ray diffraction. And then they hypothesized that DNA must replicate either conservatively or semi-conservatively. And it was Michelson and Stahl who then conducted experiments to determine it is semi-conservative replication, not conservative. Now, these experiments I'm not actually going to explain in detail in this video, because this actually comes up on the spec saying as application questions, you need to be able to understand and explain it. But I do have a full video on this, which I'll link here, but just search Miss Estruck, Michelson and Stahl, and I've got a full explanation in detail of this experiment if you've come across questions on this that you weren't sure on. Right, next then, 3.1.6 ATP. ATP is a nucleotide derivative, and it is made up of an adenine base. So it's only got one nitrogenous base and it's always adenine. It has a pentose sugar, but it's ribose, and it has 3 phosphate groups. And its role is it's an immediate source of energy in metabolic processes. And all cells must have a constant, steady supply of ATP so that they can perform their metabolic processes. So let's go through some of the key details then. ATP hydrolysis, meaning the splitting apart of ATP, which breaks off an inorganic phosphate and releases energy. That is catalyzed by the enzyme ATP hydrolase, very similar to the word hydrolysis. So that reaction breaks ATP or hydrolyzes ATP into ADP, meaning diphosphate, T means triphosphate, and PI, which is inorganic phosphate. The energy released can actually be coupled to energy requiring reactions in cells, or that inorganic phosphate that's been broken off can be added to another compound, which is known as phosphorylation, and that then makes that new molecule more reactive. That happens at the start of respiration. Glucose gets phosphorylated, and you'll learn about that in topic 5. ATP can also be resynthesized, which is when ADP and PI join by a condensation reaction. It's also an example of phosphorylation because you're adding an inorganic phosphate onto this molecule. So this resynthesis is catalyzed by ATP synthase, and that happens in both photosynthesis and respiration, both of which come up in topic 5. So here is our word equation. ATP plus water goes to ADP and PI. Energy is released, so it's not actually a product that you'd write in there because you can't produce or use up energy. 3.1.7 is water, and water is a polar molecule. One water molecule, so H2O, has unevenly distributed charge due to the fact that the oxygen is slightly negative and the hydrogen is slightly positive. So this symbol means delta, so delta negative, delta positive, which means slightly negative, slightly positive. And because of that, hydrogen bonds can form between the oxygen and hydrogen of different water molecules. And that concept underpins pretty much all of the properties we're about to see in water. So hydrogen bonds form between those different water molecules, like we just said, and there are 5 key properties of water that you need to know for AQA. I've put them here highlighted in yellow and what each one means. So it's a metabolite, which means it's used in many reactions. In this video alone, we've already seen it multiple times in condensation and hydrolysis reactions. In an exam, I would state that as well. Although it's in brackets here, I would say, for example, condensation and hydrolysis reactions. Number 2, it's an important solvent, meaning you can dissolve solutes in it. And that is really important because metabolic reactions happen in solution. Then it's got a high heat capacity, which means it takes a lot of energy to raise the temperature of water. And that's because of these hydrogen bonds. But in terms of in the exam, they just want you to say that means it buffers temperature changes. Number 4, it has a large latent heat of vaporization. You do have to get this the right way round. High heat, large latent. You can't say high latent. So think of the L's together. Large latent heat of vaporization. That means it takes a lot of energy to convert water in its liquid state to its gaseous state. And again, that's because of these hydrogen bonds. You'd have to have energy to break the bonds to then convert it from liquid to gas. So that means when you sweat, water provides a cooling effect because it takes a lot of energy to evaporate the water from your skin. And that energy comes from the heat in your skin. So it provides a cooling effect. And number 5, it has strong cohesion between water molecules. And that is because of the hydrogen bonds. Cohesion meaning like sticking together with those hydrogen bonds. So because water molecules form these hydrogen bonds between each other, they all stick together and you can get these continuous columns of water inside of plants, inside of the xylem, which comes up in the cohesion tension theory of topic 3. And also it provides surface tension where water meets air, meaning that you can actually balance some items on top of water, and that provides a habitat for organisms. Last bit of topic 1 is 3.1.8 inorganic ions. Inorganic ions are found in solution in the cytoplasm and in other body fluids. They're present in the body in varying concentrations depending on different ions, which they are. Each ion has a specific role, and most of these come up throughout the A level in different places. We've briefly talked about in this video the role of hydrogen ions, because we talked about that in terms of enzymes and how lots of H+ means that you have a more acidic solution or lower pH. So hydrogen ions affect pH. Iron ions comes up more in topic 3 when you learn about hemoglobin, because hemoglobin contains iron ions and is actually the part of hemoglobin that oxygen binds to, so it's involved in oxygen transport. Sodium ions come up in multiple places in the A level. The first one you're going to see is in topic 2 for co-transport, and then again in topic 3 because you have co-transport in absorption of digested molecules. So glucose and amino acids are transported from the ileum into the blood by co-transport. Phosphate ions has come up in this video because they're components in DNA, RNA and also ATP. AQA, a topic to the entire topic. Let's get into all of this information then, starting with 3.2.1, which is cell structure. For the eukaryotic cells, these are the 10 organelles that you need to know both the structure and how that structure links to the function. So we're going to go through all of these. Here's just showing you an overview though of what they look like inside of the cell. So we have our cell membrane on the outside. Now although it does say vacuole here and it's an animal cells don't have permanent vacuoles, but they can have temporary vacuoles, things like your phagosome that you get in phagocytes, but we can kind of ignore that for the sake of this slide. Rough endoplasmic reticulum, so you've got these folded membranes with the ribosomes on the outside, so they look rough or bumpy. Golgi apparatus, the one that looks like a Wi-Fi symbol. The cytoplasm, lysosomes, ribosomes, which are the smallest, and then smooth endoplasmic reticulum and the mitochondria. So centrosome is not on the AQA spec. In plant cells, they have all of those structures plus 3 additional ones. They do have a permanent vacuole, which is surrounded by a membrane called a tonoplast. They have the chloroplast, which is the site of photosynthesis, and then they also have the cellulose cell walls. So if we have a look at the nucleus first of all, the structures and the functions. The nucleus has got an envelope, which is this membrane around the outside, this double membrane, and there are holes within that called nuclear pores. The liquid in the middle, or the jelly-like material, is the nuclear plasm. You have the chromosomes within the nucleus, which are protein bound and described as being linear DNA. And then we've got the nucleolus, which is this additional small sphere inside of it, which in terms of the function, really that should be on that side, but the function is the site of RNA production and it makes the ribosomes. So if you were asked to describe the functions of the nucleus, one of them we've already said down here, RNA production, and it makes ribosomes. It also contains all the genetic code for each of the cells, and it's the site of DNA replication and transcription, which is the making of mRNA. Endoplasmic reticulum can be rough or smooth, and both of them are made up of folded membranes called the cystinae. But the rough has ribosomes attached to the outside, which is what makes them rough. So here it is under the microscope, and all those tiny black dots are the ribosomes, giving that rough feel or rough look. The function of the rough endoplasmic reticulum is for protein synthesis, and the smooth endoplasmic reticulum is the synthesis of the lipids and carbohydrates. The Golgi apparatus and then the Golgi vesicles that pinch off. First of all, the structure though, it's folded membranes making these cystinae again, and it creates this Wi-Fi symbol is what I hear most people describe it as, because you've got these curved folded membranes and then the vesicles bubbling off. So those secretory vesicles pinch off from the cystinae. There's a whole range of functions for the Golgi apparatus and the vesicles. So you could have carbohydrates being added to proteins to form glycoproteins. You also get secretory enzymes being made, secrete carbohydrates. They transport, modify and store the lipids. They form lysosomes. Molecules are also labelled with their destination here, meaning you might add on some sort of chemical tag so that it can only bind to a target cell. And also we've got the finished products are transported to the cell surface through these Golgi vesicles. They move up and fuse with the cell surface membrane, and that then releases the contents of the vesicle. The lysosomes are these bags of digestive enzymes. We can see one here. So just like sacks filled with digestive enzymes. So all of their functions are going to be to do with hydrolyzing and digesting. So they could be hydrolyzing pathogens inside of the phagosome. So in your phagocytes in topic 2, which we'll get into later on in this video, you've got your phagosome that has got a pathogen engulfed within it. The lysosome contains digestive enzymes, it fuses with the phagosome and that digests the pathogens. It could be to completely break down dead cells within the body that you don't need anymore. You can have exocytosis, meaning the release of those digestive enzymes to outside of the cell to destroy material. And also it might be to digest worn out organelles so that you can reuse those materials. The mitochondria is a double membrane bound organelle. We've got the outer membrane here, and the inner membrane is folded. It's shown as this bright yellow structure here, folded so that you've got a really large surface area for some of the stages of respiration. So that folded inner membrane is called the cristae. Inside of that, you have a fluid-filled centre, which is known as the matrix. You learn more about this in topic 5 when you learn aerobic respiration. You also have loops of DNA and 70S ribosomes, which are the smaller ribosomes that you typically get in prokaryotic cells. And you need both of these structures so that within the mitochondria, you can have the creation of proteins, for example, enzymes that are needed for aerobic respiration. So the function then is it's the site of aerobic respiration, not anaerobic, just aerobic, which means you have lots of ATP being produced here, and also we get enzymes being created for respiration. Ribosomes, these are small, they're the smallest of the organelles, made up of 2 subunits of protein and RNA. Those are the 2 molecules that a ribosome is made up of. You have 2 different sizes, ATS, which are the larger ones, which are found in eukaryotic cells, and 70S are smaller in size, which are found in prokaryotic cells, but also inside of the mitochondria and chloroplasts in eukaryotic cells. And the function is for protein synthesis, meaning making proteins. Then if we have a look at a vacuole, so we're thinking about a permanent vacuole that you get in plants here. This structure is filled with fluid and it's surrounded by a single membrane, which we can see here as this darker layer, which is known as a tonoplast. The function is by having liquid in the centre, it pushes everything outwards and that makes the cell turgid and therefore it provides support. It can also be a temporary store of sugars and amino acids, and the pigments that may colour petals to attract pollinators can be found there as well. Next then, chloroplasts, which again are found in plants. This is very similar in structure to a mitochondria. It's also double membrane bound, but this time we have the outer membrane and the inner membrane directly next to each other, both creating a sort of oval shape. Instead, these folded stacks are additional membranes called thylakoids. So we've got these folded thylakoid membranes that fold up to create almost like pound coin or disc-shaped stacks. And those stacks are known as granum for singular or grana for plural. You learn more about that when you get on to topic 5 for photosynthesis. You also have a fluid-filled section here, which is known as the stroma. And the function is photosynthesis. Cell walls, which are found in plants and fungi, but also in bacteria, but we'll come back to bacteria in the prokaryotic cell section. So the cell wall in plants is made of cellulose. The cell wall in fungi, which are actually also eukaryotic cells, is made of chitin. And whatever it's made of, the function is the same. It provides structural strength to the cell. Plasma membranes, this is found in animals and plants. In fact, it's found in all living cells. So it's the phospholipid bilayer, which we can see here. We've got one layer of phospholipids a second. We've got the hydrophilic heads on the outside, hydrophobic tails pointing inwards. And within the plasma membrane, we also have proteins embedded. We have some carbohydrates on the outside as well. And you also have a lipid called cholesterol embedded within. And the function of the plasma membrane is to control what can enter and exit the cell. And this comes up again later in this video when we look at transport across membranes. So that's the eukaryotic cells and their organelles. Next, let's have a look at prokaryotic cells, for example, bacteria. And we can see a range of the organelles here. Some on here you don't need to know. But you do need to know that it has a plasma membrane, a cell wall. Sometimes it has a capsule. You don't need to know the pili for AQA. We've got a flagellum here, which some of them have, and some of them may have one or more plasmid. You do need to know that they have cytoplasm, ribosomes, and then the DNA is as a loop of DNA in the cytoplasm. So let's have a look at that information. The key differences then are between a prokaryotic cell and a eukaryotic cell. Prokaryotic cells are much smaller. In exams, they insist that you say much smaller, not just smaller. They don't have any of the organelles that have membranes, and we describe that as membrane-bound organelles. They do have ribosomes, just like eukaryotic cells, except they're smaller in size, they're 70S. They do not have a nucleus. Instead, their DNA is found as a single circular DNA molecule in the cytoplasm. They do have a cell wall, but it's made up of a glycoprotein, not cellulose, not chitin, and that glycoprotein is called murine. Some of them might also contain any of these three extra structures. They might have one or more plasmids, which are small loops of DNA that only have a few genes on them, often the genes for antibiotic resistance. Some of them have a capsule around the cell wall as well. This prevents the cell from desiccating, meaning drying out, and also it helps to cover the antigens to evade the white blood cells. And they might have one or more flagella, which is for movement or locomotion. Viruses, these are not classed as living, so they're non-living and they are acellular. This has come up as an exam question before to define what those two terms mean. So non-living, it's classed as not being alive because it doesn't do any metabolic reactions within the structure or within that particle. It cannot move, it cannot respire, it cannot replicate without using a host, and it has no means of getting nutrition. It's called acellular, meaning it's not a cell, basically, and it's not classed as a cell because it doesn't have a cell membrane. Viruses are even smaller than bacteria, and they only contain three structures. They contain the genetic material, capsid, and attachment protein. The genetic material is this here in the middle, and that can be DNA and it can be RNA. It'll be one or the other. That genetic material is surrounded by a protein shell called the capsid, not to be confused with the capsule that some bacteria have. So capsid is the protein shell that protects the genetic material. And then the only other structure that all the viruses have are these attachment proteins on the outside, and that's what they use to attach to receptors on host cells. Now we can see additional structures in this picture. We've also got an envelope, and some viruses do gain a lipid envelope by taking it from the host cell that they infect. They aren't able to produce their own one. So that is the three things that all virus particles contain. And that's what we call them, a virus particle, because it's not a cell. So next then is looking at how you can study cells and how this has evolved so that we've been able to understand these organelles that different cells have. And you can use a microscope, cell fractionation and ultracentrifugation. These are things that we're about to go through. The types of microscope, magnification calculation, calibration calculations linked to this idea of the eyepiece graticule, and then cell fractionation and ultracentrifugation. Starting with these two key definitions, knowing what's meant by magnification and resolution. Magnification of a microscope refers to how many times larger the image size is compared to the actual size of the object you are viewing. Resolution of a microscope is the minimum distance between two objects in which they can still be viewed as separate, and therefore you get a clear, crisp image still. The resolution of any microscope is determined by the wavelength of the energy source. So for an optical microscope, light, visible light is the energy source, so the wavelength of visible light is what determines the resolution. For an electron microscope, beams of electrons energy source, so the wavelength of electrons is what determines the resolution. So if we have a look then at some of these differences between optical, which are the light microscopes, and electron microscopes, which can be scanning or transmission. First then is what it is that creates the image. So for light or optical, it's a beam of light, which is condensed to a fine point using a glass lens. An electron microscope, it's a beam of electrons, which is condensed to a fine point using an electromagnet. Optical microscopes have a much poorer resolution, and that's because the wavelength of light is longer. Electron microscopes have a much higher resolution or resolving power, and that's because electrons have a shorter wavelength. Optical microscopes have a lower magnification, and electron microscopes have a higher magnification. Optical, you can get colour images, whereas you can only get black and white images with an electron microscope. You can then artificially add colour to that image using different Photoshop kind of programs, but with the actual microscope, you only get black and white. And then optical microscopes, you can view living samples, but you can't in an electron microscope. And that's because when you're preparing the sample, it all has to be inside of a vacuum, meaning there is no air. And that's because the air would absorb the electrons and then you wouldn't get an image. So it has to be a vacuum. But because it's in a vacuum with no air, that means you can't create samples with living things because they would die. So just to see an image here, we've got what we can see with an optical microscope. Because they have a much poorer resolution, that means that we can't see the small organelles in a cell. We can see the cell membrane, not in detail, but we can see it's there, cytoplasm and the nucleus and the chromosomes when they condense, but we can't see all of the organelles and we cannot see inside the organelles, but we do get a colour image and we can look at living cells. With an electron microscope, the electrons, as we said, can be absorbed by air, so it has to be prepared in a vacuum, which is why we can't have living samples and it's why it's black and white. A transmission electron microscope, these are the ones where the electrons transmit, meaning pass through the specimen. And that is why we have to have extremely thin slices of the specimen. They are stained and placed in the vacuum, and an electron gun produces a beam of electrons that passes through the specimen. Some parts of the specimen absorb the electrons and appear darker. So that's how we get these different grayscale, black and white parts of the image. So you end up with a 2D image, but you can see inside of the organelles. So this here is a chloroplast, and we can see the membrane, we can see the thylakoids, we can see those stacks, the grana, so you can see details internally. A scanning electron microscope, the specimen does not need to be thin because the electrons are not passing through the specimen. Instead, the electrons are beamed onto the surface and the electrons scatter back. And because they scatter back in different places because of the contours, you end up with these 3D style images. You can't see the internal structures because the electrons bounce back or they scatter back, but you can get an idea of the contours creating a 3D image. The math skill linked to this then is magnification, and it's being able to calculate the magnification, or you might have to rearrange the formula to work out the actual size. So the key thing is knowing the formula. I always remember it as I am, so image equals actual size times magnification, and then I'd rearrange it if I needed to work out magnification. And you usually have to do unit conversions with this because most organelles, the actual size will be measured in micrometers because they're very, very small structures. But when you're measuring your image size, that is usually going to be in millimeters. So that means when you do your calculation, let's say we're working out the magnification and we're doing image size divided by actual, they both need to be in the same units. So you'd either need to convert your millimeter image size into micrometers, and to convert millimeters into micrometers, you would times by 1,000. Or you'd need to convert your actual size, which would be micrometers, into millimeters, and that'd be divided by 1,000. Now you could also be asked to calculate the actual size, and in which case we need to swap actual and magnification here. So actual size would equal image size divided by magnification. And if they want you to work out actual size, they'll probably state that they want the answer in micrometers. So again, you might need to do a conversion. Then we move on to what is the eyepiece graticule. So inside of the eyepiece, which is this part here, there is a scale on a glass disc, which is called the eyepiece graticule. So when you look through the eyepiece, you will see what looks like a ruler. And this can be used to measure the size of objects that you are viewing underneath the microscope. But you need to know what each of these divisions on this graticule or ruler is worth. And that is going to vary depending on what magnification you are viewing the specimen at. So that's why we have to calibrate the eyepiece graticule every time we change the magnification. And when we say calibrate, what that means is work out what each of these divisions is worth in terms of a distance. So the way that we would do that is you have your eyepiece graticule in the eyepiece, but you also place a stage micrometer on the stage, which is this bit here. And a stage micrometer is typically 2 millimeters long, and it has subdivisions 10 micrometers apart. So it's a glass slide that already has known distances etched onto it. So you line up your stage micrometer on the stage of the microscope, and you would maybe need to twist the eyepiece a little bit so that the eyepiece graticule is now completely lined up next to it. And we then need to look at how many divisions on the eyepiece graticule fit into one division on the stage micrometer. And we know that one division on the stage micrometer is worth 10 micrometers. And we can see that we've got 2 fit into one division. So that then means if that is worth 10 micrometers, we need to do 10 divided by 2, which is 5. And that tells us that each of these small divisions on the eyepiece graticule is worth 5 micrometers at that particular magnification. So then anything you view, you can use your eyepiece graticule to measure the size, and then we know that the number of divisions times by 5 is the actual size. And then if you're going to go up and change the lens to a different magnification, you would do this process again to work out what one of these divisions is worth on the eyepiece graticule. Next is cell fractionation, and this is used to isolate different organelles so that those organelles can then be studied. And this has helped us to identify organelle structures and functions. So there's 2 stages to this. We do homogenization and then ultracentrifugation. And homogenization, this is when the cells are broken open to release the contents, and the organelles are then separated by ultracentrifugation. But throughout this process, the cells must be in a particular prepared solution. It has to be cold, isotonic, and buffered. And you need to be able to explain separately for all of these points why it has to be cold, then why it has to be isotonic, and then why it has to be buffered. So because you're breaking open the cell, that means you're going to have some organelles now exposed to enzymes that they might not usually be exposed to in the cell because we've broken it all open. And those enzymes might damage the organelle before we can examine them. So we make sure it's very cold, it's ice cold, to reduce enzyme activity, to prevent any enzymes potentially damaging the organelles before we can view them. It has to be isotonic, which means the water potential of the solution is the same as the water potential inside of the organelles. And this is to make sure that there is no osmosis, because we don't want any water moving into the organelles because that could cause them to burst. And we don't want water moving out because that could cause them to shrivel. And then it has to be buffered, meaning you use something to maintain the pH, because we don't want the pH to change because that could damage the organelles. And the key thing I've underlined each time is organelles. If you say damage to the cell, that is incorrect because we've already broken open the cell. The cell doesn't exist anymore. It's the organelles that we need to now make sure they're undamaged so that we can go on to examine them. Okay, so a bit more about this homogenization process, basically like a blender. And in fact, when I've done this in lessons, that is literally what we used, a blender. So homogenization is when you break open the cells, and we do that with an ice cold isotonic buffered solution. Then you have to filter your solution to remove large cell debris. And then that filtered solution can be spun in a centrifuge at varying speeds, starting at low speeds. And this is going to separate organelles out from that solution according to their densities. So that's what we can see here. Here is our original solution that we've filtered. the centrifugal spin and the centrifugal forces cause pellets to form at the bottom of the most dense organelles. So the first time we spin it, we start at a low speed and then we increase the speed each time. But our lowest speed, that is going to cause the nuclei to sink to the bottom in the first pellet because the nuclei are the most dense. You then remove the pellet and you then put the rest of that liquid or the supernatant into another tube, spin it at a slightly higher speed. And this time you'd get the chloroplast if you're using plants and also the mitochondria because they've got similar density. Again, remove the pellets and the supernatant can be spun again. Next then it'd be lysosomes, after that endoplasmic reticulum, after that the ribosomes. And you do need to remember that order. They have done exam questions where they've asked how would you isolate mitochondria, and for the mark you needed to know it'd be on the second spin. So chloroplasts and mitochondria are both on the second. We then move on to 3.2.2. All cells arise from other cells, or in other words, we're looking at the cell cycle and mitosis. Now, not all cells are able to continually divide in multicellular organisms, but the ones that can go through the cell cycle. And in eukaryotic cells, when they enter the cell cycle, they will then divide by mitosis or meiosis. Prokaryotic cells replicate by binary fission, and viruses do not undergo cell division because they are acellular, meaning they don't have a cell to divide, and they are non-living. Viruses can still replicate though, but only once they are inside of a host cell, which is the cell that they are infecting. And they have to invade by injecting their nucleic acid into the cell to replicate the virus particle there. And that comes up later in this topic in immunology. So binary fission then, we said this is how prokaryotic cells or bacteria replicate. And this involves, first of all, we have the circular DNA and the plasmids, if they have plasmids, will replicate. And that's what we can see here. We've got circular DNA and we've got plasmids. That is going to replicate. The cytoplasm splits to then create two daughter cells. And each daughter cell has one copy of the circular DNA, but the plasmids, if they are present, don't necessarily split equally, so they might get different numbers of plasmids. It's shown here that they have the same, but it could be different. So then let's have a look at the cell cycle. There are three key stages: interphase, nuclear division, and cytokinesis. Interphase is the longest stage, and we can see that here. Interphase is shown here as G1, S phase, and G2. So we can see about 90% of the cell cycle is interphase. And this is when the organelles double, the whole cell grows, and also DNA replication occurs. So G1 of interphase, that is the growth phase. So that is when we're going to have the growth of the cell and also some of the organelles doubling. S phase is when the DNA replicates. And G2, we have more growth. Also, we have checks for any DNA replication copying errors, and we have final preparations for mitosis, or it could be meiosis. And then this stage is our nuclear division, which can either be mitosis or meiosis. In this topic, topic 2, we only look at mitosis, and that is when you create identical diploid cells. Diploid, di meaning two. Diploid cells are ones that have two copies of the chromosomes. Meiosis, which you learn in topic 4, you end up with 4 genetically different daughter cells are haploid. Haploid meaning one copy of each chromosome. Mitosis creates cells with identical DNA, and therefore it's useful for growth and repair, whereas meiosis creates gametes, which are all genetically different. The final stage, which is just shown as this red line here, it's a very, very short stage, is cytokinesis. And this is when we get the division of the cytoplasm and the cell membrane to create the new cells. So we're going to focus on mitosis, which is just one of these stages in the cell cycle. And mitosis is split up into multiple steps as well. We have prophase, metaphase, anaphase, and telophase, or PMAT, to remember the order. Key facts about mitosis: you only have one division occurring, so you go from having one parent cell to two daughter cells. You get identical, genetically identical cells. They are diploid, meaning two copies of each chromosome. We've only got one chromosome shown here, but we can see we've got the red copy and the blue copy. That's in the parent cell, and it's the same in the two daughter cells. And as we said, the function, growth and repair, and we're going to see that later in this topic when we see clonal expansion in immunity. So prophase, this is our first stage. What you need to know throughout mitosis is what is happening to the chromosomes. In the spec, they describe it along the lines of the behaviour of the chromosomes. So that's what we're going to focus on. What's happening to the chromosomes and why? So in prophase, the chromosomes condense and that makes them visible. So that is what is happening. And in animal cells, these centrioles will also move to be at the opposite poles, which means opposite ends. The centrioles are responsible for creating the spindle fibres, and because they move to both ends, they start to releasing it from both poles, and they'll start to attach to the centromere on chromosomes and the chromatids on the chromosome in later stages as well. Plants have spindle apparatus, but they don't actually have centrioles. Metaphase. So in terms of what is happening to the chromosomes will now line up in the middle of the cell, which is known as the equator. Spindle fibre that is released from the centrioles will now attach to the centromere of these chromosomes, and that's what causes them to go in this single file line at the equator. In anaphase, the behaviour of the chromosomes is now that they'll be split apart at the centromere, and then each chromatid is pulled to the opposite poles of the cells. So those spindle fibres that were produced start to retract, and that is what pulls and splits or divides the centromere in two, so that those individual chromatids are pulled to the opposite poles of the cells, so they're now separated. And once they are separated, we can now call those chromatids, once they're at the opposite poles, chromosomes again. This stage does require energy in the form of ATP, which is provided by respiration in the mitochondria. Telophase is our last stage. The chromosomes are now at each pole of the cell and they're going to uncondense. So they become longer, thinner and no longer visible. Spindle fibres will start to disintegrate, the nucleus starts to reform, and then the very last step, which is not in mitosis but it is in the cell cycle, is cytokinesis, which is when the cytoplasm splits, so does the cell membrane, so you end up with two genetically identical daughter cells. You could be asked to calculate the mitotic index, and this is a way of looking at the proportion of cells that are currently undergoing mitosis compared to the number of cells present when you're looking at a sample underneath a microscope slide, which links to required practical too. So in order to calculate the mitotic index, in your field of view, which means the section that you can see when you look through a microscope, you need to count how many cells are currently in mitosis. The way that you can tell that is whether you can visibly see chromosomes, which is squiggly lines. If it just looks like grey, or in this case it's stained purple, if it just looks like dots almost and you can't see any squiggly lines, that's an interphase, so that's not in mitosis. And most cells, if you remember, we said 90% of the cell cycle is interphase, which should mean about 90% of the cells will be in interphase, and only about 10% will be currently going through mitosis. So we'd count how many cells are in mitosis, divide that by the total number of cells present, and that gives you a mitotic index. You might get questions on how can you standardize this. So you could say you're only going to count cells in which you can see the entire cell in the field of view. Now, uncontrolled cell division comes up again later when you learn more details about cancer in topic 8. But mitosis is a highly regulated process, meaning it's very controlled to make sure that cell division is accurate and it's only happening when it's needed. But disruptions to this control do occur, and that then results in uncontrolled cell division, which means you're going to get lots and lots of cells being made, even if you might not need them. And that is how you get a tumour forming, which is just a mass of cells. And those tumours could be benign, which means they're not cancerous, or malignant, which means they are cancerous. And because these tumours are formed because of the uncontrolled cell division of mitosis, many cancer treatments will target mitosis to try and control the rate. Now, this mainly comes up as application questions for topic 4. You learn more theory on this in topic 8 in year 13. 3.2.3 is the next part of topic 2, transport across cell membranes. So this takes us back to the plasma membrane, the cell membrane, which could be a cell surface membrane, but also an organelle membrane. So when we talked about the chloroplast being a double membrane bound organelle, they have one of these membranes. Well, they have 2 of them because it's double membrane bound. So the membrane is described as a fluid mosaic model, and that's due to the fact that it's a mixture of different molecules. That's why it's a mosaic. So it's made up of the phospholipid bilayer. We've got proteins embedded within it, but also just pinching in externally. We have glycoproteins and glycolipids. And it's described as fluid because there is some slight lateral movement of those phospholipids. All of these molecules are arranged within that phospholipid bilayer, and that is what creates the partially permeable membrane, which is that property that we talked about, or the function. It can select or control what can pass through the cell surface membrane. So let's just go through this in a bit more detail. The main component of the membrane is phospholipids, and this links back to topic 1, which if you haven't already seen my entire topic 1 video where I go through lipids, or if you haven't seen my lipids video in detail, then just search misestric lipids or misestric phospholipid bilayer, misestric entire topic 1, you'll find it. So the phospholipids align as a bilayer, bi meaning 2, so 2 layers, that's what it means. And we have the hydrophilic heads on the outside, the hydrophobic tails are on the inside. This is actually zooming in on one phospholipid, and we've got these structures here that need labeling. So 1 is the phosphate group, 2 is the glycerol, 3 and 4 are both fatty acids, but 3 is a saturated fatty acid, 4 is an unsaturated fatty acid because of that double bond between the 2 carbons. A is the hydrophilic head. Hydrophilic because of this negative charge on the phosphate group. And then B is the hydrophobic tail. Those fatty acids are non-polar, so they are hydrophobic, meaning they will repel water, whereas hydrophilic, hydro meaning water, hydrophilic heads will interact with water. So that's why phospholipids in aqueous solution or aqueous environments will create this bilayer. The heads can interact with any of that solution, the water-based solution on the outside, but it will repel the tail, so they spin inwards, and that's how you get this bilayer. Now this goes towards making this partially permeable membrane property because molecules can only move through the phospholipid bilayer by simple diffusion if they can dissolve in lipids and if they're small enough. So it has to be a lipid-soluble substance, which means it has to be non-polar, so it can't be charged. And that's going to be some hormones like estrogen, and also very small molecules like carbon dioxide and oxygen gas are able to. Molecules that can't pass through the membrane, and by that I mean dissolve through the phospholipid bilayer by simple diffusion, is anything that is polar or water soluble, so it has a charge, because that will be repelled by those phospholipids. So things like sodium ions. Or any molecule that is too large to fit through the gaps, so glucose for example. Now you do have other components in the cell membrane, making that mosaic feature of the fluid mosaic model. So cholesterol is another type of lipid, and that's present in some membranes in varying amounts. And cholesterol restricts the lateral movement, meaning the sideways movement of other molecules in the membrane. So that's going to be useful as it makes the membrane less fluid at high temperatures, and we want that so that it can prevent water and dissolved ions from leaking out. You also have proteins in your membrane, and proteins can be embedded across the cell surface membrane, or they could be just on the outside. So if it's embedded across, it's known as integral. If it's just on the outside, it's known as peripheral. Sometimes that's intrinsic and extrinsic. So the peripheral proteins, those can provide mechanical support, or they can connect to carbohydrates to make glycoproteins, and carbohydrates can bind to the lipids to make glycolipids. And those are normally, the function is for cell recognition as receptors. The integral proteins are going to be carrier proteins and channel proteins, which are involved in active transport, co-transport, and facilitated diffusion, which is coming up shortly. Protein channels, which we can see one here, these form tubes. We can see like a hollow-like tube, and that fills with water. That will enable any of our polar substances, so they're going to be water soluble, to dissolve in that water, and then they can move through the membrane in that way because they can't dissolve in the phospholipid bilayer. Whereas carrier proteins, these will have a molecule bind with them, such as glucose or amino acids, and when that molecule binds to the carrier protein, it causes it to change shape, and that then releases it on the other side of the membrane. Which leads us into the 5 key ways that molecules can transport across these plasma membranes. We have the top 3, which don't require energy: simple diffusion, facilitated diffusion, and osmosis. And the bottom 2 are both active types of transport, meaning they require energy from ATP from respiration. Active transport and co-transport. So let's go through all 5 of these. Now, one thing that they'll all have in common is there are certain adaptations to increase the rate at which those types of transport happen. So you could be asked to identify adaptations that cells have for rapid transport. And the sorts of things you need to look for are features that increase the surface area. So for example, microvilli, which is highly folded membranes, increases the surface area. Or if it's a type of transport that goes through a protein channel or a carrier protein, then there might be more of those proteins embedded in the membrane. So again, you've got a larger surface area for that to happen. If it's a cell where there'll be a lot of active transport or co-transport, because those require ATP from respiration, you'll also have many mitochondria as an adaptation as well. So let's have a look at simple diffusion. This is the net movement of molecules from an area of higher concentration to an area of lower concentration. So it's going down the concentration gradient. And that will occur until equilibrium is reached, meaning same concentration in both sides. This process doesn't require any additional energy from ATP. The molecules do still have energy because they're moving, but this is due to kinetic energy that they possess, and that's what enables them to constantly move in fluids, whether that's gas or liquid. For small molecules to diffuse across a membrane, they must be lipid soluble and small. So that is the criteria for simple diffusion across a membrane. It has to be lipid soluble, so that would be a non-polar molecule, meaning it doesn't have a charge, or a small molecule. Facilitated diffusion is still going down the concentration gradient. So it's still a passive process. No extra energy is added in the form of ATP. It's going down the concentration gradient, but it's for any molecule that doesn't fit the criteria we said here, lipid soluble and small. So it's going to be for molecules that are polar. So that means they have a charge. They are not lipid soluble. They're water soluble, and they are too big, or they might be too big. So this is going to happen through either protein channels or protein carriers. It can be through either of those types of membrane proteins for transport. If it's a protein channel, this is the ones where we have the tube-like center filled with water. That enables the ions or polar molecules, water-soluble molecules, to dissolve and then pass through the membrane through that tube filled with water, that protein channel. It is still selective though, as the channel proteins will only open in the presence of certain ions when they bind to the protein. So it is still selective transport across the membrane. Facilitated diffusion can also happen with carrier proteins, and in this case the molecule will bind to the carrier protein, such as it could be glucose, and that causes a change in shape to that carrier protein, and that change in shape causes it to release the molecule to the other side of the membrane. Osmosis, so this is still passive, meaning no extra ATP or energy is needed, and it is going down a concentration gradient. But we need to use the terminology water potential, not concentration gradient, for osmosis to get the marks. So for this, water potential is the pressure created by water molecules, and it's measured in kilopascals and represented by this symbol here. This means water pressure. Pure water has a water potential of 0. So 0 is the highest water potential you can ever get, and that's for pure distilled water. As soon as you dissolve any solutes in it, you get a negative water potential. And the more negative a water potential is, that indicates more solutes are dissolved in it. So that's what we mean by water potential. And osmosis is the net movement of water from an area of higher water potential, even though it will still be negative unless it's pure water, but it's a higher water potential to a more negative or a lower water potential, and that's the direction that the water will move, and it's across a partially permeable membrane. So water is the thing that moves, not the solute, and it's always moving towards a more negative water potential, which means more solutes are dissolved in the water at that location. Now, you could get questions linked to this, or mainly actually links to the required practical to do with plant tissue, but the terms isotonic, hypotonic, and hypertonic. Isotonic means the water potential of a solution is the same as the water potential in a cell. And because there's no difference in the water potential, you don't have any osmosis happening, or there's no net movement of water, so the cell will stay exactly the same size and shape. A hypotonic solution is when the water potential of the solution is more positive, meaning closer to 0 than the cell. And that means there's more water in the solution than the cell. So water is going to move into the cell by osmosis. And in animal cells, that can cause it to swell and even burst. Hypertonic solutions is when the water potential of the solution is more negative than the cell, and that will cause water to leave the cell by osmosis, and the cell will then shrivel up. Next, then we move on to our active types of transport. So we have active transport first, and this is the movement of a substance from a low concentration to a high concentration. So it's going against the concentration gradient. To do this, it has to use metabolic energy, so that means ATP from respiration, and also it always goes through a carrier protein. So you have to state it's a carrier protein for active transport. So the way that this happens then is, step number 1 is the transport is going through, or transport is occurring with a carrier protein, and those are ones that are going to span the entire cell membrane. A molecule will bind to a receptor on that protein, complementary in shape. ATP will then also bind to the carrier protein from the inside of the cell. So we can see here in the cytoplasm on the inside, it then binds, and it's hydrolyzed into ADP and PI. That releases energy, and that is what causes the carrier protein to change shape and release the molecule to the other side of the membrane. This inorganic phosphate ion was attached the whole time after ATP has been hydrolyzed, and that then gets released, and that causes the protein to return to its original shape. So in order for active transport to continue to occur, you need a constant supply of ATP from respiration. Next then is co-transport, and this is a type of active transport because one of the steps in the process is active transport. So to absorb glucose from the ileum lumen in the gut, which we'll see in topic 3 in digestion and absorption, because there's a higher concentration of glucose in the cell than in the ileum, it's hard to get the glucose to move in because it's going against a concentration gradient. So that's why we need a type of active transport, which is co-transport. So the first thing that happens is actually here. Sodium ions from within a cell are actively transported out of the cell and into the blood in capillaries. That then reduces the concentration of sodium ions in the cell. And as a result, we have now created a concentration gradient from the ileum to the cell. So sodium ions are able to move from the ileum into the cell down a concentration gradient. But they are an ion, they're sodium ions, so they have to do this through a protein. And the protein that they do this through is a co-transporter protein, which means it transports 2 molecules at the same time. And it transports the sodium ions plus glucose, or it could be amino acid as well. That then gets the glucose into the cell. And then we have a high concentration of glucose in the cell compared to in the blood. So glucose can now move down its concentration gradient from the epithelial cell into the blood and the capillary by facilitated diffusion through a protein. And it has to be facilitated because glucose is too large to move by simple diffusion. Next then is 3.2.3, cell recognition and the immune system. So the first part is all about cell recognition and identifying what are self cells, meaning part of your body, and non-self cells, which are not part of your body. And your immune system has cells to identify the presence of pathogens, which are non-self cells, and other non-self cells, which are potentially harmful. And then your immune system will either destroy those or neutralize them to prevent harm. So those cells are the lymphocytes, and each type of cell has a specific molecule on its surface to identify it. And those molecules are usually proteins, and because they're proteins, they have a unique 3D shape. There's overlap here with biological molecules from topic 1 because common marking points linked to this are stating that it has a tertiary structure because it's a protein and that's what gives a unique 3D shape, but tertiary structure would be the key marking point, and that would be complementary in shape to, and that's actually going to link to some of the cells that we learn about later, but it's the idea of tertiary structure, complementary in shape. That's how you can work out if it's a self or non-self cell, and a bit more on that when we get to the T and B cells. But thinking now more about what counts as a non-self cell. So if a non-self cell is detected, a response will be triggered to destroy that cell. And these are the 4 examples that you need to know that could trigger an immune response because they're detected or could be detected as non-self cells. So number 1 is a pathogen. So bacteria, fungi, they'll have antigens on their surface that can be used to identify them as non-self cells. It could be cells from another organism of the same species. So for example, if you've had an organ transplant, that could be detected as non-self. And we don't want that to happen because then your immune system will start to attack and destroy the cells from that transplanted organ. It could be abnormal body cells. So for example, cancer cells, their antigens on the surface will slightly change. They now don't get detected as self cells, they're now detected as non-self. Or it could be toxins because some pathogens release toxins into the blood, such as cholera. So I use this word antigen quite a bit, but an antigen is a foreign protein that generates an immune response by the lymphocytes when it's detected, and it's located on the surface of cells. That is quite a common 2-mark question. You need both of those points to define what an antigen is. Antigens can change, though, over time, and this links to antigen variability. So a pathogen's DNA can mutate frequently, and if the mutation occurs in the gene that codes for the shape of the antigen, then that shape of the antigen will change. And therefore, any previous immunity that you might have had for that pathogen, based on the recognition of the old shaped antigen, it will no longer be effective because the memory cells have a memory of the old antigen shape. So this is known as antigen variability. And for example, the influenza virus, which is the virus that results in the flu, that mutates and changes its antigens very quickly. And that's why you need a new flu vaccine every single year. So let's go on to the immune response then. If you do have this non-self cell detected, we said an immune response occurs. So if a pathogen gets past any chemical or physical barriers, which you don't learn for AQA for A level, but that's things like your skin barrier, your stomach acid, and it can then enter the blood, then this is when the white blood cells are the second line of defense, and you either get a specific response by the lymphocytes or a non-specific response. And in fact, you get both, starting with the non-specific response of the phagocytes and then the lymphocytes. So the non-specific response by the phagocytes is phagocytosis. And a phagocyte is a type of white blood cell. For example, a macrophage is an example of a phagocyte. And phagocytes are white blood cells that can do phagocytosis. They're found in the blood, but also in the tissue. And phagocytosis is a non-specific immune response. What that means is any non-self cell that is detected, and that means any of those 4 items that we listed a couple of slides ago, if they are detected, it will trigger phagocytosis to happen every single time. So phagocytosis then, the first thing that happens is our phagocyte, for example a macrophage, which is in the blood or in the tissues, and any chemicals or debris that are released by the pathogen will attract the phagocyte to move towards the pathogen. So that's how the phagocyte is able to detect where the pathogen is. Now on the phagocyte, there are many receptors which are binding points on the surface of that phagocyte that can then bind to the antigens or any chemicals on the pathogen. The phagocyte, once it's then bound to it, the phagocyte changes shape, which is known as engulfing. So it completely surrounds and engulfs the pathogen, and it then will put it inside of a vesicle inside of the cell. And that vesicle is known as a phagosome. So we can see here the pathogen's been engulfed and it's now trapped inside of the phagosome. You then have the lysosomes within the phagocyte will move towards, so the lysosome shown here in blue, it will move towards and fuse with the phagosome, and it will then release its contents. And the contents of a lysosome is digestive enzymes. And in this case, they're known as lysozyme enzymes. They're digestive, which means they're lytic, or they're going to split, they're going to break apart, hydrolyze the pathogen. So that is how the pathogen is then destroyed. From the broken down, hydrolyzed, digested pathogen, any soluble products that are going to be useful can be absorbed by the phagocytes, and then the antigen is placed on the surface of the pathogen, and that is going to be used later on as a way to signal to other white blood cells known as lymphocytes that a pathogen has been detected and destroyed. So that takes us on to the first type of lymphocyte we're going to go through, T lymphocytes, or T cells for short. These lymphocytes are involved in the specific immune response, meaning they respond in a particular way based on the particular shaped antigens that are detected. Lymphocytes are made in the bone marrow, and this is extra information, you don't need to know it, but they're called T lymphocytes because T cells then go to mature in the thymus, which is this structure here in the body. And they are involved in the cell-mediated response. So this is where we look at how the antigen-presenting cells are going to help trigger the cell-mediated response. And the antigen-presenting cells could be, like we just said, a phagocyte, for example, a macrophage, which has engulfed and destroyed a pathogen and then it presents it on its surface. But it's actually any cell that has a non-self antigen on its surface. So that could be one of your own body cells that's become infected. It could be one of those phagocytes presenting the antigen on the surface. It could be cells of a transplanted organ. It could be cancer cells. And because they have antigens presented on their surface, that is going to enable lymphocytes to respond to those antigens in this cell-mediated response. So let's carry on from this bit we were at. So once the pathogen has been engulfed and destroyed by the phagocyte, antigens are then presented on the cell surface. It's now called an antigen presenting cell. And we can see an example of that here. We've got an antigen presenting cell. One type of T lymphocyte is known as a helper T cell. And helper T cells, they have these receptors on their surface, and that can attach to complementary shaped antigens on antigen presenting cells. Once it then attaches, that activates the helper T cells to divide by mitosis, and that will then replicate and make large numbers of clones of that exact helper T cell that had the receptor complementary in shape to that exact antigen. That stage is known as clonal expansion. Clonal because you're making identical copies, expansion because you're making large numbers. And this is where there's that link back to mitosis. We said the role of mitosis is growth and repair. In this case, it's growth and making large numbers of identical T helper cells. Now we've got that large number of identical T helper cells, they actually differentiate into different types of T lymphocytes. So some will actually just remain as helper T cells because those are then going to go on to activate the B lymphocytes, which we're going to go on to shortly. Some of them are able to stimulate macrophages so that they will perform more phagocytosis and engulf and destroy more of those non-self cells. And then some of them become cytotoxic T cells, sometimes called killer T cells. AQA use the term cytotoxic T cells. And a cytotoxic T cell is able to destroy your own body cells that are infected with a pathogen, typically a virus. And the way that they do this is cytotoxic T cells release a protein called perforin, and that protein can embed in the cell surface membrane to make a hole or a pore. And that means that either lots of water will move in and cause the cell to burst, destroying the infected body cell, or it will cause lots of water to move out, causing the cell to shrivel, and again, it will cause cell death. So this is most common in viral infections because viruses can only replicate when they're inside of the body cell. So if you destroy the body cell, the virus can't replicate, and that gives more time then for the phagocytes to locate and destroy the virus before it can replicate and cause more damage. And this is why you get a sore throat when you have a cold. The cytotoxic T cells are destroying the infected body cells in your throat. You're sacrificing body cells to prevent the virus replicating and infecting even more cells and tissues. Now, B lymphocytes, this is the humoral response. Again, all lymphocytes are made in the bone marrow. You don't need to know this, but the B is because it matures in the bone marrow as well. This is a humoral response, meaning within liquids or within body fluids, because in this instance, the B cells will only respond to antigens that are floating within the blood or within body fluids. They don't respond to the antigen presenting cells in the same way as a cell mediated response with the T cells. So let's have a look at the B cell activation. So you have approximately 10 million different B cells which have antibodies on their surface complementary in shape to 10 million different antigens. And you have these circulating in your blood, and if you get an antigen in your blood, collide with the complementary shaped antibody on a B cell, that is going to cause the B cell to take in that antigen by endocytosis, which means basically surround and engulf it. So this is responding to the antigens in the blood, not the entire cell. When the B cell then collides with the helper T cells that we talked about in the previous slide, that activates the B cell to go through clonal expansion and differentiation. Now this can happen because when that B cell takes in the antigen, it's going to break it down, but it can then actually also present it on its surface. So that means it's able to bind to helper T cells that have that receptor complementary in shape to the antigen. That is then going to trigger the B cell to be activated to do the same thing as the T cells. It undergoes mitosis, you get a large number of clones. clones of that B cell that has that exact shape, antigen and antibody on its surface, complementary to that foreign protein, the antigen. But this time, the cells differentiate into different molecules. They can either become plasma cells or memory B cells. The plasma cells, which you can see here, these are the ones that make antibodies. Memory B cells are the ones that remain in your blood for long periods of time, and they can divide rapidly into the plasma cells to release large quantities of antibodies so quickly if you get reinfected with the same pathogen. So they're responsible for you being immune, basically, to particular pathogens and disease. So let's have a look at this. Memory B cells, as I said, they can live in your blood for a long time. They can be decades in your blood, whereas plasma cells are very short-lived. And those are the ones that make the antibodies, so that's why we need to store a memory of that antibody. So if you are reinfected, you can then have the memory B cell rapidly differentiate into plasma cells, which can then make lots of antibody so quickly that it should help to destroy the pathogen before you get any symptoms, which means you are immune. Or it might just mean you get very, very minimal symptoms. And this is known as active immunity. You are immune to that pathogen, and it's active because you have memory B cells against that pathogen. So this links this concept of primary and secondary response. Primary immune response is the first time you are exposed to a pathogen. And because it's the first time you're exposed, it can take a longer period of time for the B cell to collide with that antigen, causing it to engulf the antigen, and then you get the clonal expansion, differentiation. All of that process of the humoral response has to happen. So it takes a longer time to make antibodies, and you don't get as high a concentration. The second time you're exposed to that same antigen, because you now have the memory B cells present in your blood, those can then collide with the pathogen's antigen again and differentiate rapidly into plasma cells. So you end up with large quantities of antibody much, much quicker. And this is why the secondary response should mean that you get no symptoms or very few, because you get the antibodies being produced so rapidly and in large concentrations. So let's have a look at what antibody is. They are proteins that have a quaternary structure, meaning more than one polypeptide chain, and they happen to have 4 polypeptide chains. The end bits here are the variable regions, meaning those are the parts that are going to be different in shape and combined to the different shaped antigens. The red part is constant, meaning it's the same in all antibodies. The heavy chain is this longer one, and you have 2 heavy chains. The shorter chain is known as the light chain. Here are the antigen binding sites. You can bind one here and here. And then you also have these bonds here and the hinged section, which makes the antibody flexible, so it can easily bend and attach to one antigen over here, bend and attach to another over there, which is how we get agglutination happening. And agglutination is the idea that multiple antibodies combine together lots of different antigens at the same time, so they all clump together. That makes it easier for the phagocytes to, first of all, locate where all the pathogens are, and then they can more efficiently engulf and destroy them. So antibodies, they create these antigen-antibody complexes, and we get agglutination happening. But they don't directly kill the pathogen. They just make it far more efficient for the phagocytes to then destroy the pathogen. So different types of immunity. First of all, passive immunity. Passive immunity is when the antibodies are introduced into the body and the pathogen doesn't enter itself. So you don't actually get any antigen or pathogen in you. So that means the plasma cells and memory cells are not made. You just get a direct source of antibody. So you don't get any long-term immunity because there's no memory B cells. A natural example of this is in breast milk. So through breastfeeding, breast milk contains antibodies. That means babies will have passive immunity whilst they are breastfeeding. And when I say whilst, it's not literally just while they're breastfeeding. The antibodies can last for a short period of time. Active immunity is when you have been exposed to the pathogen or the antigen, and therefore you do have memory B cells in the body. And that could be natural active immunity following infection, or it could be artificial active immunity following a vaccination where you might have a weakened version of the pathogen or the antigens injected in. So vaccines, that means a small amount of either weakened or dead pathogen or the antigens itself introduced to the body. And that could be as a liquid through the nose or mouth, or it could be an injection. And exposure to those antigens activates the B cells to go through clonal expansion and differentiation, just as we saw in the humoral response. So those B cells will then undergo mitosis, make large numbers of identical cells, which will then differentiate into plasma cells and memory B cells. Plasma cells will make antibodies, which will then destroy any of that weakened or dead form of the pathogen. And the B memory cells will then remain in your body for decades. So if you are reinfected with the same pathogen, you can make large numbers of the antibodies so rapidly that you shouldn't get symptoms. Herd immunity is if enough of the population are vaccinated against a particular pathogen. That means that the pathogen cannot spread or transmit amongst the population. And that's because if you have most of the population immunized, that means that they can't transmit the pathogen onto someone else, and therefore hardly anyone in the population is going to have the pathogen. And this provides protection for those who are not vaccinated. So maybe they are too ill to be able to be vaccinated, or maybe they're pregnant, or they have some lowered immunity that means they aren't able to. The next bit is then looking at HIV, an example of a virus, and it is a retrovirus. It has a lipid envelope around it, but that is taken from the host cell membrane. The envelope has the attachment proteins, which are glycoproteins, which is how it attaches to the host cell. So here are the key structures that you have in the HIV particle. You have the core, and that is the genetic material, which is RNA, and it has the enzyme reverse transcriptase as well, which is needed for the virus to replicate. You have the capsid, which is the outer protein coat protecting the genetic material. There is the envelope, that is an extra outer layer, but that is made out of lipids taken from the host's cell membrane. And then you've got the protein attachments on the exterior of the envelope to enable the virus to attach to the host's helper T cell. So let's have a look at how HIV replicates in helper T cells, because those are the host cells that it infects. HIV is transported around in the blood until it attaches to a CD4 protein on a helper T cell. So here is a helper T cell. Here is a CD4 protein, which is a receptor. And we can see that the HIV with the attachment proteins binds to that receptor. The HIV protein then fuses with the helper T cell membrane, and that enables the RNA and the reverse transcriptase enzyme to enter with the capsid, and then it does get released from the capsid. The HIV enzyme reverse transcriptase copies the viral RNA into a DNA copy and moves into the helper T cell nucleus, and this is why it's called a retrovirus, because it's doing the reverse of transcription. You've got an RNA copy being turned back into DNA. The DNA then moves into the nucleus. It combines with the host's DNA, and that is where the mRNA is then transcribed along with the host cell's mRNA to create the viral proteins. The viral proteins are then reassembled, and they'll then move towards the cell surface membrane, and they are released from the cell. And that then goes on to make a new virus particle. Now, the difference between HIV and AIDS is HIV is the virus, and it specifically targets and it destroys helper T cells, because as it replicates inside of them, it destroys the helper T cells, which activates B cells. So if we don't have those helper T cells, they're not there to activate B cells. That means we have now significantly reduced the immune system. And if enough helper T cells are destroyed, then you end up with AIDS, acquired immunodeficiency syndrome, where you've become so vulnerable to opportunistic infections and cancers and you can't defend yourself. So symptoms of AIDS are chronic infections, weight loss, fever, night sweats, and they can be followed by more serious illnesses like pneumonia, tuberculosis, and certain cancers. A person is HIV positive if they're infected with HIV. AIDS is when the replicating virus in the helper T cells interfere with the normal function of the immune system, and it's normally classed by how many of the helper T cells have been destroyed. The last bit of this topic is monoclonal antibodies. Mono meaning one, clonal meaning identical. And a monoclonal antibody is a single type of antibody that can be isolated and cloned. And antibodies are proteins which have binding sites complementary in shape to certain antigens. Now this is what is created in the immune response, monoclonal antibodies, lots of identical copies of the same antibody. But this has been manipulated to create monoclonal antibodies for medical treatments, medical diagnosis, and even pregnancy tests and other chemical tests. Now, targeted medication, this can use direct monoclonal antibody therapy, and this is when you have some cancers that can be treated using monoclonal antibodies, which are designed with a binding site complementary in shape to the antigens on the outside of cancer cells. Those antibodies are then given to the cancer patients and they'll attach to the cancer cells. While the antibodies are bound to the cancer cell antigens, this prevents chemicals from binding to the cancer cells, which enable the uncontrolled cell division. And therefore, the monoclonal antibody prevents the cancer cells growing anymore. And as they're designed to only attach to the cancer cells, they should only affect the cancer cells and not healthy body cells. Indirect monoclonal antibody therapy can also be used, and this is when you would attach a drug to the monoclonal antibody. So in this instance, you've still created the antibody to be complementary in shape to the antigens on the cancer cells, but the cancer drugs are then delivered directly to the cancer cell, and it kills the cancer cell rather than lots of healthy body cells. So this should reduce the harmful side effects that affect traditional chemotherapy and radiotherapy can produce. And this is known as a bullet drug. It's going straight to where it's needed. So if you have a look at medical diagnosis, monoclonal antibodies can be used to test for things like pregnancy, influenza virus, hepatitis, chlamydia, prostate cancer, but also it's not on the spec because this happened after the spec was created, but COVID-19, all of those lateral flow tests that we used to have to do all the time use monoclonal antibodies. And this works via an ELISA test. So an ELISA test, the enzyme linked immunoabsorbent assay, it uses two antibodies. We can see here in the pregnancy test, we've got, first of all, a mobile antibody, and then we have a immobilized antibody. And we actually have a third one here. We've got an immobilized one as well. And in this instance, what happens is you would dip your pregnancy test into the urine that you are testing to see if someone is pregnant or not. Because when you are pregnant, you create this hormone HCG and it can be secreted in your urine. So this is absorbent paper. That means the urine with any hormones dissolved within it are going to move up through that absorbent paper. The hormone is complementary to the mobile antibody, which also has a coloured dye attached to it. So that will then move up the pregnancy test. And at point C, you have immobilised, meaning it can't move, antibody that is also complementary in shape to the hormone. So that will then cause the first antibody that has the hormone attached to it to attach to the immobilised one. And it also has that coloured dye on it. And that's why at that exact position you get a coloured line. Any antibody that didn't bind to the hormone will move past section C, but we do have antibodies also immobilised at point D that are complementary in shape to the end of that antigen. So those will bind and you get a second line. And in this way, whether you have that hormone or not, you should always get the line at D, and that is to help prevent false negatives, because it shows us that you don't have a coloured line here if you don't, but the antibodies are definitely able to move, so the test is working. Whereas if you have a line here, that means you are pregnant because you do have that hormone present. And this line here confirms that the test was working, so antibodies did move through the test. Now you can also have different versions of this. So we've got one here where we would add the test sample from a patient to the base of a beaker or the base of a well. And we'd then wash to remove any unbound test sample. Then you'd add an antibody complementary in shape to the antigen you are testing for, and that will bind if it's present. Again, wash to remove any that didn't bind. We then add a second antibody that is complementary in shape to the first one, so that's only going to bind if the antigen was present and therefore the first one bound. Again, wash to remove any that's unbound. Then you'd fill the wells with a liquid which is a substrate for this enzyme. And when those react together, you get a colour change. So the presence of the colour indicates that you do have that antigen present, and the intensity of the colour indicates the quantity present. Now, there are some ethical issues around monoclonal antibodies because creating monoclonal antibodies requires animals such as mice to produce those antibodies in tumour cells, which leads to ethical debates as to whether this use of animals is justified to enable better treatment of cancers in humans and detect disease. Because in order to do this, there will be suffering to the animal, and then at the end they would be killed because they wouldn't be needed. So topic 3 for AQA A-level biology, and we start with 3.3.1 surface area to volume ratio. Exchange surfaces in organisms, so where you might have diffusion occurring across, for example, have many similar adaptations to make that transport across the surface more efficient. And surface area to volume ratio is one of those adaptations. And the relationship between the size of an organism or the structure, so like the shape of a cell or the organism, and its surface area to volume ratio plays a significant role in the types of adaptations an organism will have. And we're going to see this repeatedly throughout topic 3. So the concept of surface area to volume ratio is if you were to work out the surface area of the object, and in the case of a cube, that would be the area of one side of the cube, so the length times the width, and then we times that by 6 because there's 6 sides to the cube. So our surface area for this cube is 6 centimeters squared. The volume would be the height times the length times the width, and 1 times 1 times 1 is 1 centimeters cubed. The surface area to volume ratio means divide the surface area by the volume. And that gives us 6, or a 6:1 ratio, but we can just present that as 6. If we then were to calculate that for these larger cubes, we can see this one, the surface area to volume ratio would be 3, and this one would be 2. And this is demonstrating a key pattern. The larger the object or the organism, so the bigger the size, the smaller its surface area is compared to its volume. And that means that small organisms, for example an amoeba, has a very large surface area compared to its volume. And this means that there is a big enough surface for exchange to happen across to compare to its volume. But also there's a smaller distance from the outside to the very middle of the organism. So you have a short diffusion distance as well. Now, as a result of this, very small organisms don't have special exchange systems that they've evolved. For example, a lung system or respiratory system, we should call that. The lungs is the organ. And therefore, they can just simply exchange substances across their surface. So as an organism increases in size, their surface area to volume ratio gets smaller. So that means larger organisms have a relatively small surface area compared to its volume. So they don't have a very big surface area available for exchange of substances like oxygen or heat compared to their volume. Whereas in the smaller organisms, as we said, they have a large surface area to volume ratio, and that enables efficient exchange directly across their body, so they don't have to have any specialized exchange or transport systems. Now, smaller mammals have a high surface area to volume ratio, and that means that they lose heat rapidly, across their surface. So to replace that heat that they lose, they have to have a high rate of metabolism, and therefore they will respire rapidly. And this high metabolic rate provides the energy needed to provide more heat and maintain a stable body temperature, especially if they're in colder environments. Larger animals, because they have a smaller surface area to volume ratio, they will lose heat more slowly due to that. And their metabolic rate can therefore be relatively lower in comparison to the smaller mammals. So larger organisms then will have adaptations to increase the surface area to volume ratio at these exchange surfaces. So for example, villi and microvilli, which we'll see come up in the digestion topic in this video. Alveoli and bronchioles, spiracles and tracheoles, and gill filaments and lamellae, and also the thin wide leaves are all linked to gas exchange. This one here is in mammals, this one is in terrestrial insects, fish and plants, and this is all going to come up in this video. And then also many capillaries, so having a capillary network is going to be relevant at exchange systems for gas exchange, but also for the absorption of digested food. So these concepts will come up over and over again in this video. 3.3.2 is gas exchange, and some key terms to be familiar with this topic are breathing. This is the movement of air into and out of the lungs. Ventilation is the scientific term for breathing. Respiration doesn't really come up in this topic too much, but I just bring it up because that's commonly mistaken for breathing at GCSE. So let's just clarify. Breathing and respiration are not the same thing. Breathing is air moving in and out. Respiration is the chemical reaction that releases energy in the form of ATP. And then gaseous exchange, that is diffusion of oxygen from the air in the alveoli into the blood, and of carbon dioxide from the blood into the air in the alveoli. Now, gas exchange just means the exchanging of gases across a surface, and that will be by diffusion. So gas exchange surfaces all have the same three features that they'll have adaptations for. Large surface area to volume ratio, short diffusion distance, maintaining a concentration gradient. And when we have a look at the gas exchange system in mammals, for example, humans, in terrestrial insects, in fish, and in leaves, we'll look at what do they have as adaptations to meet these needs. And that's the common marking points that come up for all three. And it links to this equation here, which you don't have to know, but it just helps to explain it. The rate of diffusion is proportional to how big the surface area is, how steep the concentration gradient is, divided by the length of the diffusion pathway. So if we have a look at the human gas exchange system, these are the 5 structures that you need to be familiar with. So the alveoli, which are the air sacs. We then have the bronchioles that attach to those, and they're branching. You then have the bronchi, which is the 2 splitting tubes from the trachea, which is your windpipe. And the lungs, you have a left and a right lung. Ventilation is how we get air in and out of the lungs. And you need to know about the role and what happens to the diaphragm, the rib cage, and the intercostal muscles. So ventilation is inhaling and exhaling, and it is controlled by these muscles. And when it comes to the intercostal muscles, you have to be specific, talking about the external or internal. It's normally only the external that they refer to on the mark scheme. So let's have a look at these two. For inspiration, which is inhaling, the external intercostal muscles contract, and that pulls the rib cage up and out. That would be 1 mark. I've not seen on the mark scheme anything linked to the internal intercostal muscles, but just so you're aware, they'll be relaxed because they're antagonistic muscles, which means if one is contracting, the other does the opposite, so relax. The diaphragm is a muscle, so it contracts, and that then pulls it to move down and flatten. That would be a mark on the mark scheme. This then causes the air pressure in the lungs, or you could say the thoracic cavity, to decrease. And the reason the air pressure is decreased is because the lung volume is increased, or again, you could call that the thoracic cavity. Because that rib cage has moved up and out, the diaphragm has moved down, you now have a bigger space, so a larger volume, but it contains the same number of air particles. So that means that the pressure decreases. And for that reason, air moves in. So your key marking points would be this row here about the external intercostal muscles, this row about the diaphragm, and then these two combined. So that would be a 3-mark answer. Expiration or exhaling, exact opposite. So the external intercostal muscles relax, and that causes the rib cage to move down and in. The internal intercostal muscles, again, it doesn't really come up in the mark scheme that you'd have to say that they contract. So the pull the rib down and in, that would link to this mark here. The diaphragm relaxes and that causes it to dome and move upwards. And because of that, you've now decreased the volume in the lungs or the thoracic cavity, and therefore that increases the pressure in the thoracic cavity. And because you've got a higher pressure, that is moving air out. Now, there is a maths equation linked to this, pulmonary ventilation, and that is the total volume of air that is moved into the lungs during 1 minute. So decimeter cubed is our unit for volume, per minute is how we get it as a rate. So you'd need to do the tidal volume times ventilation rate. And the tidal volume is the volume of air inhaled or exhaled in one breath or breathing cycle. Ventilation rate is the number of breaths per minute. So if we have a look at some adaptations then to increase the rate of gas exchange or diffusion of the gases, once the gases are in the alveoli, so because of that ventilation, the gas exchange occurs between the epithelium and the blood. Alveoli are the tiny air sacs at the end of the bronchioles, and in each of your lungs you have about 300 million, and that is how we get the large surface area. One alveoli does not provide a large surface area, it's the fact that there are so many in each lung. That's adaptation number 1. The short diffusion distance, that is because the alveoli epithelium is just a single layer of cells. That's a very thin layer to minimize the diffusion distance. It's the same thing with the capillaries as well. It's just one layer of cells making up that epithelium. So that is our short diffusion distance. The maintaining the concentration gradient is nearly always the same adaptation, surrounded by a network of capillaries. Because what that means is, as the gas diffuses into the blood, it's then carried away and replaced by blood that is low in that gas, in this case oxygen. So those are our key adaptations for gas exchange in the human lungs. We then move on to gas exchange in fish, and fish get their oxygen from the water. The challenge they face though is there's about 30 times less oxygen dissolved in water than there is present in air. So they have to have special adaptations to really ensure that they maintain that concentration gradient even when there's so much less oxygen dissolved in water. And we'll come to that when we get to the countercurrent flow. So fish gill anatomy first then. So the gills are found on both sides of the head, and they're found as 4 layers of gills. The gills are made up of these stacks of gill filaments. So this long structure sticking out is a gill filament. Each gill filament, so we're zooming in on one gill filament here, has lamellae at right angles going all the way along it. And the lamellae is the site of gas exchange. So because you have gills on both sides of the head, you then have lots of gill filaments, and every gill filament is covered in lots of gill lamellae. That is what provides the large surface area for gas exchange. And when the fish opens its mouth, water rushes in and it goes through. There's a hole in the side of the head, which you can see here. So that causes the water to move in, goes over the gills and comes out the other side. And as the water flows over the gills, the gas exchange will happen at the gill lamellae. So other adaptations then. We've talked about how they have a large surface area to volume ratio. Next then is, how is the short diffusion distance maintained or provided? And that is, first of all, because the gill lamellae are very thin, but also within every gill lamellae there's a capillary network, and the gases are diffusing from the water into the blood in the capillaries, and that's a really short diffusion distance. Finally, then, it's how do they maintain that concentration and diffusion gradient across the entire lamellae, even though there's a low amount of oxygen in water. And that's when we get to the countercurrent flow mechanism. And the countercurrent flow mechanism means that the blood and the water flow in opposite directions. So this is when water flows over the gills in the opposite direction to the flow of the blood in the capillaries. That would be one of your key marking points for what is the countercurrent flow mechanism and how is it advantageous. That means that the equilibrium of oxygen is never reached, because as the blood flows in one direction and the water flows in the other, that means you should always have blood which has a lower concentration of oxygen reaching water which has a higher concentration of oxygen. So equilibrium is not reached. And your key marking point, your third mark, would be this ensures that a diffusion gradient is maintained across the entire length of the lamellae. Because if it was concurrent flow, that should be con, not co, that would mean that the blood and water flow in the same direction, and by the time you're about halfway or 3/4 of the way across the lamellae, you would reach equilibrium, so there'd be no gas exchange or diffusion in the end section. And that's why the key mark is across the entire length of the gill lamellae. Next, then, we look at terrestrial insects. They have an exoskeleton made of hard, fibrous material, and that is an advantage because it's protection and it prevents water loss, but it reduces the space that they could do gas exchange across. So they don't have lungs, and instead they have a tracheal system. And the tracheal system is made up of a trachea, tracheoles, and spiracles. Whenever you're describing this in an exam question, there's normally 1 mark just for stating those 3 structures. So always start by stating those. The spiracles are these round valve-like openings that run along the length of the abdomen, and they can open and close like stomata in plants. And that is where the gases can enter and leave. The trachea is a network of internal tubes. So once the gases enter through the spiracles, they then go through the trachea, and they do have rings within them to keep them open the whole time. And then the trachea branch into tracheoles, and these are much, much smaller tubes that are going to reach the abdomen of the insect. And they extend throughout all of the tissue in the insect to ensure that oxygen is delivered to all respiring cells. So those are the 3 key structures. Now, in terms of the adaptations, we need to think about large surface area, short diffusion distance, maintaining the concentration gradient. So first of all, tracheoles, which are the branching structure where gas exchange actually happens, have very thin walls. So that provides a short diffusion distance. Tracheoles are also highly branched, which provides a short diffusion distance because they're reaching so many cells that there's never going to be a big distance between the tracheoles and these cells. And also because the tracheoles are highly branched, that provides a large surface area for gas exchange. So these are our adaptations for short diffusion distance and large surface area. Linking to the maintaining of a concentration gradient, at rest, so when the insect isn't flying, diffusion is the main method of gas exchange happening at those tracheoles. And when the cells are respiring, they're using up oxygen and producing carbon dioxide. And that is enough to create a steep concentration gradient from the atmosphere compared to the tracheoles for the insect. So that is how the concentration gradient is maintained, is through respiration. And that's when they're at rest. During periods of high activity, so for example when they're flying, there will be mass transport of the gases occurring. So what happens in this case is the insect's abdomen will contract and relax, which actively moves air in and out of the tracheal system, and that helps to maintain the diffusion gradient. Finally, when the insect is very active, so they might be flying, they will end up respiring anaerobically. And lactate or lactic acid is produced during anaerobic respiration. And when that dissolves in the cells, it's going to lower the water potential of those muscle cells. And this causes water, which is naturally present in the very tips of the tracheoles, to move into the cells from the tracheoles by osmosis. And because water has now left the tracheoles and gone into the cells, there is now an empty space to be filled with air in the tracheoles. And that is going to cause gases to move more quickly into the tracheoles. Now, when it comes to reducing water loss, in the spec, it says you need to know how terrestrial insects reduce water loss and plants, and you need to know comparisons between the two. So let's have a look at terrestrial insects first of all. Insects have that waterproof exoskeleton, and that prevents evaporation of water across their surface. The spiracles that they have can open and close, and when they close, that prevents water from evaporating out. So that reduces water loss. And the spiracles also have tiny, tiny microscopic hairs around them as well, and that will trap any humid air, reducing the water potential gradient and therefore reducing evaporation. We'll come to the examples of the plants once we've gone through gas exchange in plants. So the structure, or the cross-section of a leaf, we have at the very top the waxy cuticle, the upper epidermis, the palisade mesophyll, which is the site of photosynthesis, spongy mesophyll, that's where we have lots of air spaces. We then have the lower epidermis, and within the lower epidermis there are guard cells which create these pores known as stomata. This bit here is the vascular bundle, which is made up of the xylem and the phloem. And we can see those structures here under the microscope as well. So gas exchange in plants then, the palisade mesophyll, as we just said, that is the site of photosynthesis. So that is where lots of oxygen is going to be produced and carbon dioxide is going to be used up. That is what is creating the concentration gradient. So because we have that concentration gradient, oxygen will then travel through the air in those spaces in the spongy mesophyll and diffuse out of the stomata created by the guard cells, assuming that oxygen is not being used in respiration. Carbon dioxide, because that's being used up, we also have a concentration gradient compared to inside the leaf and the outside, so carbon dioxide will diffuse in through the stomata. So that is how the concentration gradient is maintained. To reduce water loss, the stomata will close when it is not light, so at night, because they won't be photosynthesizing, so they don't need carbon dioxide. The short diffusion distance is the fact that leaves are so thin, so it's a very short distance for the gases to diffuse from the outside in the atmosphere through the stomata to the palisade mesophyll. And the large surface area, leaves are very broad, so you're going to have lots of these cells and also lots of stomata, so there's a large surface area for that gas exchange as well. So the gas exchange at the stomata, oxygen diffuses out, carbon dioxide diffuses in, and we've just said to reduce the water loss, stomata close at night. Now, the other adaptations, in particular in xerophytic plants, which are plants that are adapted to survive in really hostile abiotic factors where there's very little water, the structural adaptations that they have are, first of all, a lot of the time the leaves curl up, and that means all of the stomata are facing inwards, and any water that does evaporate gets trapped, and it creates a very humid atmosphere to reduce the water potential gradient, so less water evaporates out. You also have these hair-like structures, which again trap humid air to increase the humidity. Sunken stomatas, again, that increases the local humidity. And you have a thicker waxy cuticle as well to help reduce evaporation too. You do have a longer root network. Now that's not going to reduce water loss, but that means you can reach more water to more readily replace the water that might be lost. Next then, 3.3.3, digestion and absorption. So during digestion, the definition of digestion is large biological molecules are hydrolyzed into smaller molecules that can be absorbed across cell membranes. And in mammals, we will learn about how carbohydrates, lipids, and proteins are digested. Those are the three biological molecules that you need to know. So first of all, the carbohydrates, they require more than one enzyme to hydrolyze them into the monosaccharides. It's amylases and membrane-bound disaccharidases. And the amylase is produced by both the pancreas and the salivary glands. So that's why digestion begins in the mouth, because the salivary glands will release amylase, and then in the mouth, any starch that is there will start to be digested. That will then be swallowed, goes into the stomach. The stomach acid will denature that amylase, and that is why the pancreas then releases more amylase into the very top part of the small intestines known as the duodenum, and you do have to say the duodenum to get the mark. And because we've now got more amylase being released there, any remaining starch can continue to be digested. Then as that food travels into the next part of the small intestines called the ileum, most of that starch should have now been digested into disaccharides, and embedded within the membrane of the epithelium of the ileum, you have membrane-bound disaccaridases, meaning enzymes embedded within that membrane that can digest the disaccharides into monosaccharides. So that could be the enzyme sucrase, digesting sucrose, or lactase, digesting lactose, or maltase, digesting maltose, all of which will hydrolyze the disaccharides into the monosaccharides, which are then small enough to be absorbed from the ileum into the bloodstream. So here are the three word equations for biological molecules, but just as a reminder here, because this could come up on paper 1, so that means even though this is paper 1 theory, it could come up linked to digestion, knowing what the three disaccharides are digested into or hydrolyzed into by those membrane-bound disaccaridases. Next then, proteins, and this starts in the stomach, it continues in the duodenum, and then it's fully digested in the ileum of the small intestines. And there's three enzymes involved in hydrolyzing these large polymers into the monomer amino acid. We have the endopeptidases. These hydrolyze the peptide bonds between amino acids in the middle of the polypeptide chain. That means that you're going to be breaking or hydrolyzing one long polypeptide chain into multiple shorter chains. And this increases the surface area for the next enzyme to work with. And that is the exopeptidases. And these hydrolyze peptide bonds between amino acids at the end of the chain. So by hydrolyzing the chain into many smaller ones, that's what the endopeptidases do, that increases the number of exposed ends for the exopeptidases to work on. So those two work together, doing the same thing, hydrolyzing peptide bonds, just in different locations to speed up protein digestion. Now, if there are any dipeptides remaining, meaning two amino acids still joined together, by the time that protein or digested protein then reaches the ileum, it's the membrane-bound dipeptidases, which are enzymes that can hydrolyze the peptide bond between two amino acids. Those enzymes will work to hydrolyze a dipeptide into amino acids, and the amino acids are small enough to be absorbed. So in terms of these proteins, you would have in the stomach the endo and exopeptidases. The pancreas will release more of those into the duodenum, and then in the ileum you have your membrane-bound dipeptidases. Next then is the lipids, and these are digested by only one enzyme, lipase, but also by the action of a chemical called bile salts. So lipase is produced by the pancreas and it releases it into the duodenum. And lipase can hydrolyze the ester bonds in the triglycerides. And that's the bond between the glycerol molecule and the fatty acids. So by hydrolyzing those ester bonds, you are hydrolyzing and releasing those fatty acids, and you do get monoglycerides as well. Bile salts are not enzymes, but they are produced by the liver. That will then be released into the gallbladder, and then the gallbladder releases it into the duodenum. And what bile salts do is emulsify these large lipid droplets into smaller droplets to increase the surface area for lipase to work on. They're also important in the formation of micelles. That comes up in lipid absorption rather than digestion. So this can be split down into the concept of physical and chemical digestion. The physical digestion is this emulsification. So the bile salts produced by the liver emulsify the lipids, meaning splitting it up into smaller droplets, but no chemical bonds are broken. So it's not chemical digestion, it's just splitting it up into smaller droplets so that there's faster hydrolysis by lipase. Chemical digestion is when bonds are broken, and lipase hydrolyzes the ester bonds so that we get glycerol, fatty acids, and monoglycerides. A monoglyceride is the glycerol molecule with one fatty acid still attached. Next then we move on to absorption, and in mammals the products of digestion are absorbed across the cells lining the ileum, which is within the small intestines. So adaptations to increase absorption, and this is going back to this concept of this is an exchange surface, so we need to look for adaptations that increase the surface area, reduce the diffusion distance, and maintain the concentration gradient. So the ileum wall is covered in villi, which are these foldings, which have thin walls surrounded by a network of capillaries, and also these epithelial cells have even smaller microvilli, which is the folding of the cell membrane. And it's those features that maximize absorption. So the increased surface area is the villi and the microvilli, because you've got that highly folded surface. Decreasing that diffusion distance is because there's thin walls, but also the capillary network branches all the way up into the villi. So it's a short distance to diffuse across to reach the capillary network. Maintaining the concentration gradient is also because of that capillary network, because any digested food molecules that do get absorbed are transported away in the blood in the capillaries. Now, I've also got a table of adaptations listed here that I repeatedly have seen come up in the mark scheme. So you can screenshot this, turn it into a flashcard. These are what come up in long answer questions, short answer questions for adaptations to maximize absorption of digested products. So learn that. So now we've had a look at the adaptations for absorption, we need to know how the products of lipid digestion are absorbed, and also how the products of carbohydrate and protein digestion are absorbed. Starting with lipid absorption. And now we get to find out more about what are micelles. So micelles are water-soluble vesicles. And these vesicles contain fatty acids, glycerol, and monoglycerides. Those are the products of lipid digestion. So your triglyceride, after it's been digested, that's what we have. And it's also bile salts combined with those. And it's the bile salts that make these products, these fatty acids, water soluble. So that's what a micelle is, but a common exam question is, how do micelles help with lipid absorption? And they do it in these ways. So we've just said that the micelle is fatty acids, monoglycerides, bile salts, and those bile salts help to make the fatty acids soluble in water. Now that is an advantage for lipid absorption because it allows the micelles to move the fatty acids and monoglycerides from wherever in the lumen they were digested towards the cell surface membrane of the epithelial cell. Because the contents of the lumen, which is like the hollow space, the hollow tube of your small intestines, that is containing lots of watery substances from digestion. And fatty acids are not soluble in water. So we need them to be soluble so they can move easily towards the epithelial cell surface membrane. So that's why being water soluble is an advantage. So the second mark links to that. The micelles carry and then release the fatty acids and the monoglycerides to the epithelial lining of the ileum. So that then means the fatty acids are all released at this point. It's actually often another marking point from here that it helps to maintain the concentration gradient of the fatty acids near the epithelial cell. And the fatty acids, monoglycerides and glycerol, those are then absorbed by simple diffusion. The micelle does not enter, the micelle releases those at the cell lining. So those will then enter by simple diffusion because the cell membrane is made up of phospholipids, and only non-polar molecules can diffuse through those, and fatty acids are non-polar. So inside of the cell, once it's been absorbed in the cell, it will then be reformed into triglycerides, and that happens in the Golgi body. So we get them packaged into these triglycerides, into vesicles, and those vesicles containing the packaged triglycerides will then move towards the cell surface membrane on the other side, fuses with the membrane, it then releases that triglyceride, which is picked up by a lacteal rather than the blood in the capillaries, and that is the end point of the absorption. Then we need to look at how the products of carbohydrate digestion and protein digestion are absorbed. So products of carbohydrate digestion are monosaccharides, for example glucose. Products of protein digestion are the amino acids. And both of these are absorbed into the blood in the same way, and it is co-transport, which you learn in topic 2. So for co-transport of glucose, and it'd be exactly the same for amino acids. First of all, we have sodium ions from the epithelial cell actively transported into the blood. This creates a concentration gradient of sodium ions from the ileum into the epithelial cell. And therefore, the sodium ions can enter the epithelial cell down their concentration gradient, but they are an ion, so they have to go through a protein. And the protein that they can transport through is a co-transporter protein, which will bind sodium ions and glucose or amino acids. And that is then how the glucose and those amino acids enter the epithelial cell. Once inside of the epithelial cell, we already have a concentration gradient then. There'll be lots of glucose in the cell compared to in the capillary. And that means then that glucose can move down its concentration gradient into the blood and the capillary by facilitated diffusion through a transport protein. And it has to be facilitated because glucose is too large to move by simple diffusion through the phospholipid bilayer. Next then, 3.3.4 mass transport. Starting with hemoglobin, and these are groups of proteins found in different organisms. And hemoglobin is a protein with a quaternary structure. Quaternary structure from topic 1 means a protein with more than one polypeptide chain. And hemoglobin has 4 polypeptide chains, each of which has a heme group attached, which is where you have the iron-ion. And it's this heme group and the iron-ion where oxygen binds. So one hemoglobin molecule can bind 4 oxygen molecules, and then it transports the oxygen to respiring cells. Myoglobin sometimes comes up in application questions. So just to give you an awareness, this is another protein that can transport oxygen. It's not hemoglobin, but it's like hemoglobin in that it can transport oxygen, but it can only transport 1 oxygen molecule at a time. And some animals have a lot of this, and we have it in our muscle tissues. So oxyhemoglobin dissociation curves. Oxygen is going to be loaded in areas in the body with a high partial pressure of oxygen. So for example, the alveoli in the lungs, and it will be unloaded in regions of low partial pressure, so where oxygen's been used up for respiration. So on our oxyhemoglobin dissociation curve, this axis is showing us the percentage saturation of oxygen on the hemoglobin at different partial pressures of oxygen. And that basically means the amount of oxygen present. Partial pressure is a way to consider concentration. So in areas where there's a low partial pressure, meaning not a lot of oxygen present, that will be respiring tissues because oxygen's constantly used up in aerobic respiration. And at areas with a higher partial pressure, for example, the alveoli in the lungs, that is because as you inhale, you're getting new air, which will have lots of oxygen within it. So this oxygen dissociation curve, or oxyhemoglobin dissociation curve, it shows us that at low partial pressures, hemoglobin is not as saturated as it is at high partial pressures. And if it's not as saturated, that must mean it's either unloading oxygen or not loading oxygen as much. And we'd say at that point, that would mean the hemoglobin has a low affinity for oxygen. Affinity being the attraction and ability to bind oxygen. Whereas at higher partial pressures, because the hemoglobin is nearly fully saturated with oxygen, that must mean that hemoglobin has a very high affinity for oxygen. It's loading lots of oxygen onto it. Cooperative binding is this concept of when you have different numbers of oxygen bound to the hemoglobin, it affects the ability of subsequent oxygen molecules to bind. So hemoglobin can associate with, which means load, 4 oxygen molecules because of those 4 heme groups. And as each molecule binds, it slightly changes the shape of hemoglobin, exposing the further binding sites, making it easier for each subsequent oxygen to bind. So if we think of this as, we've got 0, let's say that's the first one binding, second oxygen binding, third and fourth. We can see that we've got not a particularly steep gradient compared to the point of 2 and 3, showing that it must be hardest for the first oxygen to bind. And that's because at this point the binding sites are slightly covered. Once that first oxygen binds, it changes the shape and exposes the binding sites. So we can see we've now got a steeper gradient, and that is representing that it's easier for the second and the third oxygen to bind. However, when we get to the fourth oxygen, we can see the gradient is less steep again and then it plateaus. And that's because if 3 of the 4 binding sites are already saturated, you've got a 1 in 4 chance now of the oxygen binding with an empty binding site. And that's why the ability to bind the fourth one is also more unlikely or challenging. So this all shows us that in areas with a high partial pressure of oxygen, as we said, there'll be a higher affinity. In areas with a lower partial pressure, there'll be a lower affinity. The Bohr effect is looking at the effect of carbon dioxide dissolved in the blood on hemoglobin's affinity. And when carbon dioxide dissolves in the blood, and by the way, this carbon dioxide is coming from aerobic respiration. So when you're respiring lots aerobically, you will have a lot of carbon dioxide dissolving in your blood, forming carbonic acid. Carbonic acid decreases the pH of the blood, and that decrease in pH slightly changes the tertiary structure of the polypeptide chains in hemoglobin. And this is why the affinity for oxygen decreases in respiring tissues. That change in shape is going to decrease the affinity. So hemoglobin ends up loading less oxygen and unloading more. And that is an advantage because if hemoglobin is going to be unloading oxygen more readily at respiring tissues, that means it's delivering the oxygen to the site of aerobic respiration where it's needed, so aerobic respiration can continue. And the way we can see this on the graph is, as the pH becomes more acidic, we can see the curve shifts to the right each time. And the best way to consider this in an exam is, at any of the partial pressures, use your ruler, which I don't have right now, and draw a vertical line and read off what the partial pressure is. Sorry, the saturation, not the partial pressure. So read off what the saturation is for that partial pressure. And let's say this one is maybe 25, 39, and that might be 56. That's very inaccurate readings off. But the point is, even at the same partial pressure, the most acidic is 25% saturated. The least acidic is 56% saturated. So what this shows us is the hemoglobin in the more acidic conditions must have a lower affinity, indicated by the fact it's less saturated, so it's more readily unloading oxygen rather than loading. So if a curve shifts to the right, that is what it's indicating, and this is how I demonstrate it in an exam. Read it off and show that even at the same partial pressure, the saturation is lower, showing that it must have a lower affinity for oxygen. Within the spec, it does say you can get applications linked to different hemoglobins for animals. So it says you need to know that animals are adapted to their environment by possessing different types of hemoglobin with different oxygen transport properties, which basically means based on their environment, they've evolved to have hemoglobin with either a higher affinity for oxygen if they live in an environment which has a low partial pressure of oxygen, Or they might have evolved to have hemoglobin with a lower affinity for oxygen if they need to readily unload it for constant aerobic respiration at higher rates. So things like small organisms that need to maintain their body temperature and they lose more heat across their surface. You might also have questions linked to fetal hemoglobin and myoglobin as well. Myoglobin has a very high affinity for oxygen, even at very, very low partial pressures, and in that way it can act as an oxygen store. Linked to the fetal hemoglobin, this is showing us in blue the curve for fetal hemoglobin and green, the adult hemoglobin, and that would be the mother, for example. And a fetus doesn't breathe. It gets all of its oxygen from the placenta as the blood then goes from the placenta, passes across the umbilical cord and into them. And that means that it's really important that a fetus has hemoglobin with a higher affinity for oxygen than the mother's hemoglobin's affinity for oxygen. Because that then will show us, again, I'd draw a line. Let's say we were at 25 partial pressure. We can see that we're maybe 45% saturated, whereas here we're 70%. So that would mean at this partial pressure, some of the oxygen is being, almost half of the oxygen, in fact over half of the oxygen has been unloaded from the mother's hemoglobin. And that can then be picked up by the fetus's hemoglobin because they are still at a high saturation, indicating a higher affinity. So at these lower partial pressures, the adult, the mother hemoglobin, will be unloading oxygen, but the fetus's hemoglobin, which has a higher affinity even at low partial pressures, will be able to load up the oxygen still. And in that way, a fetus gets oxygen from the mother. So we move on to the circulatory system, and mammals have a closed double circulatory system. Closed meaning the blood remains within the blood vessels and the heart, whereas the double circulatory system means the blood passes through the heart twice in each circuit. So one circuit goes to the lungs, one circuit goes to the rest of the body. And mammals require a double circulatory system to help manage the pressure of the blood flow. So the lungs are directly behind the heart. So they're not a very far distance at all. So you don't need a high blood pressure to make sure that the blood reaches the lungs. And in fact, we don't want a high blood pressure because that could damage the capillaries in the lungs if the blood was pumping through at that high pressure. And also it's an advantage to have a lower pressure because that reduces the speed at which the blood flows, which means there's more time for gas exchange in the alveoli. In contrast, we do want a high blood pressure for the blood flowing in the circuit from the heart to the rest of the body because we need it to ensure it can reach all of the respiring cells in the body. You need to know some key blood vessels. It's not all of the ones in the body that we can see here, but it's the ones in the red boxes. So you need to know the coronary arteries, which we can't see here, but these are the blood vessels that are on the outside of the cardiac muscle on the heart, and it supplies the cardiac muscle itself with oxygen for respiration. And then you need to know the blood vessels that are connected to the heart, the lungs, and the kidney. So we'll start at the top of the lungs. Any blood vessel that is connected will have pulmonary if it's for the lungs. And artery means it's blood coming away from the heart. Vein, if you think in, it's going back into the heart or towards the heart. So pulmonary artery is blood coming away from the heart to the lungs. And pulmonary vein is the blood vessel that is taking blood from the lungs back into the heart. Then if we move on to the heart, we've actually covered two already because the two that are connected to the lungs are also two connected to the heart. So then if we have a look at the blood vessels carrying blood away from and into on this side, the aorta is the artery carrying blood away from the heart to the rest of the body. The vena cava is the vein carrying blood from the body back into the heart. Vena cava meaning body vein. And lastly, the kidneys. Any of the blood vessels that are attached to the kidneys have renal in front of them. So renal vein means it's the blood vessel from the kidneys taking blood back into or towards the heart. and renal artery means it's blood coming away from the heart to the kidneys. So those major blood vessels are then connected within the circulatory system via arteries, arterioles, capillaries, also venules, but you don't need to know that for AQA, and veins. And that takes us on to these blood vessels. So arteries in general, away, they carry blood away from the heart towards the arterioles. Arterioles are smaller than arteries, they're branching and they connect to the capillaries. You don't need to know about the venules, but you then have the capillary network, and then you learn about the veins, which carry blood towards the heart. And I always say, like, vein, it's got in it, it's going back in or towards the heart, away. So we can see this here just from another angle, and it's showing us the inside of the walls, which are the adaptations that you need to know for the blood vessels. Now, the aorta is the largest artery in the body. It carries blood at the very highest pressure in the body. And it carries it at high pressure because it's carrying it directly from the heart, where that left ventricle would have just contracted, forcing the blood out at high pressure. So it contains a large amount of elastic tissue in its wall, which means that it can stretch when the ventricle contracts, and it can recoil when the blood is then flowing away, causing it to move back to its original shape. And this helps to smooth out blood flow and also maintain consistent pressure. It's got a very thick muscular wall as well, and that helps it to withstand high blood pressure and prevent it from bursting, whilst also the muscular wall can contract, and that helps to smooth the blood flow as well. Arteries in general, aorta is an artery, but it's the thickest or it's the biggest. Arteries all have thick muscular and elastic walls. That means they've got a narrow lumen for the blood to flow through, and it helps them to maintain the high blood pressure as they are directing the blood from the heart away to all of the cells of the body. Arterioles are going to be branching from the arteries and help to regulate blood flow into the capillaries. So they still have a relatively thick layer of muscle, and that can contract to narrow the lumen or vasoconstriction. That reduces the blood flowing into the capillaries. And when the muscle relaxes, the lumen widens, which is vasodilation, increasing the blood flow to the tissues. Lastly, in the veins, these carry blood back to the heart under low pressure. They have thinner walls. They still have muscle and elastic within the walls, but just a much thinner layer. And because it's a thinner wall, they have a wider lumen. And this is to reduce resistance of the flow. And that's because the blood is going to be flowing at a much lower pressure because it's now further away from the heart, which was the pump that increased the pressure in the first place. It also has valves which prevent the backflow of blood. So here's a little summary table of some of those features that you can screenshot and add to your notes. Capillaries then. These are the smallest of the blood vessels and adapted for efficient gas exchange or other exchange of substances between the blood and surrounding tissues. And some of the adaptations are, first of all, very narrow diameter, which means only one red blood cell can fit through at a time. That slows down the flow of blood, which is good because that means there's more time for exchange, for example, gas exchange. The walls are also only one cell thick, so it's consisting of a single layer of flattened endothelial cells, and that reduces the diffusion distance. Capillary beds are networks of capillaries that spread through tissues, and that provides a large surface area for exchange as well. And the extensive branching also means that all cells are going to be close to a capillary, and that is reducing the diffusion distance. So again, we're thinking here about large surface area and reducing diffusion distance. This leads us into tissue fluid. Tissue fluid is a fluid surrounding your tissues that contains water, glucose, amino acids, fatty acids, ions, oxygen that have all been forced out of the blood in the capillaries. So you need to know, how is tissue fluid formed, and then how does the water from the tissue fluid get reabsorbed? So in terms of the formation, the capillaries, we've said that it's only a single layer of cells making up that endothelium wall, that lining. And between every cell, there's going to be these microscopic gaps. So any molecule that's small enough to fit through those gaps can be forced out if there's a high enough pressure. And that's what we have at the arterial end. So this is where we'd have arteries, that branches into arterioles, and then that branches into the capillaries. And at the other end, the capillaries, then there's the venules and then the vein. At the arterial end, there is a high pressure. And that high pressure is originally generated from the contraction of that left ventricle in the heart. And then also the arterioles have got a wider diameter than the capillaries. So going from that larger diameter to a smaller diameter with the same volume of blood increases the pressure. And we call this high hydrostatic pressure. And because there's such high pressure, anything that's small enough to fit through those gaps between the cells and the capillaries is forced out, and that's known as ultrafiltration. Now, the key thing here to get the mark, you would have to name what gets forced out, not just say small molecules. So always name water, and then I would name at least 2 other things from this list. So water, glucose, amino acids are forced out. That is how tissue fluid is formed. So what is forced out then? Water, dissolved mineral ions, glucose, small proteins amino acids, fatty acids and oxygen. But anything that's too big to be forced out will remain in the blood in the capillaries. And that is primarily large proteins, also red blood cells and platelets, but the marking point is normally large proteins. And this is important because it helps to explain how the water from tissue fluid gets reabsorbed. So because those proteins remain in the blood in the capillaries, you end up with a much lower water potential at the venule end of your capillary compared to the water potential of the tissue fluid. And that is how water can then move from the tissue fluid back into the capillaries by osmosis. Now, in terms of mark scheme language, you must say it's water that is re-entering, not tissue fluid. So it's water that is entering by osmosis because of this decreased water potential. Now, within that water, there might be dissolved substances that are also reabsorbed, but that's not what you need to say. Because this is osmosis, that means eventually you will reach equilibrium in the water potential because water is moving in by osmosis. And when equilibrium is reached, no more water from that tissue fluid can be reabsorbed. But we need to reabsorb as much of it as possible, otherwise you get swellings in the body where you've got too much water outside of the blood vessels. And that's where the lymphatic system absorbs the rest. So then we move on to the heart, starting with the cardiac muscle that it is made from. So you get these thick muscular layers of cardiac muscle, and that has unique properties such as it's myogenic, which means it can contract and relax without any input from the nervous system or hormonal stimulation. So as long as it has a constant supply of glucose and oxygen in the blood in the coronary arteries, it can contract and relax. So it never fatigues as long as it does have that oxygen and glucose for aerobic respiration to make ATP so it can contract. So that's then our coronary arteries and the cardiac muscle, and this is just showing us the coronary arteries in more detail. So these branch off from the aorta, carrying oxygenated blood and also glucose that's needed for aerobic respiration. If one was to get blocked, this is now actually application, it's not on the spec anymore, but if you did have a blockage in your coronary artery, that means that all of these blood vessels would not have oxygenated blood flowing through them anymore. So all of that cardiac muscle would not get oxygen, glucose, wouldn't be able to respire, and it would stop contracting. And that is what a heart attack is, or myocardial infarction. It's when the heart, cardiac muscle stops contracting. In terms of the structure of the heart, you have 4 chambers. So you have the 2 atria at the top, the 2 ventricles at the bottom. The atria have much thinner muscular walls, and that's because they don't need to contract with much force at all, because they're only moving the blood from the atria directly down into the ventricles. And they're elastic walls to stretch when the blood enters. Ventricles have much thicker cardiac muscle, and that's so that they can contract with more force because they're pumping the blood further. The right ventricle is pumping blood to the lungs. The lungs are directly behind the heart, so it doesn't have to pump a very far distance. They don't need as thick a muscular wall compared to the left ventricle, which does have to contract with a large force to create a high blood pressure to pump the blood to all of the respiring cells in the body. So the left ventricle wall has the thickest cardiac muscle. Next is knowing the different blood vessels, which we actually touched on already because we talked about the pulmonary arteries are carrying blood away from the heart to the lungs. Pulmonary veins carry blood from the lungs back into the heart. The aorta is the artery that carries blood from the left ventricle to the rest of the body. And the vena cava carries blood from the body back into the heart. Next is looking at the valves in the heart. So the semi-lunar valves are the ones between the ventricles and the arteries. So you have one in the aorta, which you can't actually see in this diagram, but it's just about here. And then we've got one here in the pulmonary artery, just there. The atrioventricular valves are found between the atria and the ventricles, and we have them both on the left and the right. The bicuspid is the name for the one on the left side, and the tricuspid is the name for the one on the right, but it's sufficient just to call them both atrioventricular for AQA. Now, valves will open when the pressure is higher behind them, forcing them open, and a valve will close when the pressure is higher in front of them. And in this way, they control the flow of blood so that it only goes in one direction, and it prevents the backflow of blood in veins, but also within the heart, making sure that the blood does go from the atria into the ventricles, out of the arteries. The septum, now this isn't technically on the spec, but just to make you aware, the septum is the part of the cardiac muscle that runs down the middle, separating the left and the right side, meaning the oxygenated blood from the deoxygenated blood, and that is going to help maintain concentration gradients for diffusion, because you're going to have far higher concentration of oxygen being pumped around, whereas if this all mixed together, it's going to decrease the concentration of oxygen in the blood. We then move on to the cardiac cycle, and there are three stages: diastole or diastole, atrial systole or systole, ventricular systole or systole. People pronounce it differently, so I'm going to keep switching between the two because I haven't decided how I'd pronounce it yet. So if we start with diastole, this is relaxation, and diastole is when both the atria and ventricle cardiac muscle are relaxed, and all of this pale green bit is our ventricular diastole, and all of this section is atrial diastole. But diastole means the cardiac muscles are relaxed. And this is when the blood is going to be entering from the body into the atria, when we've got both the atria and the ventricles in diastole. And it's going to enter through the vena cava and the pulmonary vein into the right and the left atria. The blood flowing into the atria is going to increase the pressure within the atria compared to the ventricles, and that causes the atrial ventricular valves to open, and so some blood will start to move into the ventricles at this point. Atrial systole, wherever you see systole, it means contract. Atrial systole is a very, very brief period. It's not a long stage, and this is when the muscles in the walls of the atria contract, increasing the pressure further, forcing all of the remaining blood that was within the atria into the ventricles. The ventricular muscle walls are relaxed at this point. We can see they're still in diastole. Then we have ventricular systole. So this is when the ventricles contract. And this happens after a short delay, meaning the atria contract, there's a short delay, and then the ventricles contract. And as they contract, that's going to increase the pressure in the ventricles to the point where the pressure is now higher in the ventricles than in the atria. And that is what causes the AV, or the atrioventricular valves, to close. And then when the pressure is even higher compared to the arteries, it causes the semilunar valves to open. And that is what causes the blood to be pushed out of the ventricles through the two arteries, either to the lungs or to the rest of the body through the aorta. Now there is a math skill linked to this, the cardiac output, and that is the volume of blood which leaves one ventricle in 1 minute. And it can be calculated by doing the heart rate times the stroke volume. And heart rate is beats of the heart per minute. Stroke volume is the volume of blood that leaves the heart each beat. The final part is mass transport in plants. So we're going to look at transpiration first, the mass transport of water. And transpiration is the loss of water from the stomata by evaporation. And there are 4 key factors that can affect the rate of transpiration. Light is one of them because the brighter it is, the more the stomata open. So there's a larger surface area for that evaporation or transpiration of water. Temperature, if you've got a higher temperature, there's more kinetic energy, so the water molecules will move faster. So again, more evaporation and transpiration. Humidity, if it's more humid, there will be less transpiration. And that's because the more humid it is, the more water vapor in the air, and that will reduce the water potential gradient, so you get less water evaporating. The windier it is, that's going to blow away humid air containing that water vapor, and that can help to maintain the water potential gradient, so increasing that rate of transpiration. Then if we have a look at the movement of water up the xylem, water has to move from the roots all the way up a plant against gravity. And sometimes it's a small plant, sometimes it's a plant that might be several meters tall, moving water against gravity. And this is possible because of the cohesion tension theory. And that's what we're going to have a look at next. What cohesion is, what capillarity or adhesion is, and what we mean by the tension part in this theory. Now this links back to module 1 with the properties of water. Water is a polar molecule, meaning it's got a slight negative charge on the oxygen atom, slight positive charge on the 2 hydrogen atoms. And for that reason, hydrogen bonds can form between the oxygen and hydrogen of different water molecules. That is cohesion, those hydrogen bonds between water molecules sticking the water together. And that means that water can move up the xylem as a continuous column of water. Now you also get hydrogen bonds forming between the water molecules and the walls of the xylem, and that is adhesion, the sticking of those water molecules and then the entire water column to the walls of the xylem. And that can result in the xylem being drawn in more and more narrow, because we're going to look at how the water column gets pulled. And if you're pulling upwards on that column and the column is all attached and it's stuck to the wall, as you pull it up, it's like an elastic band. It's going to cause the xylem to become longer and narrower. And the reason that helps move water up against gravity is the narrower the tube, the more surface area you have for the water column to stick to, and that means it can actually move up the tube with more ease, which is just shown here with like different diameter straws. So the cohesion tension theory then. Water evaporates from the stomata. That is transpiration. That would be your first mark. Water evaporates from the stomata. As that water evaporates, it decreases the water potential in those surrounding cells, and that creates tension in the water column and on the xylem, and that draws and it pulls the water up. So that'd be your next mark. As that water is lost by transpiration, that creates tension on the water column and it pulls the water column up. The water molecules stick together because of cohesion. So that's your third mark. All of those water molecules form a continuous water column because they stick together because of the cohesion by the hydrogen bonds. And then at the same time, we do also have the column of water sticking to the walls of the xylem, adhesion, and that helps to move it up and it also draws in the xylem narrower under that tension. So that is our cohesion tension theory. As I've read this through, this is a lot of text here. That's to give you the full understanding, but the bits that are in bold, those are your key marking points typically, and the ones that I pointed out as well. So then we have a look at the mass transport of sucrose in plants. And this is one of our products of photosynthesis. And all cells need this sucrose to then break down or hydrolyze into glucose for respiration. So this is going to be transported from the leaves where the organic substances are made in photosynthesis, to all the respiring cells through the phloem. And the sugary solution, that sucrose solution, can move up or down through the phloem, whereas in the xylem, it only ever moves upwards. So the phloem tissue, there are two key cell types. You have the sieve tube elements and the companion cells. The sieve tube elements are here in the middle. They are living cells, but most of the organelles have disintegrated. So there's no nucleus and they don't have many other organelles. And that's why they need these cells next to them, called companion cells. And the companion cells will have lots of mitochondria in them so that they can do lots of aerobic respiration and provide the ATP needed for active transport in this process. So this links to the mass flow hypothesis, and this is looking at how the sucrose that's made in the leaves, which is our source, can be transported to the respiring cells, which is known as the sink. So we're going to go through this process of how it happens. There's a lot of text here, which I'm not going to talk through at this point. By all means, you can pause, write it down, but it's also going to come up in the subsequent slides. So I'm going to talk through it with those. This has just got a lot of the information in one place for you to screenshot if you want. So the source to sink explanation with this model helps to explain the mass flow hypothesis in terms of how the hydrostatic pressure causes the mass movement of sucrose solution. So in this tank, this model, we've got a tank of water, we've got one tube representing the phloem, another tube representing the xylem. And our source cell would be our photosynthesizing cell, where you'd have lots of sucrose being produced. And sucrose lowers the water potential of the cell and also of the phloem. And that means water is going to enter from the xylem into the phloem. At the sink cell, because lots of respiration is occurring, that's going to be using up sucrose, and that means you'll now have a more positive water potential, or less negative. So water will actually move from the phloem in this region back into the xylem, and that will decrease the pressure. So at this end, we have an increase in the hydrostatic pressure as water moves in by osmosis. And at this end, we have a decrease in hydrostatic pressure because water leaves by osmosis. And as a result, because we've got high pressure and low pressure, that forces the liquid through the phloem from the source to the sink. So you also need to know, well, what is it that is creating this hydrostatic pressure and how does the sucrose move? So our source cell, we're looking at it slightly differently here now because it's actually in the plant rather than a model. The source cell is the leaf. That's where photosynthesis happens, sucrose is produced. Sucrose is actively transported from the source cell into the sieve tube element. So it has to go from the source to the companion cell to the sieve tube element. And that is done using ATP because it's active transport. And the ATP is provided by the companion cells. So this is your mark number 1 in translocation. Sucrose is actively transported using ATP from the companion cells. Now we've got the sucrose into the sieve tube element. That lowers the water potential of the sieve tube element, and therefore water from the xylem will enter the sieve tube element by osmosis. So here would be our mark 2. That increases the hydrostatic pressure. That would often be mark number 3, or these 2 points will be split between 2 and 3 marks. And because we've got a high hydrostatic pressure, that forces that sucrose solution to move en masse. So this is our mass transport towards the sink cell. And that is because at the sink cell, sucrose is being used up in respiration. And therefore, we're going to have sucrose from the phloem being transported into the sink cell, initially by facilitated diffusion, but when the concentration gradient is equal, you have equilibrium, you'll then have active transport of the sucrose from the sieve tube element into the sink cell. But either way, that means you have less sucrose here. That now means you have an increase in the water potential gradient, and water will move from the sieve tube element by osmosis back into the xylem. So it's that pressure gradient, the high hydrostatic pressure compared to a low hydrostatic pressure, because water enters by osmosis, water leaves by osmosis. That's what causes the mass flow, the mass transport of the sucrose solution from the source to the sink. Now, there are investigations that have been linked to translocation and working out the involvement of the phloem, which are tracers and ringing experiments. Tracers involves radioactively labeling carbon, and then that carbon is provided to the plant in the form of carbon dioxide. So by only providing the plant with radioactive carbon in the carbon dioxide, that means over time, all of the carbon-containing compounds in the plant should contain the radioactive carbon, because the carbon dioxide is taken in for photosynthesis and turned into sucrose, other sugars. So that means that you can take thin slices through the stem, and under the X-ray film, any radioactive carbon is going to show up. So in this way, you can track the route that the radioactive carbon takes through the plant, and that is how they could see where the phloem was, and that it must be the phloem that is transporting the sugars, because they are what lit up with that radioactivity under the X-ray. Ringing experiment is when a ring of bark, which will contain the phloem, are peeled and removed from a tree trunk. This can actually also be done on stems of smaller plants as well. And the result of removing the phloem is the tree starts to bulge here and here, so above where it's been removed. And that bulging, you can remove the liquid and analyse it to see it contains sucrose. But what this indicates is if you remove the phloem, the sucrose is made in the leaves further up, and when you take the phloem out, you get this bulging of liquid. The liquid contains sucrose, and that shows us that it must be the phloem transporting sugars, because when you remove them, the transport stops. AQA topic 4, and we begin with 3.4.1, DNA, genes and chromosomes. So comparing the DNA in a eukaryotic and prokaryotic cell is the first part of the spec, and this has been quite a common long answer question to do this comparison. So I've put it together here as a table so that you can see that direct comparison. And by the way, it's fine to present your answers as a table like this or a bullet point list in the exam, except for the essay. So eukaryotic DNA and prokaryotic DNA both are made up of DNA nucleotides. So that means they both have nucleotides that contain deoxyribose, the pentose sugar, phosphate group, and those nitrogenous organic bases. The nucleotides are joined together by phosphodiester bonds still, but the differences are eukaryotic DNA is longer than prokaryotic DNA. It's linear in shape, whereas it's circular for prokaryotic DNA, and then associated with the histones in eukaryotic DNA and not associated with histones in the prokaryotic DNA. So in eukaryotic cells, a DNA molecule is associated with the histones, forms a chromosome. So that's what we can see here. We've got the chromosome, and that is made up of tightly coiled DNA that is wrapped around these histone proteins. There is also DNA inside of the mitochondria and inside of chloroplast, which you learn about in topic 2. And the mitochondria within those organelles in eukaryotic cells contains DNA which is very similar in structure to the DNA that you get in prokaryotic cells. And the similarities are it's very short, it's circular, and it's not associated with proteins. So that is the properties of the DNA in mitochondria and chloroplasts, and that is a similarity to the DNA in prokaryotes. A gene is a base sequence of DNA, and it codes for the amino acid sequence of a polypeptide and functional RNA. And the mark scheme is really specific that you do say a base sequence of DNA, not just it's a section of DNA. And every gene occupies a fixed position, which means a certain place on a chromosome, and that's known as the locus. So for example, here we've got the locus for flower colour. The gene is for flower colour, that's the locus, meaning the location. You would always find it on the chromosome. A sequence of 3 DNA bases is a triplet, and that codes for a specific amino acid. And that is the fundamentals of the genetic code. There's 3 features of the genetic code that you need to know as well. So the genetic code is degenerate, it's universal, and it's non-overlapping. Degenerate then, this is based on the fact that there are 20 amino acids that the genetic code is able to code for. there are 4 DNA bases: guanine, cytosine, thymine, and adenine. And just a note here, although I've abbreviated it, you are not allowed to abbreviate those in the exam. You have to write the full name. And therefore, 3 bases are needed to make enough combinations to code for at least 20 amino acids. And we can prove this mathematically. So 4 to the power of n, there's 4 bases. N is how many of those bases you would need to get enough to code for 20. So if it was 1 base, that would be 4 to the power of 1, which is 4. So that would mean we'd only be able to code for 4 amino acids. Guanine would code for 1, cytosine would code for 1, thymine would 1, and adenine, and that's not sufficient. If it was 2, that would be 4 to the power of 2, and that gives us 16 combinations. So that is not enough to code for 20 amino acids. 4 to the power of 3 is 64, so that is sufficient to code for 20 different amino acids, plus there are extra. And that is what leads to this property of the genetic code being degenerate. Because there's 64 combinations, which is more than the 20 amino acids, as a result, each amino acid is coded for by more than 1 triplet of bases. So that is what we mean by the genetic code being degenerate. And we can see that here in this genetic code wheel, where you would start on the inside, work your way out. So GUC, that codes for Val, that's one of the amino acids. But GUU, GUA, GUC, all of those code for the same. By the way, this is a genetic wheel which always has the codons, which are mRNA. That's why we've got U for uracil instead of T for thymine. Now, it's an advantage that the genetic code is degenerate, because if a gene mutation occurs and it's a substitution mutation where one of the bases is now swapped and we have a different base, it might mean that the triplet of bases still codes for the same amino acid, and therefore, even though there was a mutation, you still code for the same amino acid, so it has no effect on the overall protein that is made. Universal is the next feature, and this means that the same triplet of bases codes for the same amino acid in all organisms. So that is your definition. Now, this has been used to our advantage because we can do genetic modifications, because we know that you can take a gene from one organism, put it into the DNA of another, and because the genetic code is universal, it'll still code for the same amino acids and make that protein. Non-overlapping means that each base in a gene is only part of one triplet of bases that codes for one amino acid. So therefore, each codon or triplet of bases is read as a discrete unit. So we can see here it's read as a discrete unit of 3, this is a discrete unit of 3, and so on. And that, again, is an advantage in case there is a gene mutation. Because if one base does get substituted and it does code for another amino acid, at least it's only affecting one triplet, whereas if it was an overlapping code, that one mutation would affect 3 amino acids. So 2 other key concepts are knowing what coding and non-coding DNA is within your DNA. So there are non-coding multiple repeats of bases that you find between genes. But in addition to that, within a gene, you do also have sections that code and sections that don't. So within a gene, only some sequences, and these are the exons, code for amino acid sequences. And you also have sequences within a gene that do not code for sequences of amino acids, and those are known as the introns. So these non-coding sequences, you do have them between genes, but also within genes. And it's the ones that are within genes that are called introns, and those get removed during one of the stages of protein synthesis that we'll have a look at shortly. So 3.4.2 is DNA and protein synthesis. Now, I've mentioned this term codon, but the definition of codon is 3 bases on mRNA that code for a specific amino acid. The start codon is a particular set of 3 bases, and it's the 3 bases at the start of every gene that initiate translation, which is the second stage of protein synthesis. The stop codon is 3 bases at the end of every gene, and this causes a ribosome to detach in translation, and therefore it stops translation. So it stops protein synthesis. Genome and proteome. The genome is an organism's complete set of DNA in a cell, whereas the proteome is the full range of proteins that a cell is able to produce. So the genome should stay the same. It's not going to change unless there are mutations. Whereas your proteome, that does stay the same because it's the full range of proteins, but the reason it says is able to produce is not every cell is going to make all of the proteins that it's able to, but the proteome is the full range that the cell is able to make. Doesn't mean it does make all of those proteins. So next, let's have a look at the three types of RNA. We have M, T, and R. mRNA is short, it's single-stranded, and you find it in the cytoplasm and the nucleus. It's made during transcription, which we're going to see shortly. That's the first stage of protein synthesis. And it's a copy of one gene. So it's going to be a complementary copy to that DNA sequence in the gene. And as we said, 3 bases on mRNA are a codon, and that codes for 1 amino acid. tRNA, or transfer RNA, this is found in the cytoplasm, and specific amino acids become attached to transfer RNA molecules. Each tRNA is specific for 1 amino acid. Each tRNA molecule has an anticodon, which is these 3 bases here, which are complementary to the bases in a particular codon. You also have an amino acid binding site at the top, which is where a specific amino acid will bind, and that's how tRNA can bring a particular amino acid. This is involved in translation. It brings the amino acids to the mRNA on the ribosome. It's still single-stranded, it's folded to create what's known as a clover leaf shape. And those bases, as they're folded, will form hydrogen bonds, and that's what holds it in this structure. Now, I did say we talk about rRNA. That actually mainly comes up in topic 1. rRNA, you just need to know that combines with proteins to make ribosomes. So let's move on to protein synthesis. This happens at the ribosome, but it does actually, the first step where you make mRNA happens in the nucleus. So we're going to go through transcription and translation. Transcription is when a complementary mRNA copy of one gene of the DNA is created in the nucleus. As we said, the mRNA is much shorter than DNA, and that's because it's only a copy of one gene rather than the entire genome. And because it's shorter, that means it's able to fit through the nuclear pores in the nuclear envelope. It moves out, and then it can attach to a ribosome in the cytoplasm to be used in the next step. So if you were asked to describe transcription, here are your key marking points for transcription. First of all, the hydrogen bonds between the DNA bases break, and that causes the DNA double helix to unwind and the 2 strands to separate. Only one of the DNA strands acts as a template though. You will then get 3 mRNA nucleotides from within the nucleus, attracted to and aligning opposite their complementary bases. And in RNA, the complementary bases, just bear in mind, because it's RNA for mRNA, you have uracil instead of thymine. So any place on DNA where there'd be adenine, uracil would be the complementary base pair. Now that's not normally a mark, point 4, if it's a describe transcription question, but it's just worth noting. The enzyme RNA polymerase is then responsible for catalyzing the condensation reaction, which joins the adjacent RNA nucleotides together, and in doing that, phosphodiester bonds are formed. Once that mRNA is copied, it then gets modified, and then it's ready to leave the nucleus. So these modifications then, we actually call the mRNA before it's been modified pre-mRNA. Once it's been modified, then we call it mRNA. So the modification that happens is, once you've made your mRNA, and we've now got pre-mRNA, the introns get removed. And that's known as splicing. So the introns are spliced out, which just means they're cut out and removed. So then you're just left with the exons. And the exons are the sequences of bases on mRNA that code for sequences of amino acids. Now that happens in eukaryotic cells. It won't happen in prokaryotic cells because prokaryotes, such as bacteria, they don't have introns in their DNA, so they don't have to go through that step. The next stage is translation, and this is when the mRNA has now been modified, it leaves the nucleus, and it's going to attach to the ribosome, and tRNA is involved as well. So here are our key steps. Number 1, I've just said. Number 2, the ribosome, once that mRNA is attached to the ribosome, will start at the start codon and then it moves along the mRNA. But the key thing here is knowing that the mRNA attaches to the ribosome. Sometimes there's an extra mark for saying at the start codon, or it will move to the start codon. Mark number 3, always going to be a mark. A molecule of tRNA has an anticodon complementary to the codon on mRNA, and that is due to complementary base pairing. Number 4 is always a mark. Each tRNA molecule carries a specific amino acid, and that is determined by the anticodon. The ribosomes will then join those amino acids, and a peptide bond forms between the amino acids. ATP is required for the formation of that peptide bond, but it's actually also required to join the amino acid to the tRNA molecule. The ribosome will then move along the mRNA to the next codon. The process repeats with new tRNA molecules bringing more amino acids and binding them. And this will continue until a stop codon is reached. So your absolute key marking points that you should always include would be number 1, number 3, 4, 5, 6, and 7. So 1, 2, 3, 4, 5, 6. This is often a 4 to 6 mark question. So they might be, here's your range, you have to say 4 of those, 6, or if it was a 6 marker, you'd have to say all of them. Next then we move on to 3.4.3, genetic diversity, and this is looking at mutations and meiosis. So genetic variation can be introduced in these 3 ways: through meiosis, mutations, and random fertilization of gametes. So meiosis is the type of cell division that creates genetically different cells, which are the gametes, so sperm and egg cells. And unlike mitosis, there are 2 rounds of division. In meiosis 1, that is when homologous chromosomes are separated. In meiosis 2, it's when the centromere splits and the sister chromatids are separated to the opposite poles. And by the end of both divisions, you have 4 cells. They are haploid, meaning only 1 copy of each chromosome, and they are genetically different. So I've just put this down here as a reminder of what haploid and diploid means. Haploid means 1 copy of each chromosome, and that is what you'd have in the gamete. So for example, the haploid number for a human is 23. Diploid means 2 copies of every chromosome. So for humans, the diploid number is 46. Now, the way that meiosis introduces genetic variation is the key focus here. You don't have to know PMATs, prophase, metaphase, anaphase, telophase, like you did for mitosis. You just need to know for meiosis how the genetic variation is introduced. And that occurs in 2 processes: independent segregation of the homologous chromosomes and crossing over between homologous chromosomes. You have to say homologous chromosomes for both of those. And if you're not sure what that means, homologous chromosomes are 2 chromosomes that have the same genes. So for example here, these 2 are homologous. They are the same size, same shape, and it's because they contain the same genes. You just have 2 copies of them. One that would have come from the mother, one from the father. Now, both of these processes here occur in meiosis 1, which is the first round of division. So let's go through independent segregation then. As we said, it happens in meiosis 1, and that is when the homologous pairs of chromosomes line up opposite each other at the centre of the cell, or the equator of the cell. But it's random which side of the equator the paternal and the maternal chromosomes will line up. They're always in their pairs, but it's random which side they go onto. And that matters because when the homologous pairs get separated, so those chromosomes are pulled apart to the opposite end, depending which side of the equator they're on, will determine which chromosomes you get in these cells here, and then those get separated, the chromatids. So that is how the genetic variation is introduced. And there is actually a maths question linked to this quite often, using this formula here, 2 to the power of n, because you can use this to work out how many possible different ways are there for these homologous pairs to line up at the equator, and therefore how many different gametes could be made. And it's 2 to the power of n, 2 because we have pairs of chromosomes, n is for the number of homologous pairs you have. So for humans, that would be 2 to the power of 23, which is over 8 million. So because of independent segregation, there's over 8 million different ways that our homologous pairs of chromosomes can line up at the equator, and therefore over 8 million different potential gametes one human can make, just from independent segregation. But on top of that, crossing over can also occur. Now, crossing over is rare. That becomes important when you get to topic 7, learning about autosomal linkage. But for now, we're just going to go through what crossing over is and explain how it introduces genetic variation. So it also happens in meiosis 1. Again, it's linked to this homologous pair lining up. So we get the homologous pairs lining up at the equator. And when you have a pair next to each other at the equator like this, we call it a bivalent. So they line up and they form a bivalent. The non-sister chromatids, one chromatid is one of these arms of a chromosome. Non-sister means it's the chromatids from different chromosomes. So the blue and the pink here are non-sister chromatids. They cross over and entangle, and the exact point where they cross is called a chiasmata. Chiasma is the singular, chiasmata is the plural. And at each of those chiasma, you can get a break happening, and then you exchange that section of the non-sister chromatids. And as a result, when these chromatids separate to go on to make the final 4 gametes from one round of meiosis, you end up with new combinations of alleles in the chromatids because they've swapped sections of the chromatid. So if we just compare meiosis to mitosis, meiosis is 2 nuclear divisions versus 1. Meiosis, you get a haploid cell versus diploid. Meiosis introduces genetic variation. Mitosis, you get genetically identical cells. You could also be asked for meiosis to identify where meiosis is happening within an unfamiliar life cycle. And the key thing here is don't look for where gametes are made, because for some organisms, meiosis isn't making gametes. So instead, what you need to look for is where you go from having a diploid cell to a haploid cell. And they might write diploid or haploid, or they might write 2N, meaning diploid, or N. So that's what you need to look for on a diagram. And this is a bit blurry, but we can see here that we've got N, 2N, N, N, N. So where we go from the zygote to the zoospore, we've got 2N to N. That would be the meiosis stage because we've gone from diploid to haploid. Not here where we have that gamete being made. Now in this one, we can see everything on this side is 2N, everything on that side is N. So that means at this stage here, it's labelled on, but if it wasn't, this is meiosis because we've gone from having a 2N cell to all of these being N. So that's what to look for. Diploid to haploid is always where the meiosis is occurring in a cycle. Gene mutations is another way that genetic variation is introduced. And a gene mutation is a mutation in a gene, but it means you're changing the base sequence of the DNA for that gene. And these mutations randomly occur during DNA replication. So there's a link here to DNA replication in topic 1, and there's a link here to the cell cycle in topic 2, because DNA replication happens in interphase during S phase. So these random mutations are more likely to occur if you are exposed to mutagenic agents, and they can interfere with DNA replication, and that's why. So mutagenic agents include high energy radiation such as UV light, ionizing radiation, which would be gamma rays and X-rays, and chemicals, so carcinogens such as mustard gas and cigarette smoke. There are 2 types of gene mutation that you need to know for topic 4. You do learn more in topic 8, but for paper 1 or for the AS, you only need to know deletion and substitution mutations, which is modelled here. So here is our original unmutated DNA sequence. A substitution is when one base is changed, and we can see that cytosine base in the original has been changed to adenine, and maybe that might still code for the same amino acid because the genetic code is degenerate, or maybe not. A base mutation that is a deletion is when one entire base is deleted, and we can see that one of the cytosines has been deleted here, and all of the bases after the point of that mutation have now shifted left. And this is known as a frame shift because all of the codons after the point of mutation have now changed. So that means we're going to code for all of those, probably different amino acids. So a deletion mutation is far more harmful, potentially, than a substitution mutation because more amino acids will be coded for incorrectly. You can also get chromosome mutations, and this is mutations in the number of chromosomes, and that can happen during meiosis. Now, a chromosome mutation, if you're asked in the exam to name the chromosome mutation, it's non-disjunction. That's come up quite a lot recently, and sometimes students aren't sure what they're meant to say the name of the chromosome mutation is, and the name of it is non-disjunction. So non-disjunction, what this means is during meiosis, either the homologous chromosomes in meiosis 1 don't separate equally, or if it's meiosis 2, the sister chromatids don't separate equally. So as a result, you end up with more either chromosomes or chromatids going to one cell, and the other cell has a lack of chromosomes. So that is the non-disjunction chromosome mutation. Now this is just going to show you it, so you can visibly see the outcome. So in this scenario, it's in meiosis 1. The homologous pairs have lined up at the equator, and for some reason the spindle fibres are all coming from one of the centrioles, and that means all of those homologous pairs are going to be pulled to this cell. This one has got none, so that cell isn't going to survive, and this one has now got an extra copy of every chromosome in it. Meiosis 2 is when the sister chromatids separate, but because we had 2 copies of every chromosome, we end up with 2 copies in our gametes. So we have diploid gametes here instead of haploid. So this is one example of non-disjunction where we now have an extra copy of every chromosome in that gamete. Now, if that gamete was to fuse with a haploid gamete where there are no mutations, then 2 plus 1 equals 3. So we now have a triploid cell. So this is one example of non-disjunction. And this does happen quite commonly in plants, and plants can still grow and survive that, but this isn't seen in humans. Triploid would be fatal. That wouldn't lead to a fetus and a baby that could actually be born. Now, another type of non-disjunction we can see here, it's still happening due to the centrioles only producing spindle fibres on one side, but meiosis 1 happened normally. This time it's meiosis 2, where the sister chromatids are both pulled to the same side. This one, there was no chromosome mutation, no non-disjunction, but in meiosis 2, this one there was. All of the sister chromatids moved to one side, so again we end up with that diploid gamete and one gamete with no chromosomes. Now, another type of non-disjunction that can happen is if only one of the chromosomes, the spindle fibres, are attaching at one side. And as a result, both copies of only that chromosome move to the same cell. So we still end up with a haploid gametes, but you have one extra chromosome. So it's haploid, we've got one copy of all the chromosomes, except for this one, we've got one extra. And this one's haploid, one copy of all of the chromosomes, except we're missing one. So it'll be described as n+1, and these are n-1. And if you then had a gamete like this, combining with a haploid gamete, you end up with a diploid zygote plus one extra chromosome. And that's actually what happens in Down syndrome. There are 3 copies of chromosome 21 because of this type of non-disjunction chromosome mutation. If it was the n-1 gamete, then you'd end up with a zygote that was still diploid minus 1 chromosome. Next then we move on to 3.4.4, genetic diversity and adaptation. And genetic diversity is the number of different alleles of genes in a population. And this is one of the key driving factors of natural selection, because you have to have genetic diversity in order to select which allele is going to be passed on. So a gene pool can be described as all of the genes and all of the alleles in a population at a particular time. The allele frequency is the proportion of organisms within a population carrying a particular allele. We're going to see these key terms come up as we go through natural selection and evolution. So natural selection is the process that leads to evolution. Your definition of evolution is the change in allele frequency over many generations in a population. And this helps to ensure that you have a population of species better adapted to their environment. And these adaptations could be anatomical, physiological, or even behavioural adaptations. So natural selection is random mutations occur. That's always your first marking point. If it's an applied example, you have to state which allele has been made. So in this case, it might have been there was a random mutation that created the white mouse or the black mouse, whichever one it was. That introduces genetic variation. Those are often a combined mark. Some mutations might be harmful, but some are actually advantageous. So if an individual did have that allele, they might be more likely to survive. And if they're surviving, they're more likely to reproduce. And if they reproduce, then they're passing on that new mutated allele to their offspring. And if this repeats over many generations, most of the population or a lot of the population will now have this allele in their DNA. So that means the frequency of that allele has changed over many generations, which was our definition of evolution. Now, there's 2 different outcomes that you learn about, which is types of selection or outcomes. You do learn a 3rd type in topic 7, but there's only 2 that you need to know for paper 1 or for your AS. And one of those is directional selection. And this is when the allele that is providing the selective advantage is one of the extreme traits. So it could be very, very dark fur if it was the mice, or very, very light fur if it was the mice. Or the example that you actually need to know is antibiotic resistance. So bacteria that are very, very, very resistant compared to not very resistant or not resistant at all, the very resistant ones are surviving. Their alleles are then being passed on. And the outcome is we see this shift from the original red line showing us the original distribution of the different alleles in the population. Now we can see there's been a change in the allele frequency because most individuals now have that extreme trait. Stabilizing selection is if there's no change in the environment. So whatever was the most common or the modal trait, that is still the allele that's providing the selective advantage, and it still therefore remains the most common. It's just over many generations of natural selection happening, there's still a change in the allele frequency because now even more individuals have that modal trait and the extreme traits have been lost. So in terms of standard deviation, the spread of data around your mean, we've lost those extremes, so the standard deviation has decreased. The example linked to this is human birth weight. So a middling birth weight is a selective advantage, because if you're much, much lighter, then potentially you were premature, and that means maybe you're less likely to survive. And if it's a very, very big baby, you're more likely to have complications with the birth. So the middling birth weight is a selective advantage. Next then we've got 3.4.5, species and taxonomy. The definition of a species is two organisms that are able to breed and make fertile offspring. But the key thing is, can they breed and make fertile offspring? If they can, they're part of the same species. And species must reproduce and pass on advantageous alleles in order for the entire species to survive. And courtship behaviour, we did say behaviour is an adaptation. Courtship behaviour is genetically coded for, it is an adaptation, and it is essential for successful mating and species recognition to help the entire species survive. So what courtship behaviour means is, it's the different behaviours that animals demonstrate as a way to attract a mate. And each species has their own unique sequence of behaviours to ensure successful mating. And what we mean by successful mating is if the two individuals are mating and produce offspring. Now, normally the courtship ritual is performed by the male, and then the female selects which male they want to reproduce with. And the behaviours are often a sequence of behaviours. And it's not necessarily just behaviours, it could be sounds, the release of pheromones, which are chemicals. So it could be dance moves, displays of colour and feathers. There's lots of really extravagant courtship behaviours in plants. And in fact, if you just search birds of paradise courtship behaviours, there's some really cool David Attenborough videos on YouTube for that. So females will observe the courtship ritual and decide if they want to mate with that male. So the reason this is important is, as we said, it's to ensure successful reproduction, which means mating and producing offspring so that the whole species can survive. And the reason it increases the likelihood of an offspring being produced is it enables you, first of all, to recognise members of the same species. Now that sounds fairly obvious and not needed if you're thinking about humans, but for other animals, they do rely on this to make sure they are the member of the same species before they reproduce. Secondly, in order to be able to make offspring, they'd need to be mating with a member of the opposite sex. So that's the first two things that courtship helps animals to do. Recognise members of the same species, recognise the opposite sex. Next is synchronising mating behaviour. Now what this means is animals will do the courtship ritual and they'll be able to recognise what that ritual means only if they are producing gametes. So males will only do the dance when they are producing gametes. Females will only recognise and respond to the courtship ritual when they are releasing eggs. And again, that then means if they reproduce, you're more likely to have offspring because both the male and female are producing gametes, which could then fuse together to make an offspring. Now, on top of that, this isn't to ensure successful mating, because this is now saying for some animals, there's an extra importance to courtship behaviour, and that's to ensure the survival of their offspring. And that's because these courtship rituals can help to form a pair bond, which means the two parents should stay together. And we see this in penguins because it's a massive advantage to the survival of the offspring if they have a mother and a father, because one of them stays to look after the young while the other goes to find food. It also helps to enable the female to select a healthy male to mate with, and therefore their offspring is more likely to be healthy and therefore increases the survival of their offspring. And the logic behind this is, if a male is fit and healthy, then they'll have more energy that they can put into their dance routine, or their feathers will look brighter in colour, they might look better conditioned. So that is the idea behind that one. Next then is the binomial system. Bi meaning two, nomial meaning name, so the two name system. And this is when you use the genus and the species to name the organism. And this is the universally used, meaning it's the same across the world. The universal scientific way to identify an organism is the binomial system, genus and species. And this is useful because, first of all, you can share data, but also it gives you more information than the common name would. So for example, we've got two birds here. The common name is New Zealand robin, and then this is a robin. And you might think, oh, they're both called robins, so they must be closely related. But if we have a look at their name using the binomial system, their genus and species is different. So they're not actually even closely related. So it's useful to be able to see, are different species part of the same genus? Because if they are, that means they're closely related. If they're not, then they're not closely related. And that links us into phylogenetic classification. And this is arranging species into groups according to their evolutionary origins and relationships. So it's a way for us to see, for different species, how closely related they are. Do they share recent common ancestors? And that's what these phylogenetic trees show. So humans, for example, in this tree, we're most closely related to chimpanzees because we share the most recent common ancestor. Then as we go back in time, so we're going back in time this way, after that, here is the next most recent common ancestor. So that means after chimpanzees, we're most closely related to these two. And then we can go back again and see it's then these, and so on and so on. So every fork or branching point represents a common ancestor, which means that is the species, anything that branches off, evolved from. Now, linking to classification still, individuals can be classified according to their physical and biochemical similarities, and classification systems are an example of a hierarchy. And that is a really common 2-mark question. What is meant by hierarchy? And it's when you have smaller groups arranged with larger groups. That's mark 1. Mark 2 is there's no overlap between the groups. So what that means is we've got an example of a hierarchy here, which you're probably familiar with from GCSE, and you do still need to know this for A-level. We can see that within a genus, there are lots of smaller groups. And that's because in one genus, there'll be lots of different species, but there's no overlap between those species. They're all different species, they just all belong to the same genus. And then we can go up again and say the same thing with genus. Within a family, there's lots of different genuses, but there's no overlap between them. 3.4.6 is biodiversity within a community. And biodiversity refers to the variety of life, and it can be measured on different scales. It can relate to a small local habitat, such as a pond or a woodland, or the entire earth. And there's different terms to classify or to describe biodiversity. We've got species richness. That definition is the number of different species present in a community. So it just tells you how many different species there are. Index of diversity gives you more information than that because it's looking at the relationship between the number of species, so the species richness, but it also takes into account the number of individuals within each species, which is far more helpful because then we can get an idea of, are any of those species at risk of going extinct, because we now know the number of individuals within each species. And biodiversities can be used to describe, as we said, local habitats or the entire earth. And a low biodiversity isn't always a cause for concern because it might be naturally there is just a very, very low biodiversity because there's such harsh abiotic factors. So, for example, in the Arctic or anywhere with extreme abiotic factors. The concern is when you have an area that had a high biodiversity and it's starting to decrease because something is causing individuals and maybe entire species to die out. And often that is due to human activity. Now we can calculate this index of diversity using this formula. They give you this formula, but they don't always tell you what the capital N and lowercase n is. Often they do, but I'd still say learn it because the most common mistake students make on this is misremembering what the lowercase n actually means. So capital N is the total number of every organism alive in that area, whereas lowercase n is just a population size of one species. The best way, though, to get an idea of this is we'll go through an example. Now, with this formula, typically, you will get a value that is between 1 and 10. It doesn't have to be 10 as the maximum, but usually you get an answer somewhere between 1 and 10. So if you do this in the exam using this formula and you get an answer like in the 50s or 100s, you've gone very wrong somewhere. Recalculate it, try again. So to model this then, capital N we said is the total number of organisms. So that is the sum of that column. We've got 4 species, added all the individuals together. Lowercase n is the population size of each species. And we have to do n times n minus 1, and then it's the sum of that for every species. So n is 6, so we need to do 6 times 6 minus 1, which is 5. 6 times 5 is 30. Then for this one, 3 times 3 minus 1, so 3 times 2 is 6. 12 times 11, 132. 4 times 3 is 12. And then the denominator is the sum of all of those. So you do have to work this out for every single species they give you in the table. Add that all together and that gives us 180. Then the numerator is the total number of individuals, which in this case is 25. So we do 25 times 25 minus 1, which is 25 times 4. And then that comes to 3.3 when we finish the rest of that equation. Farming is one of the examples of human activity that can reduce biodiversity. And within the specification, it says you need to be awareness of the balance of we need farming to provide food for humans, but there needs to be a balance between having that farming and not having too negative an impact on the biodiversity. So some of the ways that modern farming techniques might reduce the biodiversity are the removal of habitats. So that could be removing hedges and putting in fences instead, or just not having a hedge there at all. It could be removing habitats by cutting down a forest and then just planting crops. It could be using a monoculture, which means just growing one species. And if you've removed a whole range of plant species and replaced them with only one, reduce the species richness, but also the variety of food available for herbivores. If pesticides and herbicides are used, that is going to kill animals and plants, which again is reducing the species richness. And the overuse of fertilizers can cause harm in aquatic ecosystems as well. But 2 key marking points that always come up linked to this concept of farming techniques and how this can reduce biodiversity is there will be fewer habitats. So fewer habitats and fewer food sources. Not quantity of food, it's food sources, meaning one plant might eat grass, another plant might eat daisies. I'm just doing 2 random examples. So if you only have grass now, any of the animals that ate daisies can't survive there. So it's fewer habitats, fewer food sources if you have fewer plant species present. Next then, investigating diversity. So genetic diversity refers to the variety of alleles within or between species. And you can assess this by comparing the frequency of measurable or observable characteristics. So for example, the flower colour. But this can be less reliable because characteristics are also influenced by the environment as well as the genetics. So more accurate ways to compare are comparing the DNA base sequence. This is a direct measure of how similar or how different your DNA is. You could also do base sequence of mRNA because that is copied from the DNA. Or it could even be the amino acid sequence in proteins because amino acids are coded for by DNA. However, we do need to bear in mind for this one, because the genetic code is degenerate, 2 organisms might have the same amino acid sequence for a particular protein, but their DNA base sequence might be different because the genetic code is degenerate. So the most accurate measures are comparing DNA base sequences or mRNA base sequences. So when measuring variation within a population, this is where we get the concept of sampling, because we'd take samples because it'd be too time consuming to literally measure it for every individual organism, and it might not be very accurate. So to investigate variation scientifically by doing sampling, you have to make sure that you're doing random sampling to avoid bias. You must take large samples to ensure the results are representative, and then calculate a mean to summarise the data. You can then also go on to do standard deviation and even statistics. That is going to be the most useful because if you do a statistic, you can see if you have a significant correlation or a significant difference depending on what sort of pattern it was you were looking for. AQA topic 5. And we begin with 3.5.1 photosynthesis. And everything in topic 5 is A level only, not for AS. Now the first part of photosynthesis is a little recap on the structure of a chloroplast because this is where photosynthesis happens. You have a double membrane organelle, and then the thylakoids are folded membranes which contain photosynthetic pigments such as chlorophyll, and these are going to be involved in the light-dependent reactions in photosynthesis. You also have this fluid-filled centre called the stroma, and that is where the light-independent reactions happen. Now, chlorophyll is actually made up of many photosynthetic pigments, and these are in those proteins embedded within the thylakoid membrane. And the advantage of there being multiple photosynthetic pigments is each of those pigments absorbs light at a different wavelength. And that then means there's a bigger spectrum of visible light that is able to be absorbed by the plant. And if a bigger spectrum of wavelengths can be absorbed, that means overall more light energy is absorbed. And if more light energy is absorbed, then there's more energy for the light-dependent reactions. So the light-dependent reactions then, this is the first stage in photosynthesis, and it happens on the thylakoid membranes, or a stack of them is known as a granum. The light-dependent reactions require light energy and they use water, and they produce ATP and reduced NADP. And that is through these 3 stages: photolysis of water, photoionization of chlorophyll, and chemiosmosis. So we're going to go through those 3 and see how it results in the production of ATP and reduced NADP, which are needed for the light-independent reactions. Photolysis of water, photo, if you see that in biology, it means light, lysis means splitting. So it is using light energy to split water. That is what photolysis is. And we've got an equation here. Water, and it's using the light energy, is split into oxygen, electrons, and protons, also known as hydrogen ions. The protons are going to be used to reduce NADP, and that helps to maintain a proton gradient in one of the later stages. Electrons, those are going to replace the electrons that get lost in chlorophyll in one of the stages we look at. Oxygen, this is a byproduct, it's not actually used. So a few things just to point out here. NADP is a coenzyme. These are not enzymes. They don't catalyze reactions. They are going to be carrying molecules from one point of a reaction to be used later. And if it's reduced, it means it's gained a proton and an electron. So that is what we mean by that term. Photoionization of chlorophyll. This is when light energy, again, so photo meaning light energy is absorbed by the chlorophyll, and that energy causes an electron to be released, so it's been ionized. But they are really specific with the terminology you use for this. So you'd have to say the light energy is absorbed by the chlorophyll. That's usually 1 mark. Then it's saying what that light energy does. So it results in the electrons becoming excited and raising up an energy level. That is the language they put in the mark scheme, excited and raising up an energy level. And that causes the electrons to be released, and therefore chlorophyll has been ionized. And those electrons are going to be used in one of the later stages to help make ATP and reduced NADP. And that later stage that I keep referring to is chemiosmosis. This is where we're going to see the protons and electrons being used to make ATP and to make reduced NADP. NADPH is the same thing as reduced NADP. Either of those will be accepted on the mark scheme. So step 1, here is PS2. You don't need to know that terminology for AQA. That is just a protein embedded within the thylakoid membrane. And we can see here, this is the thylakoid lumen. This is the chloroplast stroma. So within the thylakoid lumen, so within inside of the thylakoids, that's where we have our photolysis happening. Water has been split into protons and electrons, which are picked up by this protein, and the oxygen's a byproduct. So we can see there we've got our electrons, and those are replacing the electrons that are getting excited, raising up an energy level and leaving the chlorophyll, moving on to the next protein in the photoionization of chlorophyll. So we get these electrons moving along this chain of proteins, and every time they move from protein to protein, they release energy. And that energy is used to pump protons from the stroma into the thylakoid lumen. And as a result, from those protons being pumped across and the protons released from photolysis of water, we end up with lots of protons on the thylakoid lumen side compared to the stroma, which means we've created an electrochemical gradient. Those protons can move down that electrochemical gradient back into the stroma, but it has to be through a protein, and the only protein they can move through is the enzyme embedded within the thylakoid membrane, ATP synthase. So the protons move through ATP synthase, and that enables that enzyme to catalyze the phosphorylation of ADP into ATP. So that is how ATP is made in chemiosmosis. The last step then is, how is this reduced NADP made, or NADPH? Well, the protons that have moved through ATP synthase are picked up by that coenzyme NADP, and also the electrons, when they get to the end of this electron transport chain, they are also picked up by NADP. And that then forms reduced NADP, or NADPH. And that's how we get those two products that we're now going to see used in the light-independent reactions, also known as the Calvin cycle. This stage happens in the stroma. It uses the enzyme Ribisco, so it's temperature sensitive. So the key things you need to know about the Calvin cycle are, it's going to use carbon dioxide. It also uses reduced NADP and ATP. The ATP is going to be hydrolyzed to provide energy for reaction. The reduced NADP is going to donate hydrogen, which is a proton and an electron, and therefore it will reduce molecules in the cycle. We're going to have a look at this whole cycle, but that's just a summary to be aware of. So here is the cycle. Step one is carbon dioxide is going to enter the cycle, and it combines with RUBP, which is a 5-carbon compound. So a 5-carbon compound, RUBP, is going to combine with carbon dioxide, a 1-carbon compound, and that is catalyzed by Ribisco. So that adds up to 6 carbons, but we actually get that splits to make 2 3-carbon compounds known as GP. So all of that is step number 1. Those 2 molecules of GP are then reduced to form 2 molecules of triose phosphate. So the way that they are reduced is using the reduced NADP. So the hydrogen from reduced NADP is gained by the GP molecules to reduce it. The ATP is hydrolyzed to provide the energy for that reduction. Now, some of those carbons from the triose phosphate are going to be removed each turn of the cycle. And it's actually going to be 1 that is removed. And that means if we're removing 1 carbon each time, we need to make a hexose sugar, such as glucose, the cycle has to happen 6 times in order for those useful organic substances, such as hexose sugars, to be made. But each turn of the cycle, because 1 carbon is removed, that leaves 5 carbons remaining. And it'd be as a 3 and a 2. And we need to join those back together to regenerate RUBP. And that's why ATP is also needed at this stage in the cycle. ATP is hydrolyzed to provide the energy to regenerate RUBP. Now, whilst glucose is the product, generally speaking in the mark scheme, they'll accept you saying organic substances, meaning it contains carbon and hexose sugars. But you can also get questions about what could that go on to form, linking to your knowledge of biological molecules. So an organic molecule contains carbon, so you could be creating glucose, sucrose. Those could then go through condensation reactions to make polysaccharides such as cellulose or starch, but it could also be converted into glycerol, and then the glycerol can combine with fatty acids to make lipids, or it could be used to make an amino acid if you add an amine group. There are limiting factors that reduce the rate of photosynthesis. Those are the light intensity, carbon dioxide concentration, and temperature. The light intensity is going to limit the rate because it provides the energy in the light-dependent reactions. So if you don't have a high enough light intensity, then photolysis of water and photoionization of chlorophyll won't be happening at a fast rate. However, it will get to the point where it can get brighter and brighter and the rate still remains the same because maybe you now have a different limiting factor such as carbon dioxide concentration or temperature. Carbon dioxide concentration can limit the rate because it is the reactant in the Calvin cycle. Again, it will plateau eventually because maybe you don't have a high enough light intensity or the optimum temperature. Temperature is linked to the idea of it being an enzyme-controlled reaction. So at too low a temperature, there's not enough kinetic energy for successful collisions, and therefore you don't get as many enzyme-substrate complexes. And too high a temperature, the enzyme denatures, active sites change shape, so again you don't get as many enzyme-substrate complexes. Now, for maximum photosynthesis, and therefore plant growth, in farming and agricultural practices, they want to be removing limiting factors so they get more crop to sell. And that could be by increasing the light intensity, the temperature, and burning fuel to release more carbon dioxide. But all of those strategies cost money. So they will have to work out if the extra growth from photosynthesis offsets the cost of paying for extra heating, lighting, and fuel. And this often comes up as application questions linked to experiments where you have to work that out. Next is 3.5.2, respiration. There's aerobic and anaerobic respiration, and we are going to start with aerobic, which is split into 4 key stages. We have glycolysis, which happens in the cytoplasm of the cell, but the other 3 stages do happen in the mitochondria. The link reaction and the Krebs cycle happen in the mitochondrial matrix, which is the fluid-filled center. And oxidative phosphorylation happens on the cristae, or the inner mitochondrial membrane. And this is an enzyme-controlled reaction, so it is sensitive to temperature changes and pH changes. The respiratory substrate typically is glucose, and that is broken down through the reactions and it will release ATP. But if there's no glucose, then glycogen in animals could be hydrolyzed into glucose, or if it's a plant, then the starch could be hydrolyzed into glucose. Now, if there's no glucose or glycogen in animals or starch in plants, then the next option is other biological molecules will need to be used. So lipids can be hydrolyzed into glycerol and fatty acids. The glycerol then gets phosphorylated into triose phosphate, and it can enter aerobic respiration at the Krebs cycle to be used. Proteins can also be modified and used. They'd have to be hydrolyzed into amino acids, and then the amino acids would have to be hydrolyzed and the amine group removed, or deamination. The carbon compound that remains could then enter the Krebs cycle. So let's have a look at all of these stages, starting with glycolysis, because that's the first stage. And this actually happens in both aerobic and anaerobic respiration. No oxygen's needed in this stage, which is why it's in both types of respiration, and it happens in the cytoplasm. So there's 3 key stages in glycolysis. Phosphorylating glucose to form glucose phosphate, and that requires ATP. The production of triose phosphate, and the oxidation of triose phosphate to produce pyruvate with the net gain of ATP and reduced NAD. So NAD is one of the coenzymes used in respiration, and it has the same role as NADP did in photosynthesis. Do not confuse those though, it will cost marks. So the way that I always say to remember it is NADP, think P for photosynthesis. So here is the glycolysis reactions. We start with glucose, a 6-carbon compound, and we said that is phosphorylated to glucose phosphate. Phosphorylation means adding on phosphate groups, and that's why we have to use ATP. 2 molecules of ATP get hydrolyzed, and that means the inorganic phosphate removed from those 2 molecules can be added onto glucose, and that makes glucose phosphate. That is a more reactive molecule, and that causes it to split into 2 molecules of triose phosphate. That triose phosphate is then oxidized to make 2 molecules of pyruvate. And we can tell it must be an oxidation because the coenzyme NAD has gained hydrogen from the triose phosphate. And if it's gained a hydrogen, that means it's a proton and an electron. So if triose phosphate has lost that proton and lost the electron, then it has been oxidized, and the NAD has been reduced because it gained that electron in the hydrogen. So you can call that NADH or reduced NAD. Now this oxidation reaction also produces 2 molecules of ATP. So this means from 1 glucose molecule, we get 2 pyruvates, 2 reduced NAD, and a net gain of 2 ATP molecules. And that's because although we've made 4 in total, we had to use 2 to start with. So it's a net gain of 2. Mark schemes are incredibly picky that you say net gain of ATP. Even better if you can say a net gain of 2. So now we've got our pyruvate and those 2 molecules of reduced NAD. Those are going to be actively transported into the mitochondrial matrix to be used in the next stages. The ATP can be used in the cytoplasm for other reactions. The link reaction is the next step, and this is when pyruvate is going to be oxidized further into acetate. And again, we can tell it's an oxidation reaction because NAD has now formed reduced NAD. So NAD has been reduced, which means pyruvate must have been oxidized. 1 carbon has also been removed, so we've formed 1 carbon dioxide molecule. So that means we've gone from pyruvate, which is 3 carbons, to acetate, which is 2 carbons. Now acetate can't enter the Krebs cycle as it is, so it has to combine with a coenzyme called coenzyme A to make acetyl-CoA to enter the Krebs cycle. So that's a link reaction, and for every glucose molecule we end up with 2 acetyl-CoA, 2 carbon dioxide, and 2 reduced NAD. And the reason for that is for every glucose molecule, 2 pyruvate molecules were made in glycolysis. The Krebs cycle then, and this is actually just showing you the link reaction leading into the Krebs cycle. Acetyl-CoA is going to combine with a 4-carbon molecule. So we've got 4 plus 2, that makes a 6-carbon molecule. And once that has happened, coenzyme A is released and it can be reused in the link reaction. We then have a series of redox reactions where we go from having a 6-carbon molecule back to the 4-carbon molecule. And you don't need to know all of the intermediates, you just need to know that when you go from that 6-carbon molecule back to the 4, there's a series of redox reactions. And the carbon compounds are being oxidized. The coenzyme NAD and FAD are being reduced. And we get 3 reduced NAD being made and 1 reduced FAD. Because we're going from a 6-carbon to a 4-carbon, we've also lost 2 carbons to create 2 molecules of carbon dioxide. And that whole stage also releases 1 molecule of ATP. So the product per cycle is 3 reduced NAD, 1 reduced FAD, 1 ATP and 2 carbon dioxides, but per glucose molecule, which is really how we should do it, because in respiration, in aerobic respiration, we will have 1 glucose molecule entering, which means the cycle will happen twice because we get 2 pyruvate, therefore 2 acetyl-CoA. So that means from 1 glucose, we have 6 reduced NAD, 2 reduced FAD, 2 ATP and 4 carbon dioxide molecules. So if we have a look at everything we've made so far then, we have got in total from glycolysis, link reaction, and Krebs cycle, 10 reduced NAD, because we had 6 made here, 2 made here, and 2 made here, taking into account how many times each of these processes occurs. We have 2 reduced FAD from the 2 lots of the Krebs cycle. We have a total of 4 ATP molecules so far. We had a net gain of 2 in glycolysis, and we had 2 from the Krebs cycle happening twice. And we have 6 carbon dioxide, which is waste, it's not useful, but we do have 6 carbon dioxide molecules. We had 2 being produced in the 2 lots of the link reaction, and 4 being produced in the 2 lots of the Krebs cycle. These types of questions are becoming more common, where you have to say how many of a particular molecule you have and identifying where it came from. So you do need to make sure you know all of this chemistry. So the final stage is oxidative phosphorylation. And this is when most of the ATP is made, because so far we've only made 4, a net gain of 4, which is not enough for metabolism. And this stage involves the transfer of electrons and the movement of those electrons along that inner mitochondrial membrane and the protons moving across. So in this one, first of all of those reduced coenzymes that we made are going to accumulate here in the mitochondrial matrix. The hydrogen is removed, and that is then split into protons and electrons. The electrons are picked up by these proteins and are passed down a series of electron carrier proteins embedded in that inner mitochondrial membrane, and this is the electron transport chain. As they move from protein to protein, they release a small amount of energy, which is used to pump the protons from the mitochondrial matrix into the intermembrane space, which is the gap between the inner membrane of the mitochondria and the outer membrane. That then creates an electrochemical gradient, meaning lots of this ion, the hydrogen ion, on one side. And as a result, the hydrogen ion can move down its concentration gradient through the enzyme ATP synthase, and that enables ATP synthase to phosphorylate ADP, meaning ADP plus PI, into ATP. So that is how we get lots of ATP being produced. And this is almost identical to that chemiosmosis that we saw in photosynthesis as well. Final thing then is the role of oxygen. And this is a really common key marking point. Oxygen is the final electron acceptor. What that means is oxygen will pick up the electrons at the end of the electron transfer chain. And it picks up those electrons, it also picks up the protons, and that then forms water. And it's really important that oxygen does this, because by picking up the electrons at the end of the chain, it means the electron transfer chain can continue, so protons continue to be pumped, and therefore ATP continues to be made. Anaerobic respiration is when there is no oxygen, and it only happens in the cytoplasm. The pyruvate that's produced in glycolysis is reduced instead of oxidized, and it will form ethanol and carbon dioxide if it's a plant or a microbe, or lactate or lactic acid in animals. And this is by the pyruvate being reduced because it gains the hydrogen from reduced NAD. That reoxidizes the reduced NAD into NAD so that it can be used in glycolysis again to make sure that at least glycolysis can still continue even though there's no oxygen, so that you get some ATP. So let's just have a look at that then. Here's glycolysis, as we just learnt, and in animals, lactate is made. So that pyruvate is reduced into lactate by gaining the hydrogen, which is a proton and an electron. That NAD that has been made or regenerated, reoxidized, can then be used in glycolysis again, so at least you still continue to get a net gain of ATP. Exactly the same in plants and microbes, but the product's different, ethanol and carbon dioxide. Next then is 3.5.3, energy and ecosystems. Now in any ecosystem, plants are going to be the producers in a food web. And that's because they produce their own organic compounds from photosynthesis, like we just saw. And between each trophic level, meaning the stage in the food web, energy is lost. And that's because between each stage, the organism will be respiring, so using up that glucose, and therefore there is less biomass available in the organism. And also there'll be energy lost through excretion as well. Everything that remains that is available for the next organism to consume is the biomass, and the amount of biomass remaining in the organism can be measured in terms of mass of carbon or dry mass of tissue per given area, but usually it's mass of carbon per given area. So to measure that dry biomass, if you wanted to know what the amount of biomass was, because it is the dry mass or the mass of carbon, you have to dehydrate it. And in order to dry out your sample to measure the biomass, you have to heat it in an oven at temperatures lower than 100 degrees C. Now, this has come up a few times recently, and students didn't get the mark unless they said heat. You can't just say put it in an oven. You have to specify heat it in an oven, and it has to be at a temperature lower than 100 degrees C, because anything above that will cause it to burn, and if it's burning or combusting, you're making carbon dioxide, and that is removing carbon. So heat it in an oven lower than 100 degrees C, and you need to constantly weigh while you're doing this drying, and then when the mass becomes constant, that means you now have just the dry mass remaining because no more water is evaporating, which is why the mass would have been decreasing. The chemical energy stored in dry biomass can also be measured or estimated using calorimetry. And this is done by burning the sample in a bomb calorimeter. And then the energy released from that combustion is used to heat a known volume of water. What you would do is measure the temperature increase, and that is then used to calculate the amount of energy that there must have been in the biomass, because that energy has been converted into heat energy, so it would be the temperature increase. And that is measured in joules or kilograms per gram. Next is known about NPP and GPP. How productive an ecosystem is depends on the abiotic and biotic factors. So if there's plenty of favourable abiotic factors, water, a high light intensity, optimum temperature, then green plants will be able to photosynthesize at their maximum rate and therefore produce more organic substances such as the carbohydrates, and therefore it's more productive. And this can be quantified using GPP and NPP. GPP, which is gross primary production, that is the chemical energy stored in plant biomass in a given area or volume. So basically it's the total energy resulting from photosynthesis. Net primary production, or NPP, that's the chemical energy stored in plant biomass after respiration losses have been removed. And that is basically what is available for the next organism in the food chain, because you've taken away the losses, the energy losses already. Now, if you are asked what either of those terms mean, always give the equation, because that is always worth a mark. The NPP, as I said, that is the energy left over after respiration has been removed, and therefore it's what there is available to make new biomass. Another equation you need to know is the net production of consumers in a food web. And to work out the net production of consumers, known as N in this formula, you would need to do I, which is the chemical energy stored in the ingested food, minus the energy lost. And animals can lose energy through feces and urine and respiration. So we can see we've got our energy input through the ingested food minus the energy lost through respiration and through feces and urine. The productivity refers to the rate at which biomass is produced. Primary productivity is the rate at which producers convert light energy into chemical energy or biomass via photosynthesis. Secondary productivity is the rate at which the consumers produce biomass by eating other organisms. And the rates of productivity are recorded using these units: kilojoules per hectare per year. So kilojoules is the unit for energy, but we do it per area and per year. And the reason we do it per area is so that we can standardize the results, taking into account that if you have a bigger area, you probably have more plants, so you would have more energy. And we do it per year to standardize the impact of the seasons. Because if you measured one area in summer and another in winter, summer would have a higher productivity because there's more plants photosynthesizing. Next is 3.5.4 nutrient cycles. Nutrients are recycled within natural ecosystems, and we see this in the nitrogen cycle and the phosphorus cycle for AQA. And microorganisms play a vital role in both of these cycles to recycle the elements phosphorus and nitrogen. Mycorrhizae are the fungal associations that you get between plant roots and beneficial fungus. And this is going to be useful because the fungi has these hyphae which can branch through the soil and increase surface area for water and mineral absorption. The mycorrhizae also act a bit like a sponge. They can really absorb and retain water. So that makes it quite drought resistant because that water and minerals is going to be shared with the fungus and the plant. So this is vital in the nutrient cycles because if there is a soil which is quite low in either nitrate ions or phosphate ions, having mycorrhizae present will increase the ability to absorb that for the plant. And this is a mutualistic relationship because the plant also provides a benefit to the fungus because plants photosynthesize and it can share some of those carbohydrates with the fungus. So let's look at the nitrogen cycle. The nitrogen cycle is a really important one because nitrogen is needed for making proteins, ATP and nucleic acids. And the challenge in this cycle is, although nitrogen is very abundant in the atmosphere, nitrogen gas has a triple bond between those two nitrogen atoms, and we do not have an enzyme that can break it. However, microorganisms do. So microorganisms are incredibly important in this cycle. There's 4 key stages: ammonification, nitrification, nitrogen fixation, and denitrification. All of these stages use microorganisms. So step 1 is nitrogen fixing or nitrogen fixation, which I've labelled on the diagram. This is when the nitrogen from the atmosphere is fixed by bacteria, which means bacteria break that triple bond between the N2, the 2 nitrogen atoms, and it fixes it, meaning converting it into a nitrogen-containing compound, which is ammonium ions. Now that can either be done by bacteria that are free living in the soil, so the ammonium ions are fixed into the soil, or there are some plants, such as leguminous plants, which are clover and beans, that have these nodules, which means lumps, on their roots. And within those root nodules, there are bacteria that can fix that nitrogen directly into the plant itself. Step 2 is nitrification. This is a 2-stage oxidation reaction. The first step is those ammonium ions are nitrified into nitrites by nitrifying bacteria. Nitrites are then oxidized further, again by nitrifying bacteria, into nitrates. And those nitrates can then be absorbed by the root hairs of the plant and be assimilated, meaning used in the plant and converted into proteins or DNA. And to get into animals, animals eat the plants. Ammonification links to the death and decomposition of animals and the decomposition of plants. But here is where you have to be really particular with your language to get the mark. You have to name the molecule that would be in the dead animal or plant or in the waste. You can't just say a dead animal or plant is broken down. So for example, proteins in a dead animal or proteins in a dead plant, urea, which is an example of the waste, or DNA in the dead animal or plant, you'd have to name it. Those are then decomposed by saprobionts. And saprobionts are bacteria and fungi that can digest dead matter or waste outside of a cell, so extracellularly, and that then reforms the ammonium ions. Now the last stage I've not actually labelled on, it's over here, nitrification. This is not a useful stage because this is when those nitrates could be converted back into nitrogen gas, and therefore you're losing nitrogen compounds that could be absorbed. This only happens if there are anaerobic conditions And that would then mean that denitrifying bacteria that do this process would be able to thrive, and these ones that are all aerobic bacteria wouldn't. So this is why it's so important to keep soil aerated, so to plough it and toss the soil before you replant seeds, to make sure that there's plenty of oxygen so that you get the beneficial stages happening and not the denitrification. It's also why having waterlogged soils can be an issue, because the water displaces the air and then you'd have the denitrifying bacteria as well. Final note is a key tip for the exam, and that is making sure that you are saying nitrogen containing compound for any of these stages. We only say nitrogen when it is literally the element in the atmosphere. All of these other stages we have a nitrogen containing compound. So then we have the phosphorus cycle, and phosphorus is really important because it's used to make the phosphate group in DNA, RNA, ATP, and in a phospholipid to make the phospholipid bilayer. And this cycle is quite different to other ones because there isn't a stage where you have it as a gas in the atmosphere. And instead you mainly have it stored in phosphate ions in a mineral form in rocks. So here's the phosphorus cycle. If we start from phosphate ions in oceans and soils as a store, it's absorbed into the plant by active transport. That would then be converted into maybe DNA or phospholipid bilayer. Animals would consume that, and that's how it gets into animals. We would have some return to the oceans by decomposition of the plant and decomposition and excretion from animals. But we also have phosphate ions from waste and remains, such as bones and shells that don't get broken down as rapidly. And also guano. Guano is this bird pea, this white bird pea that you can see here, which is very rich in phosphorus. Now, those bones and shells that aren't going to be decomposed by the saprobionts will be broken down through erosion over time, and that returns the phosphate ions back to the ocean and soils. But we can also have rock formation happening. And this could be deposition or sedimentation. So those phosphate ions within the water or in the soil, over many, many years, heat and pressure, it creates those phosphorus sedimentary rocks. And to get that back into the water, again, it'd be erosion. Now, fertilizers, this is going to be something that you'd have to artificially add to get more phosphorus back into the soil. So here are our key stages. We've said the weathering of rocks, and that releases phosphate ions into soils and water. Plants absorb phosphate ions through their roots. Animals obtain phosphorus by eating plants or other animals. Excretion and death will return phosphorus to the soil or water. And decomposers, so those saprobionts, can break down dead organisms, releasing phosphate ions into the soil. Fertilizers are really, really important to replace nitrate and phosphate ions lost when either plants or animals are going to be harvested and removed from the cycle. Because if you're removing a plant or an animal from your cycle, then you're removing those mineral ions that are locked up in their cells, and also they're not there to decompose and return it back to the soil. So you always have to use fertilizers, and that can either be natural, so using manure, because we know from those two cycles that in feces there are phosphorus, phosphate ions and nitrogen containing compounds that can be broken down. Or you can use artificial fertilizers, which are inorganic chemicals, meaning no carbon, and there are pros and cons of these too. Natural fertilizers are cheaper. If you have animals on your farm already, it's free, it's just their manure. However, you don't know the exact mineral content, so you don't know how much nitrate or phosphate there is in there, and you can't control that. So artificial fertilizers, that is their main advantage. You can control the exact minerals and the proportions that you have. But you do have to pay for them, so they're going to be more expensive, and they're more harmful for the environment. And that's because inorganic substances are more soluble. Therefore, when it rains, they're going to dissolve in that rainwater, and they can then be washed away to water sources and have a negative impact on the environment. So the sorts of environmental impact, first of all, leaching, and that is when water-soluble compounds, such as the nitrates or phosphates, get washed away from the soil into rivers or ponds or lakes nearby. And if nitrogen fertilizers leach into waterways, That causes eutrophication. So eutrophication is when nitrates have leached from the fertilized field into the pond or lake, whatever water source it might be. And that causes the algae in the lake to grow much more rapidly. That creates a blanket of algae across the surface, and that blocks sunlight from reaching the lower levels in the water and any lower down plants. So if those plants lower down aren't photosynthesizing, they die. Bacteria within the water will then decompose those dead plants, but in order for bacteria to do that, they have to respire more, which means they're going to be using up oxygen. So using up all that oxygen eventually means there's not enough oxygen dissolved in the water for all of the other aquatic organisms, such as fish. And if fish don't have oxygen, they can't respire and they die as well. AQA topic 6, and we begin with 3.6.1 stimuli, and this is looking at the internal and external that can be detected and lead to a response. Starting with your definition of a stimulus. This is a detectable change in the environment, and these stimuli are detected by cells known as receptors. And an organism increases their chance of survival if they can respond to stimuli via different response mechanisms. So for plants, there is response as well. And we begin by looking at tropisms, which is the term given to when a plant responds to a stimulus and they respond via growth to that stimulus. A positive tropism means that the plant would be leaning towards or growing towards the stimuli. Negative means moving or growing away from it. And the two that you learn about are the stimulus of light and gravity. Tropisms are controlled by specific growth factors, and the one example you learn about is indole acetic acid, or IAA, and you can abbreviate to that in the exam. An IAA is a type of auxin, and therefore it can control cell elongation in the shoots and inhibit the growth of cells in the roots. It's made in the tip of the root in the shoot, but then it diffuses to other cells within the shoot and the root. So if we have a look at phototropisms first of all, photo meaning light, so this is the response to light. In the shoots, light is needed for the light-dependent reactions that you learn about in topic 5. So the plant is going to benefit by gaining more light energy and therefore leaning or bending towards the light source, the stimulus. And this is positive phototropism. So the shoot tip is where IAA is produced. IAA will diffuse to the shaded side. So if you've got light from one side over here, the IAA will diffuse to the shaded side. And that is what we call unilateral light source, and therefore we get this uneven distribution of IAA. And the cells are exposed to the IAA are going to elongate more, and as a result the shaded side cells will be longer than the light side, so it's longer and it bends over towards the light source. In the roots don't photosynthesize, they don't have chloroplasts and chlorophyll, so they don't require light energy, so there's no advantage of growing towards the light source. And instead there's actually an advantage to grow away, because that means the roots are anchoring the plant deeper into the soil. And in the roots, if cells are exposed to a high concentration of IAA, that inhibits cell elongation. So you still have IAA being produced in the tips of the root, but the IAA diffuses to the lower side, so the direction where you would have gravity, the lower side, and that inhibits the elongation of the cells on the lower side of the root. The ones on the top side are still going to grow, and therefore that top side is longer and it bends downwards away from the light source and towards gravity. But this would be our negative phototropism because it's away from the light source. With regards to gravitropism, in the shoots, again, IAA is produced in the tip of the shoot, and IAA will diffuse from the upper side to the lower side of a shoot. And if a plant is vertical, not lying down like we can see in this picture, this causes the plant cells to elongate and the plant grows upwards because the IAA would be moving down towards the bottom, and those bottom cells will grow more, and it just makes the shoot grow upwards. If a plant is on one side though, like we can see in this picture, the side that is exposed to the auxin will elongate, and that can cause it to grow upwards. And that would be negative gravitropism. It's growing away from the direction of gravity. In the roots, it's the opposite. The IAA will diffuse to the lower side of the root so that the upper side elongates and the lower side doesn't. So the root bends downwards towards gravity, which is a positive gravitropism. Next then, the other simple responses are taxis and kinesis, which keep organisms within their favourable conditions of their environment. For example, light intensity, moisture levels, and chemicals. Taxis, this is when an organism will move its entire body towards a favourable stimulus or away from an unfavourable stimulus. So this is directional. When an organism moves towards a stimulus, this is known as positive taxis, and when it moves away, it's negative taxis. Kinesis, then, this one is not directional, but it's a change in the speed of movement and the rate it changes direction. If an organism moves from an area where there are beneficial stimuli to an area where there are more harmful stimuli, its kinesis response will be to increase the rate it changes direction, and therefore it's more likely to locate and return to the favourable conditions again. However, if it tries that and the organism is completely surrounded by negative stimuli, so no matter how many times it changes direction, it can't find favourable conditions, then the rate of turning actually decreases to keep it moving in a relatively straight line to increase the chances of it finding a new location with favourable conditions. And just linked to this, most of the responses will be that they'll want an area where it's dark, and that would be to not be easily seen by predators or to stop them from drying out. Moisture, they'll want a more moist area, again, to stop them from drying out. Chemicals, they might be moving towards certain chemicals because that might indicate the presence of food. Simple reflexes are something that we have in more complex organisms with a nervous system, and this would involve a receptor detecting a stimulus. That would then result in coordination occurring with the coordinator, then a response carried out by an effector. And the nervous system is made up of the peripheral and the central nervous system. The peripheral, abbreviated here to P, nervous system, NS, is the receptors, sensory and motor neurons. The CNS, or the central nervous system, that's the coordination centres, so the brain and the spinal cord. So here we have a simple reflex, and a reflex is a rapid automatic response to a stimulus. Reflexes are involuntary, so they do not require conscious thought. You don't have to learn to respond in that way to protect yourself. And the point of a reflex is it protects against damage to body tissues, and it could also help you to escape from a predator. There are 3 neurons in a reflex arc, so it's very, very simple. You have the sensory neuron, which is going to be connected to the receptor. That detects a stimulus and sends an impulse to the spinal cord. The relay neuron is going to be located in the central nervous system, which will be the spinal cord for a reflex. And then the motor neuron that carries the impulse to an effector to carry out the response. And the response in terms of a reflex reaction is very fast because the impulse bypasses the conscious part of the brain and it travels through the reflex arc. And as we can see, the reflex arc only contains 3 neurons and therefore 2 gaps, 2 synapses. And because we've only got 2 synapses, that means it's faster because it's slower transmission at synapse because it's diffusion of a neurotransmitter. Now, actually, in this image, you can't see the relay neuron, but there would be a sensory neuron, relay neuron here in the spinal cord, and then the motor neuron. Receptors detect a stimuli, as we've said, but there are specific receptors that you need to learn about. And each receptor responds to a different stimulus, and this stimulus of a receptor leads to the establishment of a generator potential, which can then cause a response. And we'll come to the idea of establishing a generator potential and an action potential shortly. So the 3 receptors that you need to know are the Pacinian corpuscle, rod cells, and cone cells, both of which are in the retina in the eye. So your Pacinian or Pacinian corpuscle, this receptor responds to pressure changes. They are the receptors that you find lots of deep in the skin, mainly in the fingertips and in feet. And it consists of a single sensory neuron wrapped in layers of lamellae separated by gel. The sensory neuron in the Pacinian corpuscle has a special channel protein, or has special channel proteins in its plasma membrane. So here's our structure we can see. And those special channel proteins are stretch-mediated sodium ion channels. And these will open and therefore allow sodium ions to diffuse into the sensory neuron when those channels are stretched and deformed. And when pressure is applied to a fingertip or feet, that is applying pressure to the skin, and these layers of lamellae get pushed and deformed, and that stretches and deforms the shape of the sodium ion channels so that they open and sodium ions diffuse in. So when pressure is applied, it causes these sodium ions to diffuse in, and that will then result in the establishment of a generator potential. More on that very soon. Next, then we look at the rod and cone cells, which are the two photoreceptors, or light receptors in other words, in the human retina. Rod cells process the images in black and white. To create that generator potential, the pigment inside of these rod cells, known as rhodopsin, has to be broken down, and that is done by light energy. They can detect light of very, very low light intensities, as many rod cells connect to one sensory neuron. In this case, we can see it's called a bipolar cell before it gets that sensory neuron. And this is known as retinal convergence because we have multiple rod cells converging or meeting at one bipolar cell. And this means that collectively they are able to trigger that action potential, but because you have got multiple rod cells feeding to one bipolar cell, it means the brain cannot distinguish between the separate sources of where that light energy came from. So that's known as low visual acuity, which means you don't have that sharp, clear, crisp vision. So when it's very, very dark, you can still see, but you only see in black and white, and it's blurry. So next time it's nighttime, have a look and you'll notice you pretty much only see in black and white, and you don't have that clear, crisp vision, meaning low visual acuity, and that is why the retinal convergence. Cone cells process images in colour. There are three types that contain different types of pigment known as iodopsin that you get in the cone cells. You have red iodopsin, green and blue iodopsin, which all absorb different wavelengths of light. Now, iodopsin found in these cone cells is only broken down in high light intensities. So you do need a high light intensity to be able to see in colour vision. You also need to have a high light intensity to see in colour vision because you have one cone cell connecting to one bipolar cell. And therefore, you need to have a high enough stimulus to trigger that action potential. So when it is much brighter light intensities, you can see colour, and you have a high visual acuity because you've got one cone cell connected to one bipolar cell, so the brain can distinguish between separate sources of light. Now, the distribution of the rods and cone cells is uneven in the retina of the eye. And here is the fovea. That is where the highest light intensity hits the retina because of where the light is focused through the lens. So at the fovea, you have most cone cells. We can see here that's where the fovea is. You have lots of cone cells there. Whereas the rod cells, because they still work at lower light intensity, those are mainly located further away from the fovea because they'd still work in that location. Next, then we move on to the control of the heart, and cardiac muscle is myogenic, and that means it contracts on its own accord without any input from the nervous system, but the rate of contraction is controlled by the nervous system and also electricity. So that's what we're going to have a look at. The sinal atrial node, or SAN, is located in the right atrium, and that is known as the natural pacemaker. The AVN, or atrioventricular node, is located near the border of the right and left ventricle. It is still within the atrium though, just here. You have a bundle of His which runs through the septum, and then the Purkinje fibres branch into the walls of the ventricles. So these two are nodes that will emit the electrical activity. The bundle of His and Purkinje fibres are able to transmit and conduct that electrical activity through the myogenic muscle. So the order that this happens then, the SAN releases a wave of depolarization, which means electricity, across the atria. The AVN will then release another wave of depolarization when that first wave of depolarization reaches it. However, there is a layer of non-conductive tissue between the atria and the ventricles, and this prevents that wave of depolarization travelling straight down through the cardiac muscle in the ventricles. That means instead the wave of electricity has to go through the bundle of His and then the Purkinje fibres. As a result, that means the ventricles actually contract at the apex, which is this very tip here, and then the walls of the ventricles contract after a short delay. And the advantage of this is the atria would have fully emptied the blood before the ventricles contract, and also the ventricles contract from the bottom upwards, so you've got a more efficient pumping of that blood out of the heart. Now, the control of that heart rate is where we see the nervous system taking its role. So the medulla in the brain controls the heart rate via the autonomic nervous system. Autonomic meaning it's automatic, there's no conscious thought behind it. So there's two parts. We have a centre link to the sinoatrial node, so the SAN, to increase the heart rate via the sympathetic nervous system. And then there's also another that decreases heart rate via the parasympathetic nervous system. And that's what we can see here. We've got the medulla, and then we've got these two. We've got the parasympathetic nervous system and the sympathetic nervous system. So the heart rate changes in response to pH and blood pressure. And these stimuli are detected by receptors. Chemoreceptors detect changes in pH, and we have pressure receptors, also known as baroreceptors, detect changes in blood pressure. And both of these receptors are found in the aorta and the carotid artery, which is just after the aorta. So the response to pH, the pH of the blood will decrease, meaning more acidic, during times of high respiratory rate or respiration, because if it's aerobic respiration, there'll be lots of carbon dioxide produced, which will dissolve to make carbonic acid, or if it's anaerobic respiration, you're producing lactic acid. Either way, this excess acid needs to be removed from the blood rapidly, because if it's really acidic, it could denature enzymes and proteins as well, such as hemoglobin. So this is achieved by increasing the heart rate so that carbon dioxide will get to the alveoli, diffuse out via gas exchange more rapidly. So that means there needs to be more impulses via the sympathetic nervous system to the SAN to make that natural pacemaker, the SAN, fire more rapidly. The response to pressure is, if the blood pressure is too high, this can cause damage to the walls of the arteries, and it's important that mechanisms are put in place to bring down that blood pressure to prevent that damage. And that results in more impulses via the parasympathetic nervous system to decrease the SAN firing, so the heart rate decreases. Now, if the blood pressure is too low, there may be insufficient supply of oxygenated blood to respiring cells. So in that case, we'd have more impulses via the sympathetic nervous system to increase the heart rate to bring the blood pressure up. So here is the process. The stimulus in this case we've got is an increase in carbon dioxide, meaning lower pH, and an increase in hydrogen ions. Chemoreceptors in the walls of the aorta and the carotid artery will detect that. Then the coordination is more electrical impulses are sent to the medulla, which is the cardiac centre, and that will then result in more impulses sent via the sympathetic nervous system to the SAN to increase the frequency of electrical impulses. The effector is the cardiac muscle, so the SAN tissue, and the response is an increased heart rate because the SAN will be firing more rapidly. Now, for high blood pressure, the stimulus is increased blood pressure. The receptor would be the pressure receptors in the wall of the aorta and the carotid artery, and these stretch when there's high pressure. Coordination would be more electrical impulses sent to the medulla, and then more impulses sent via the parasympathetic nervous system so that the SAM will fire less frequently. The effector is the cardiac muscle, that's in tissues, response, reduced heart rate, and that lowers the blood pressure. Next is 3.6.2, nervous coordination. And here we have a motor neuron, which is actually the only neuron that you need to know the structure of. So we've got the cell body of the neuron, and that contains organelles that you'd find in typical animal cells. So the ribosome, nucleus, mitochondria, for example. And proteins and neurotransmitter chemicals are made there. Then we've got the dendrites, which carry action potentials to surrounding cells. The axon is, we can't see it because it's actually surrounded by the Schwann cell, but it's the long, thin, wire-like structure. And it's a conductive long fibre that carries a nervous impulse along the motor neuron. Schwann cells, so these are these Schwann cells here, and they wrap around the axon to form the myelin sheath, which is a lipid and therefore does not allow charged ions to pass through it. There are gaps in these along the axon, and those gaps are known as the nodes of Ranvier. So we then move on to the concept of an action potential and resting potentials. Resting potentials are when a neuron is not conducting an impulse. So there's not been a stimulus detected, it's just your resting standard state. But even when at rest, there is a difference between the electrical charge inside the neuron, specifically we tend to talk about the axon, and the outside of the axon. And that difference in voltage is our resting potential. So there are more positive ions, so sodium ions and potassium ions, outside compared to inside, and therefore the inside of the neuron is comparatively more negative at minus 70 millivolts, and you do need to know that value. So this is quite a common 2 or 3 mark question to explain how that resting potential is established or maintained. So first of all, there is a sodium potassium ion pump, which involves active transport and therefore ATP. So that's what we can see here in the membrane of the axon. So this co-transporter pump, we have 2 potassium ions being pumped in and 3 sodium ions being pumped out by active transport. That creates an electrochemical gradient, so we end up with more potassium ions inside of the cell and more sodium ions outside of the cell. Because we have this electrochemical gradient, those ions can then move down their concentration gradient. But the membrane is more permeable to potassium ions because there are more of these potassium ion channels, and also there's some that are permanently open, whereas the sodium ion channels, there are fewer of them and some aren't always open. So although you have sodium ions diffusing in, you have far more potassium ions diffusing out, and that's why it's more positive outside than it is inside the axon, and you get this minus 70 millivolts. Just point out that is the minus there. That minus has just gone onto a different row. Now, an action potential is when there is a response to a stimulus, and you only get this response to the stimulus if the stimulus is big enough to increase the neuron's voltage beyond a set point, a threshold. So an increase in voltage or depolarization is due to the neuron membrane becoming more permeable to sodium ions, and therefore it becomes more positive. And once an action potential is generated, it will happen initially at this point, and then it moves along the axon from node to node of Ranvier like a Mexican wave. And we'll come back to that concept once we go through the action potential. So here we can see we've got our resting potential at minus 70 millivolts, but then we have a stimulus. And the stimulus provides energy that can cause the sodium voltage-gated ion channels, so those voltage-gated sodium ion channels, to open. And if those open, we then start to get sodium ions diffusing into the axon, which increases the positivity inside of the axon. So we can see that it is becoming more positive. When you have the threshold potential reached of minus 55 millivolts, that then causes even more sodium ion channels, but they're voltage gated sodium ion channels, to open. So we get even more depolarization because more sodium ions diffuse in, up to a maximum of positive 40 millivolts. And at that point, when we get to positive 40 millivolts, that causes the voltage gated sodium ion channels to close, and we still have potassium ion channels open. So we've got potassium ion channels permanently open, so you constantly have positive potassium ions moving out, but now at this point we no longer have positive sodium ions moving in. And that's when repolarization occurs, so it starts to become more negative. And we get even more potassium ion channels opening, so even more positive ions are now moving out and none are moving in. And that's when we get this overshoot, this hyperpolarization going below the resting potential to at least minus 80 millivolts. And this is then when we'd have that resting potential being re-established through active transport with the sodium ion potassium ion co-transporter pump to bring it back to its resting state. So a stimulus has to be large enough to cause the initial voltage gated sodium ion channels to open to reach the threshold. And once that threshold is reached, an action potential will always occur. But if it's a very small stimulus and that isn't enough to open the voltage gated sodium ion channel, then you don't reach the threshold, and it's known as this failed initiation. You don't get an action potential and it goes straight back to resting. Now that is a good thing because we don't want to respond to every single slight change in our environment because we just overwhelm our senses. We only want to respond to those stimuli that are large enough, that are worthy to respond to, so that it could potentially cause danger and therefore it is worth us responding to it. Now the refractory period is just labelled here, but you actually have the refractory period happening through all of this point when you've reached that action potential, and we'll come back to the refractory period in a minute. But just showing you this concept of the Mexican wave. So that action potential that we just saw here, this curve, will happen at all of these points along the axon at the nodes of Ranvier. So it's like a Mexican wave because we have this increase and that triggers then an increase here, and that triggers it here, here and here. So each time you reach that threshold, that can then trigger an action potential at the next node of Ranvier. So the all or nothing principle, if the depolarization does not exceed the minus 55 millivolt threshold, an action potential does not occur. So we previously talked about that, and that's the nothing part of the principle. Any stimulus that does trigger depolarization to at least that minus 55 millivolt threshold will always trigger an action potential, and it will always peak at the same maximum. Bigger stimuli does not increase the voltage any further. Instead, it increases the frequency at which the action potentials are generated. So that's the all or nothing principle, and it's important because it makes sure that animals only respond to these large enough stimuli rather than responding to every slight change in our environment. The refractory period, so after the action potential has been generated, the membrane enters this refractory period, which is when it cannot be stimulated to go through another action potential. And it's because the sodium ion channels are recovering and can't be opened. So that should say ions there, sodium ion channels. Now this is important because it ensures that discrete or separate impulses are produced. An action potential cannot be generated immediately afterwards, so it has to be separate, and that makes it easier to process the stimulus. It also ensures that the action potential only travels forwards because it can't go backwards because those sodium ion channels are recovering. So it moves forwards, constantly moving its way around the reflex arc or the nervous pathway. And also it limits the number of impulses, and this is important to prevent overreaction to a stimulus. The factors affecting the speed of conductance are myelination and saltatory conduction, axon diameter, and temperature. Myelination and saltatory conduction is linked to the importance of these Schwann cells, which create the myelin sheath. So Schwann cells wrap around the axon to form that myelin sheath, which does not allow charged ions or the impulse to pass through it. So that leaves you with gaps where action potentials can be generated, known as the nodes of Ranvier. And as a result, the action potential jumps from node to node, and that is called saltatory conduction. And this is faster because that means you only have to generate action potentials at these nodes of Ranvier rather than every single position along the axon, and that is why it travels faster. A wider diameter will increase the conductance, and that's because it offers less resistance to the flow of ions inside of the axon, so depolarization spreads more quickly. A higher temperature increases the speed of conduction for two reasons. That conductance will be faster because the ions diffuse faster because they've got more kinetic energy. And also the enzymes involved in respiration will be working faster. So that means there's more ATP for active transport of that sodium ion, potassium ion pump. Synapse is the gaps between the end of the axon of one neuron and the dendrite of another. And that is where the action potential is then transmitted as neurotransmitter that diffuses across the synapse. This here is the whole process of what happens at a synapse. This is worth pausing, screenshot, and you can turn it into a flow diagram, mind map, because this would be a long answer question. So the action potential arrives at the synaptic knob. Depolarization of the synaptic knob causes calcium ion channels to open, and therefore calcium ions diffuse into the synaptic knob. That causes vesicles that contain the neurotransmitter to move towards and fuse with the presynaptic membrane, and that releases the neurotransmitter into the synaptic cleft. That neurotransmitter diffuses down its concentration gradient across the synaptic cleft, and it will bind to receptors on the postsynaptic membrane. When it binds to the receptors on the postsynaptic membrane, that causes sodium ion channels to open. Therefore, sodium ions will diffuse into that postsynaptic membrane of the neuron. And if enough neurotransmitter is bound to cause enough sodium ions to diffuse in to reach that minus 55 millivolts threshold, then it's going to generate that action potential. Lastly, that neurotransmitter needs to be broken down so that it is then released from the receptor to cause the sodium ion channels to close. Because otherwise, even after the stimulus has stopped, that will still be triggering an action potential in the postsynaptic membrane, which we don't want to happen. So that is then going to happen because of enzyme. Enzymes will degrade the neurotransmitter, causing them to be released. Therefore, the sodium ion channels close and it goes back into the resting potential stage. And the neurotransmitter that's been broken down gets recycled and reused. Now, the bits that are in bold and yellow are the parts that I've seen repeatedly in mark schemes that you have to say those terms, and sometimes they're even underlined that you have to use literally that language. So make sure you're really specific with how you word this. A cholinergic synapse is a particular synapse with the neurotransmitter acetylcholine. So it's exactly the process we just went through, except you need to say acetylcholine whenever you would have said neurotransmitter. And you need to know that the enzyme that breaks down the acetylcholine is acetylcholine esterase. And that will break down or hydrolyze the acetylcholine into acetate and choline, which then gets recycled. Summation is what happens at a synapse, and it's the rapid buildup of neurotransmitter in the synapse to help generate an action potential, because we have to have enough neurotransmitter to cause enough sodium ion channels to open for enough sodium ions to diffuse in to trigger and generate that action potential. So the two methods are spatial or temporal summation. Spatial summation is when you have many different presynaptic neurons converging at one postsynaptic neuron. So collectively, these multiple presynaptic neurons release so much neurotransmitter that it should result in the generation of an action potential. Temporal summation is when you just have one presynaptic neuron connecting to one postsynaptic neuron. And in this case, it is the repeated release of neurotransmitter from that one presynaptic neuron over a short period of time that should add up to enough neurotransmitter to cause enough sodium ion channels to open to reach that threshold potential in the postsynaptic neuron. Now, the movement at a synapse is unidirectional. The impulse only travels in one direction. And there are a few reasons why that's the case. Number one is the neurotransmitter is only released by the presynaptic neuron, and therefore you get this concentration gradient. So it can then only diffuse from that location to where there's a lower concentration at the postsynaptic neuron. And secondly, you only have the receptors that the neurotransmitter binds to on the postsynaptic neuron's membrane. You can also get inhibitory synapses, which will prevent an action potential being generated. And these can cause chloride ions to diffuse in instead of or in addition to sodium ion channels. And as a result, because you've got chloride, which is Cl-, you have negative ions moving in, and that makes that postsynaptic membrane potential even more negative. It could go down to minus 80 millivolts going into hyperpolarization. That makes an action potential highly unlikely because you need even more sodium ions to diffuse in before you'd reach that minus 55 millivolt threshold. A neuromuscular junction is very similar to a synapse, except instead of it being the gap between two synapses, it's actually the end of your nervous pathway where you have a gap between your motor neuron and your effector, which is a muscle. And striated muscle will contract when it receives an impulse from that motor neuron through that neuromuscular junction, and it works in a very similar way to synapses. So first of all, an action potential reaches that presynaptic membrane of the motor neuron, causing calcium ion channels to open, calcium ions diffuse in. That causes the vesicles that contain the neurotransmitter to move towards and fuse with the presynaptic membrane, releasing the neurotransmitter, which is acetylcholine, that diffuses across this gap, and then it will bind to receptors on the postsynaptic membrane. But the postsynaptic membrane here is the sarcolemma, which is the membrane of the muscle fibers. The binding causes sodium ion channels to open in that sarcolemma and sodium ions diffuse in. So it's pretty much identical, except we've got not a postsynaptic neuron, we've got muscle fiber, so that receptors are on the sarcolemma, and it's always going to be acetylcholine as the neurotransmitter. So that depolarizes the membrane of the muscle fiber and creates an action potential in that muscle that moves along these T tubules towards the center of the muscle fiber. And those action potentials prompt the opening of voltage-gated calcium ion channels in the membranes of the sarcoplasmic reticulum, situated near these T tubules. And as a result, calcium ions diffuse out of the sarcoplasmic reticulum around the myofibrils. And that is what causes muscle contraction via the sliding filament theory. So here's just a summary table comparing the neuromuscular junction to the cholinergic synapse. So you can screenshot that in case you did get a long answer, compare and contrast question for those. Now this flows into what we were just talking about, muscle contraction, 3.6.3, skeletal muscles and looking at how they can contract. So we talked about the myofibril. Here are the structures that you need to be aware of. Again, this could be a good one to screenshot, turn into flashcards for each of these structures. The myofibril, these are long cylindrical structures found inside muscle fibers. made up of repeating sarcomere units, which we're going to have a look at. Because the sarcomere is the functional unit. It's defined as a region between two Z lines, and you have actin and myosin where you can have your sliding filament theory occurring. The sarcolemma, that's the plasma membrane of a muscle fibre. It conducts electrical impulses, action potentials, and it triggers a contraction to help maintain ion balance within the cell as well. Sarcoplasmic reticulum, that's a specialised form of smooth endoplasmic reticulum that surrounds myofibrils, and it stores and releases calcium ions, which are essential for triggering muscle contraction. The sarcoplasm, that is the cytoplasm of a muscle fibre. So it contains the organelles, enzymes, myoglobin needed for energy release and muscle function, and it also has glycogen stores for ATP synthesis. So looking at the sarcomere in a bit more detail, this is what we said on the previous slide. So this is one long cylinder, which is the myofibril, made up of these repeating units called sarcomeres, which are identified by two Z lines. There are other structures within the sarcomere that you need to be able to name. So here are the Z lines, which indicate the start and end of one sarcomere. The A band is the myosin. So that is the distance of myosin, which is shown in blue here. An I band is the distance where you have actin not overlapped at any point with the myosin. So just red lines here, which are our actin protein. We have an M line, which is the center of where the myosin is attached. We have an H zone, which is where you just have myosin without any actin overlapping it. So those are your key structures, all to do with basically whether you have actin by itself, myosin by itself, and where it's overlapping. And the kinds of questions you might get is, what happens to the size of those sections when a muscle contracts? And when a muscle contracts, the actin slides inwards over the myosin, which we can see here. So as a result, the H zone decreases in length because we now have less myosin by itself. The I band will also decrease in length because we have less actin by itself because it's slid over. The A band never changes because that is the length of the myosin. Myosin doesn't change in length. The M line will always stay in the same position as well, but the Z lines move closer together. So that's something you need to know as well, those different bands and what happens to them when a muscle contracts. The sliding filament theory explains why you have the actin sliding over the myosin. So when that action potential reaches a muscle fibre, like we saw in the neuromuscular junction, it triggers the release of calcium ions from the sarcoplasmic reticulum into the sarcoplasm. And those calcium ions cause tropomyosin, which is a protein that is normally blocking the binding sites on actin, to move, and it exposes the binding sites for myosin on actin. Now, because of that, we get this cross-bridge formation. So with ADP and PI still attached to our myosin head, which is a structure branching off myosin, that can now bind to the myosin binding sites on actin. And when that happens, it creates what's known as an actino-myosin cross-bridge. The power stroke, which is when the actin filament is moved, once that cross bridge is formed, the myosin head pivots, so it bends here, and that pulls the actin filament inwards, so it slides it in. And that movement is known as the power stroke, and when that happens, it causes the release of ADP and PI. It also generates tension, and that is causing the actin filament to slide. So we then get this ATP molecule binding, and that new ATP molecule, when it binds to the myosin head, it causes it to detach from the actin filament. And this ensures that the myosin head doesn't remain permanently attached to the actin. That ATP then gets hydrolyzed, and it resets the myosin head. So within the sarcoplasm, there is the enzyme ATPase, and that is activated by calcium ions. That will hydrolyze the ATP back into ADP and PI, and the energy released from that ATP hydrolysis repositions the myosin head back to its original state, ready to form a new cross bridge and therefore go through that process again to bring the actin even closer for a full muscle contraction. So as long as calcium ion levels remain high, the cycle repeats, continuously pulling the actin filaments towards the center of the sarcomere, shortening the muscle fiber and causing contraction. When the nervous system stimulation stops, calcium ions are then actively transported back into the sarcoplasmic reticulum, and that allows the tropomyosin to move back to block the myosin binding sites on actin to prevent any further contraction, and the muscle will relax. So some common questions. What's the role of ATP and phosphocreatine? Here are your standard answers if this came up. So the ATP is hydrolyzed and the energy will then break the actinomyosin bridges, or it's to move or bend the myosin head back to its original state, and for the active transport of those calcium ions. So if ATP levels are low, muscle contraction can't happen effectively. And that is where phosphocreatine plays a role, because this acts as a rapid source of ATP in muscle cells by donating a phosphate group to ADP to be able to make ATP, so that you do have ATP during short bursts of intense activity. You could also be asked what's the role of glycogen granules in skeletal muscles. So it's a store of glucose that can be hydrolyzed into glucose, and that glucose can be used as a respiratory substrate to provide the ATP needed. The role of calcium ions then, so when an action potential reaches the muscle fibre, calcium ions are released from the sarcoplasmic reticulum and diffuse into the myofibrils. Those calcium ions cause tropomyosin to move and expose the binding sites on the actin filament. And with these sites now accessible, the myosin heads attach to actin, forming the actinomyosin cross bridge. The last bit of this topic is knowing the difference between slow twitch muscle fibres and fast twitch muscle fibres. So knowing the difference between the structure, location, general properties, the type of respiration that happens, ATP production and energy storage. So I recommend screenshot this page, or if you've downloaded my free booklet, fill it in so you've got this for your revision. So in terms of structures, slow twitch, large store of myoglobin, rich blood supply with many mitochondria, compared to fast twitch, thicker with more myosin filaments, large store of glycogen and a store of phosphocreatine. Location, you get a lot of slow twitch in calf muscles, whereas fast twitch might be places like biceps. General properties, slow twitch contract slower, and they can sustain aerobic respiration for longer periods. So these muscles are used in endurance activities like marathons. Fast twitch contract faster and provide short bursts of powerful contraction for things like sprinting or weightlifting. Slow twitch will mainly be aerobic, fast twitch is anaerobic, and in terms of ATP production, the slow twitch produce ATP slowly, but they'll generate more, and that's because it's aerobic. And because fast twitch are anaerobic, they produce ATP quickly, but less. Lastly, in terms of the glycogen store, they both have stores of glycogen, but slow twitch will have a lower store of glycogen, and you'll have a large store in the fast twitch. Next then is 3.6.4, homeostasis. Homeostasis involves physiological control systems that maintain the internal environment within restricted limits. And it's important in maintaining things like a stable body temperature, stable blood pH for enzyme activity. Negative feedback is involved, and this restores systems to their original level. And the possession of having separate mechanisms, so one that can bring levels back up, one that can bring it back down, give better control to get it back to the original state that it should be at. So for body temperature, if it's too low, there'd be insufficient kinetic energy for enzyme-controlled reactions. If it was too high, enzymes would denature. Either way, metabolic reactions would stop or slow and cells would die. If you have too high or too low a blood pH, it will result in enzymes denaturing, fewer enzyme substrate complexes, and again, that are going to affect metabolism. Blood glucose control. So glucose is needed for respiration. So a lack of glucose in the blood could result in cell death. And if blood glucose levels are too high, then this lowers the water potential of the blood, and water then leaves the surrounding cells by osmosis, and that prevents the normal function of those cells because now they're low on water. And also it increases the blood pressure because there's more liquid in the blood. The water potential of the blood then, if it's too high, water moves into cells by osmosis, and that could cause them to burst. And if the water potential is too negative, the water leaves the surrounding cells into the blood, and that can cause the cells to crenate. So you need to know in detail blood glucose concentration. And that is going to increase after you've eaten or drunk something containing carbohydrates, or it will decrease if you haven't eaten something containing carbohydrates or following exercise, because respiration rates would be higher using up your carbohydrates. Here are the different things involved. So the pancreas, that is the organ that detects changes in blood glucose levels. And specifically, you have the islets of Langerhans cells that are going to detect and release insulin and glucagon to bring blood glucose levels back to normal. Insulin is a protein hormone released when blood glucose levels are too high, so it causes a decrease in blood glucose levels. Glucagon is also a protein hormone, and it's released when blood glucose levels are too low, and that will cause an increase in blood glucose levels. Now, adrenaline, this is released by the adrenal glands when your body anticipates danger. So it's not to do with homeostasis, but it has a very similar effect to glucagon because it causes glycogen to be hydrolyzed, so you increase the blood glucose levels. So some key terms that you need to know for this topic are glycogenesis, glycogenolysis, gluconeogenesis. So learn those terms, get them on a flashcard. Wherever you see glyco, that means it's about glycogen. So these top two are about glycogen. Where you see gluco, that means it's about glucose. Genesis means to make, lysis means to break. So glycogenesis means making glycogen. So it's the process when excess glucose is converted into glycogen. And that happens in the liver quite a lot and muscle cells. Glycogenolysis, so glycogen lysis, is the hydrolysis of glycogen into glucose. Gluconeogenesis. Gluco means glucose, genesis means to make, neo means new. So it's making glucose from something new. And by that we mean a non-carbohydrate source. So for example, from amino acids, or it could be from glycerol from lipids. So your normal blood glucose levels might increase if you've just eaten a carbohydrate-rich meal. That is detected by the beta cells in the islets of Langerhans in the pancreas. The beta cells will then release insulin. That causes liver cells to become more permeable to glucose, so more glucose is taken from the blood into the liver cells, and then that glucose is converted into insoluble stores of glycogen. That means that we've removed glucose from the blood and brought our blood glucose levels back to normal. Now, maybe you haven't eaten anything containing carbohydrates for a while, or you've done high levels of exercise, so lots of respiration using up the blood glucose levels, so they've decreased. A decrease is detected by the alpha cells in the islets of Langerhans of the pancreas. In response, the alpha cells release glucagon. That triggers the second messenger model, which activates enzymes to hydrolyze glycogen in the liver back into glucose, which is then released into the blood to bring it back to normal. But we're going to go through that in a bit more detail. So here's our action of insulin. We said that's the hormone released by the beta cells in the islets of Langerhans when blood glucose levels are too high. And insulin will then be transported in the blood. When it gets to the liver cells, it will bind to complementary shaped receptors on the cell surface membrane of those liver cells, also muscle cells. When it binds, it increases the permeability of that membrane to glucose by stimulating the insertion of more glucose channel proteins, GLUT4, into the membrane. And that allows more glucose to move from the blood into the liver cells by facilitated diffusion. Now, once that happens, the glucose has to be converted to glycogen because that is insoluble and therefore it's not going to lower the water potential of the cell like glucose would, and therefore it won't affect osmosis. So these actions help to reduce the blood glucose levels back to normal via negative feedback. And this is just showing us here when the insulin binds to complementary shaped insulin receptors embedded within the membrane of the target cell, the liver cell. That causes a cascade of chemical reactions, which then causes vesicles, which contain the glucose channel protein, to move up and fuse with the cell surface membrane, and it embeds more of those channel proteins, so more glucose can then move from the blood into the cell. The action of glucagon then, the alpha cells in the islets of Langerhans detect the blood glucose level is too low and secrete glucagon, which will travel around in the blood, and when it gets to the complementary shaped receptors on the cell surface membrane of liver cells, it activates a second messenger model, which is described here. So it causes a protein to be activated into adenylate cyclase. That is converting ATP into cyclic or cyclic AMP. Cyclic AMP activates another enzyme, protein kinase, and that's the one that hydrolyzes glycogen into glucose to release it back into the blood. Now, if your glycogen stores are used up, then you can also have the activation of enzymes that can convert glycerol and amino acids into glucose to be used as a respiratory substrate instead. So here's our second messenger model. The first messenger is glucagon. That hormone binds and it activates enzyme number 1, which is adenylate cyclase. Now, adenylate cyclase is going to convert ATP into cyclic AMP, which is our second messenger. And that will then activate the protein kinase, which can hydrolyze glycogen into glucose. Now, adrenaline also goes through that same second messenger model, so you get that same process happening. Diabetes, you have type 1 and type 2 diabetes. Type 1 is when you're unable to produce insulin, usually starts in childhood, and it could be a result of an autoimmune disease where the beta cells are attacked. So the treatment would be injections or some way of getting insulin into the blood. Type 2 diabetes is when the receptors on the target cells lose their responsiveness to insulin. So insulin is still made, it's just the receptors don't respond as they should. And this usually develops in adults because of obesity and poor diet. It can be controlled by regulating the intake of carbohydrates, increasing exercise to use up that glucose for respiration, and sometimes insulin injections are still needed as well. Osmoregulation. So this is now looking at the regulation of the water potential of the blood. And blood with too low a water potential is described as hypertonic. And that might be if you've been sweating lots, not drunk enough, or there's lots of ions in your diet. Hypotonic is if the blood has too high a water potential, and maybe you've drunk too much water or you don't have enough salt in your diet. The corrective mechanisms would be, if you've got too low a water potential, then more water is reabsorbed by osmosis into the blood from the tubules of the nephrons in the kidneys. So that would mean you make urine, which is more concentrated and lower volumes. If you've got too much water in your blood, so too high a water potential, then less water is going to be reabsorbed by osmosis back into the blood from the nephrons. So you end up with larger volumes of dilute urine. So let's have a look at this nephron then. Osmoregulation occurs within the nephrons in the kidneys, and nephrons are these long tubules surrounded by capillaries, and there are around 1 million per kidney. The blood gets filtered here to remove waste and selectively reabsorb useful substances back into the blood. So the key stages, which we will go through each in more detail, we have ultrafiltration first, and that occurs due to high hydrostatic pressure. That will force out from the blood water and small molecules like glucose. And that gets forced from the glomerulus capillaries into this renal capsule. Then in the proximal convoluted tubule, we have selective reabsorption occurring. And that is where we're going to have all the glucose and some water being reabsorbed. Then we've got the loop of Henle, and this maintains a sodium ion gradient, so water can be reabsorbed into the blood. And water then will continue to move out of the distal convoluted tubule and collecting duct by osmosis, and the collecting duct carries the remaining liquid, which is the urine, to the ureter. So the structure of the nephron then, the formation of the glomerular ultrafiltration, that occurs here in the renal or Bowman's capsule. And there is a high hydrostatic pressure in these capillaries, the glomerulus, and as we said, that forces out water, glucose, ions and urea from the blood into that capsule. Large molecules like proteins and red blood cells remain in the blood. So here we have what causes that high hydrostatic pressure first of all. As the blood enters the glomerulus via the afferent arteriole, which is a wider blood vessel, the difference in that size creates a high pressure inside the glomerular capillaries, and it's the high hydrostatic pressure that forces out all of those small solutes and the water. Now you do have 3 filtration barriers. We first of all have the capillary endothelium. So the glomerular capillaries have fenestrations, which are these tiny gaps between the endothelial cells. And that will allow things like plasma and dissolved substances to pass through, but it blocks the red blood cells. So by plasma, I mean it'd be like the water, urea, and glucose amino acids, but blood cells can't fit through there. The next layer is the basement membrane. And this is a gel-like protein-rich layer between the capillary and the Bowman's capsule. It acts as the main filtration barrier and it prevents the passage of the large proteins. So small molecules can pass, but large proteins cannot. And then finally, we have podocytes. And podocytes have foot-like extensions that wrap around the capillaries that create these gaps, these filtration slits, to further ensure only small molecules can enter. So that's how we have the filtration. Then we have the selective reabsorption. So all glucose is reabsorbed by active and co-transport, and most water is reabsorbed by osmosis at this stage. The proximal convoluted tubule, which it says PCT here, do not abbreviate that in the exam. It is not an accepted abbreviation. The proximal convoluted tubule has cells have many microvilli and mitochondria to maximize this active transport and co-transport. So those cells lining the proximal convoluted tubule actively transport sodium ions out of the epithelial cells into the blood via sodium-potassium pump. That creates a lower concentration of sodium ions in the proximal convoluted tubule compared to the lumen. And therefore sodium ions diffuse into the proximal convoluted tubule down their concentration gradient, but that has to be via a co-transporter protein which carries glucose with it as well. So that's how we get the glucose inside of the proximal convoluted tubule, epithelial cells. And then once inside there, the glucose moves by facilitated diffusion into the blood, and that is how all of the glucose re-enters the bloodstream. So adaptations include these cells have microvilli, so these highly folded cell surface membranes to increase the surface area for that transport. They also contain many mitochondria so that lots of ATP can be produced for this active transport. And then there'll also be lots of carrier proteins and co-transporter proteins embedded in the cell surface membranes for all of that transport. And then water is reabsorbed by osmosis because following the absorption of the solutes, that creates a water potential gradient, then water can move in by osmosis as well. Then we get to the loop of Henle where we have the maintaining the sodium ion gradient. And in the descending limb, water leaves by osmosis, but sodium ions cannot. So the descending one is the one going down. The ascending limb is the one going up. Sodium ions are actively transported out, but water cannot leave. And this creates a water potential gradient in the medulla, so this surrounding area outside of the nephron, which allows even more water to be reabsorbed by osmosis at the distal convoluted tubule and the collecting duct. And water reabsorption is regulated by the hormone ADH, which we're going to come on to shortly. So here's a bit about the loop of Henle in a bit more detail, because we said that descending limb, the walls are thin and permeable to water, but impermeable to ions. So as the filtrate moves down the descending limb, water will move out by osmosis into the surrounding medulla due to that lower water potential. and the concentration of the filtrate will increase. As it moves up, the filtrate moves up the ascending limb. The walls are thicker and they're also impermeable because they don't have aquaporins within them, so water cannot move out by osmosis. But sodium ions can be actively transported out, and that then means we get a really negative water potential gradient. Now, at the base of the ascending limb, where the filtrate is very concentrated, some sodium ions will also move out by facilitated diffusion. But this whole process creates a high concentration of sodium ions in the medulla, lowering the water potential, which is essential for water reabsorption in the distal convoluted tubule and collecting duct. So, how the hypothalamus and the posterior pituitary gland are involved. The hypothalamus is going to detect changes in the water potential. Specifically, we have osmoreceptor cells, which are the receptors that detect the changes in water potential. So if the water potential of the blood is too low, water is going to leave the osmoreceptors that are in the hypothalamus, causing those cells to shrivel, and that stimulates the hypothalamus to produce more of the hormone ADH, antidiuretic hormone. And if the water potential of the blood is too high, water enters the osmoreceptor cells by osmosis, and that stimulates the hypothalamus to produce less ADH. Now, the hypothalamus is where ADH is produced, but ADH then moves to the posterior pituitary, and that is where it is released from into the blood. You do need to know that distinguishment. It's come up as an exam question before. And once that ADH is released in the blood, it then travels in the blood to its target organ, which is the kidney. So ADH, antidiuretic hormone, this is going to cause an increase in the permeability of the walls of the collecting duct and the convoluted tubule. And as a result, more water is going to move from the nephron by osmosis back into the blood. And that results in urine that is more concentrated and in smaller volumes. So there are receptors on the cell membranes of the distal convoluted tubule and the collecting duct to which ADH bind. When ADH reaches the kidney, it binds to those receptors, and when bound to the receptors, it activates a phosphorylase enzyme in the cells. The phosphorylase causes the vesicles containing aquaporins to move and fuse with the cell membrane to get more of these aquaporins, which are protein channels that water can move through. Now, this is a little bit extra information. You could include it in an essay. You could still get marks sometimes in the mark scheme for it, but it's not essential that you need to know that process. You do need to know that ADH binds to the receptors, increases the permeability of the cell membrane to water. Aquaporins is often a mark as well. So therefore, ADH causes an increase in the permeability of the walls of the collecting ducts and the convoluted tubule, which means more water will move into those cells and then into the blood by osmosis. And therefore, urine is more concentrated and you return more water to the blood. So in summary then, if the water potential of the blood increases, meaning too much water, so you might have drunk a lot of water, that's detected by the osmoreceptors in the hypothalamus. Hypothalamus produces less ADH. The distal convoluted tubule, which again, that is not an accepted abbreviation. Do not abbreviate it like that in the exam. You have to write it out in full. ADH you can abbreviate though. So the distal convoluted tubule and the collecting duct walls become less permeable to water. So that means less water is reabsorbed in the blood and more remains in the filtrate in that collecting duct and goes on to create larger volumes of more dilute urine, bringing your water potential of the blood back to normal. Now, maybe you've sweat a lot and you've lost a lot of water and your water potential of the blood decreases. That is going to be detected by the osmoreceptors in the hypothalamus. The hypothalamus will produce more ADH, which is released into the blood by the posterior pituitary. The distal convoluted tubule and the collecting duct walls become more permeable to water, so more water is reabsorbed into the blood by osmosis and less is lost in the urine. So you get more concentrated urine, and that means you get your normal water potential of the blood.
AQA topic 7, and it starts with 3.71 inheritance for A level only. There are lots of key terms in this topic that you could be asked for a 1 or 2 mark question. So I recommend that you screenshot this or when the answers are there, or turn this into flashcards because these are essential to learn. Genotype, that's the genetic constitution of an organism, which basically means the alleles it has for a particular gene. Phenotype is often 2 marks because it's the expression of the genetic constitution for 1 mark. Second mark is interaction with the environment. Homozygous and heterozygous I haven't seen come up as definition questions, but it's important that you know what those mean. Homozygous meaning you've got 2 alleles that are the same for a particular gene. Heterozygous is 2 different alleles for the same gene. Recessive allele and dominant allele have both been assessed before. Recessive is an allele that's only expressed if there is not a dominant allele present, whereas a dominant allele will always be expressed in the phenotype. Codominant is when both alleles for a gene are equally dominant and are both expressed in the phenotype. Multiple alleles is if there are more than 2 alleles for a single gene. Sex linkage is when the gene of interest is located on the X chromosome. So locus means the location of the gene on a chromosome. Autosomal linkage is when genes are located on the same chromosome. So it's going to be 2 different genes are found on the same chromosome, but it's not the sex chromosomes. Epistasis is when one gene modifies or masks the expression of a different gene at a different locus. Monohybrid and dihybrid don't come up as definition questions, or at least they haven't, but it's just knowing what they mean. So monohybrid, mono meaning 1, it's looking at the inheritance of just 1 gene. A dihybrid cross would be looking at the inheritance of 2 genes. So inheritance then, we're going to go through all of the types that you need to know for AQA, but there's different ways to present what allele you have for a particular gene. And that's what I mean here by this coding. This is what we'll use when we do Punnett squares or flow diagrams to work out predictions of what you might see in offspring. So for a simple monohybrid cross, that's what you would have done at GCSE. There's just 1 gene with 2 alleles, the dominant or recessive. So you pick 1 letter to represent the gene. Capital is dominant, lowercase is recessive. But then when we get to codominance and multiple alleles, we need to do the coding differently. Because for codominance, it means both of the alleles are dominant. So we can't pick 1 letter and have it capital for dominant because they're both dominant. So instead, you pick a base letter to represent the gene. And then superscript, you have the 2 alleles. So a dominant B, dominant A. And it's not going to be the same letter because they have to be capitals to show they're dominant. Multiple alleles, because it's more than 2 alleles for a gene, again, you can't pick 1 letter because it only gives you 2 versions. So we use the base letter to represent the gene. Superscript, you pick the allele letter. And for all of these, if they haven't told you what letters to use, you can pick any letters that you want. But I recommend that you pick letters where the lowercase and capital letter are completely different shapes, like all the examples I've done here, so that it couldn't be confused by an examiner whether you did put the capital or lowercase. Sex linkage, you have the base letter as the chromosome, so whether it's the X or the Y chromosome, and the allele is superscript next to that. There won't ever be anything written next to the Y chromosome because you only get the gene on the X chromosome. Autosomal linkage, this is where we said you have 2 different genes on the same chromosome. And usually this is just done with simple monohybrid inheritance. So 1 letter to represent the gene and then capital and lowercase for dominant and recessive, and that's for both genes. Same with epistasis. This is when 1 gene masks or modifies the expression of the second gene. So we've got 2 different letters representing those 2 different genes, and then dominant and recessive is shown by capital or non-capital letter. So let's have a look at some examples, starting with monohybrid inheritance. Cystic fibrosis is caused by a recessive allele. If 2 carriers reproduce, what's the probability they'll have a child with cystic fibrosis? And on top of that, what's the probability they will have a girl with cystic fibrosis? Now you can pause, have a go. I'm going to go straight through. One thing to be really aware on these questions is they're doing this more often now, where they specify the sex of the child as well, when it isn't a sex-linked inheritance question. So what that means is we need to do our Punnett square to work out the probability of having a child with cystic fibrosis. And it's always 50% probability of having a girl, 50% probability of having a boy. So whatever your answer is for the cystic fibrosis, you then half that because that's 50%. So here are our two carriers. I've picked the letter F here because the capital and lowercase are different shapes. We could pick A. That's even more obvious if you had a capital A, lowercase a. So we can see here that on our Punnett square, if those are our two parents' genotypes, I've then put in the possible gametes that each parent could produce. I've then done our cross to show F with this F, the two capitals, lowercase and capital, lowercase and lowercase, and capital and lowercase. And because cystic fibrosis is caused by a recessive allele, they will only have cystic fibrosis if they are homozygous recessive, because there's no dominant alleles there in their genotype. So we have 25% probability that these two parents will have a child with cystic fibrosis. Then we need to also account for a girl with cystic fibrosis. We need to half that. So times that by 50% or half it is 12.5%. Next, we have a codominant example. Cows can be red, white, or roan in colour. Red and white are both dominant. If two roan cows reproduce, what's the probability they will have offspring that are red? Again, pause now and you can have a go at this Punnett square. I'm going to go straight through it. So for this one, parental genotypes, red and white or roan. Red and white are both dominant. So that means those are our two dominant alleles. Now, they'll probably give you more information than that in a question to make it explicitly clear that roan isn't a third allele. They'd need to either tell you it was codominant or give you a hint towards that a bit more. So here, red and white are both dominant. And when red and white are the codominant genotype, you get roan. This is roan here, is when you've got both the red and the white being expressed in the phenotype. And yes, I would personally say that's more brown than red, but it's called red for some reason. So there's our two parents who are both heterozygous. They have that codominant genotype. So if we do our Punnett square, both parents would have one of the white alleles and one of the red alleles. We then combine those in our Punnett square, and we can see we'd have roan, white, roan, and this would be our only one that would be red. So 25% probability of having a red cow. Now, when you do these Punnett squares or flow diagram, it's really important that you are showing the parental genotype if that's not already been given. Then the gametes, and the difference between the two is the gametes are separating those two alleles, whereas the genotype, they're both written next to each other. You then would have to show the offspring genotype and the matching or corresponding phenotype for that genotype. That's why I always leave a space and write the phenotype in the box so it's matched, and then we've got our probability. So then we've got a blood group example, and this is not on the spec. It's just a common example because it models codominance and multiple alleles. So blood groups, you could be blood group A, B, AB or O, and this is referring to the type of antigen you have on the outside of your red blood cells. So some people have antigen A, some have antigen B, some have both antigen A and B, and some have no antigens at all. So that's what the blood groups are corresponding to. And the genotypes for this are, to be blood group A, you could either have two copies of the dominant A allele, or one copy of the dominant A and one copy of O, because O, blood group O, that recessive allele, well, it is a recessive allele. Blood group B, so you could have two copies of dominant B or B and the recessive O. Because O is recessive, you are only going to be blood group O if you have two recessive O alleles. And then blood group AB is the codominant genotype, because if you have A and B, they're both dominant, they are both expressed in the phenotype. So we've got codominance and multiple alleles because we have three alleles. So if we have a look at a genetic cross example, as I said, this is not on the spec that you need to know blood groups. So they would have to give you information about it to be able to work it out. But I went through it because it's a relatively common example, or at least it used to be. So we're told we've got parents with blood group AB and blood group O reproducing together. In the question, we're then told IA and IB are codominant and IO is recessive. What is the probability that they will have an offspring with blood group A? So the first step is working out the parents' genotypes. And if they are AB, they must be A and B, that codominant. And if it's O, that is recessive, so homozygous recessive for O. Then we can put in the parents' gametes. Then we can do the cross, and then we can add in the blood group, so the phenotype corresponding to those genotypes of the offspring. And we were asked the probability of having an offspring with blood group A, and that is 50%. Then we've got a sex linkage example. So colour blindness, again, you don't need to know this as an example. In fact, all of the examples I've done are application. There is no genetic condition or disease that you need to know for AQA. You'd always be given the information and then you do the genetic cross. So colour blindness is caused by a recessive allele, or at least one type of colour blindness is caused by a recessive allele found only on the X chromosome. If a non-colourblind male reproduces with a female carrier of the allele, what is the probability their children will be colourblind? So step one is always working out the genotypes of the parents. And when it's sex-linked, you get marks for including the sex chromosomes in the genotype as well. So you do have to include that. For the male, it's XY. For the female, it's XX. And remember, you only find the allele on the X chromosome. So the female here is a carrier, and we're told it's recessive. So there'd be X. I've picked the letter R, so it's really clear, the dominant versus recessive. So X, capital R, lowercase r. And that's a times, that's not part of the genotype. That is crossed with the male, which is X, capital R, Y. Because they are not colourblind, they only have the dominant allele. So then if we have a look at our cross, you might have noticed it's actually slightly changed. So I just went and edited it to really emphasise a key point, which I'll get to. So we have the male gametes here, X, capital R, and then the other gamete would just be the Y. Then we've got the X, lowercase r, and X, capital R for the female, the mother. Now, I've actually done that the other way round, how I've written it here, because there it was the capital first, but it doesn't matter. Your answer ends up the same. So we then fuse together the gametes to show all of the possible genotypes in the offspring. And then I've added the corresponding phenotypes for those genotypes. And you have to state the sex as well as the characteristic linked to the gene of interest. So here it's female, normal vision. They may actually call that colour vision. In the older exam questions, they used to say normal, but they've started to not use that now because it implies if you don't have colour vision, then it's not normal. So they would say colour vision or normal vision. But it's female and they have colour vision. Female colour vision, male colour vision, male colour blind. So you have to state the sex and also the characteristic in the phenotype. And for this one, that's 25% probability of having a colour blind child. Epistasis then, that's when one gene influences the expression of another. And this is often seen in colours. So it could be coat colour in mice. It could be coat colour in Labradors. It could be fruit colour of vegetables. So these are the common examples that you see. And we're going to go through an example and then do the cross. So let's have a look at coat colour in Labradors. The first gene controls whether pigment is going to be produced. And if you have the dominant version of that gene, so dominant allele, then that codes for pigment will be made. The recessive version codes for no pigment. So if a dog is homozygous recessive for this gene, they will not produce any pigment, and that's how you get the yellow dog colour. So that is no pigment has been produced in the fur. Gene 2 controls which colour pigment is expressed if they are able to make pigment. Capital B is black fur, lowercase b is brown fur. So that's how we get these three different colour furs, or hairs if it's dogs, from 2 genes. So let's have a look at the cross here. We are told straight away the parent's genotypes. And you can pause now and have a go at, first we're working out, what would the phenotype be of these 2 dogs? So we've got capital E, lowercase e. This was the one that controls where the pigment is made. This is the gene that controls what colour that pigment is. And for this one, they'd both be black because they've got the capital E, which means pigment can be made. Capital B means the pigment would be black. So if we then did a genetic cross, first of all, we have to work out all of the possible gametes both dogs could make. And because this is now a dihybrid cross, meaning the inheritance of 2 genes simultaneously we're looking at, you have to show all the possible combinations that you could get. So you need to show just 1 copy of each gene. So 1 letter E, 1 letter B, but all of the possible combinations. So we could have the capital E with the capital B, the capital E with the lowercase b, the lowercase e with the capital B, the lowercase e with the lowercase b. So we get 4 possible gametes for both of those parents. So that means our Punnett square is going to be 4 by 4. It's much bigger. And we now need to show all of the possible genotypes. And I've then added in all of the possible phenotypes as well. So here we go, we can see all of those genotypes and the corresponding phenotype. And we have 9 black, 4 yellow, 3 brown. So we've got a 9:4:3 ratio for this. So that actually goes into dihybrid crosses. A dihybrid cross is the inheritance of 2 genes at the same time. And we've just done one example, but here is a more straightforward example where we don't have epistasis involved. So we've got a round and yellow pea, a wrinkled and green pea. So 1 gene is coding for shape, 1 gene is coding for colour. And with all genetic crosses, we need to have the parent's phenotype, the parent's genotype, the possible gametes the parents could make, the offspring genotype, the offspring phenotype, and then the proportion of each phenotype. Sometimes 1 and 2 might be given to you, sometimes they aren't. So for this one, the genotypes, you would need to have information to know this, but we've got round and yellow, and in this instance, both parents are homozygous for both genes. They would have needed to tell you that for this parent, which you wouldn't know otherwise, because it's a dominant characteristic, round and yellow. So they could have either been homozygous dominant or heterozygous for both genes. So round and yellow, wrinkled and green are the 2 recessive characteristics. They had to be homozygous recessive for both genes. Possible gametes then, the round and yellow can only give dominant alleles. So 1 dominant R, 1 dominant Y in every gamete. And this one here can only give recessive alleles. So all of the gametes would have 1 recessive R, 1 recessive Y. So that means we could do a Punnett square, but actually every offspring will get this gamete and this one. So combining them together, that gives us offspring that are heterozygous for both genes. And that would mean they'd be round and yellow. So 100% are round and yellow in our first generation in this dihybrid cross with these parents. Now you could be asked to then cross F1, which means first generation. And that would now mean we're crossing 2 peas that are heterozygous for both genes. And this is now when we'd get 4 possible gametes for both of those genes. So capital R with the capital Y, capital R with the lowercase y, lowercase r with the capital Y, lowercase r with the lowercase y. So looking at all the possible combinations of getting just 1 of the R's and 1 of the Y's in every gamete. And this is then when you get that big Punnett square again, the 4 by 4 Punnett square. And if we combine all of those together and then add in all of the phenotypes to the corresponding genotype, we can see we get a 9:3 to 3:1 ratio. And in a dihybrid cross, if you have 2 parents that are both heterozygous for both genes, like we saw here, then you will always get 9:3:3:1 because you'll always have this pattern of gametes. Therefore, crossing it together, it doesn't matter what the genes are, you'll still get this outcome. So sometimes for 1 mark, you don't have to do the Punnett square and they just ask you and they tell you, you've got a dihybrid cross, both parents are heterozygous for both genes, what will the ratio of the phenotypes be? And it's just 9 to 3 to 3 to 1 because it's learning that fact. However, there are exceptions. So if there is autosomal linkage, you won't get that ratio. If there's crossing over in meiosis, you won't get that ratio. And if there's epistasis, you don't get that ratio. So sometimes that is also asked as just a 2-mark question. Maybe it's to give 2 reasons why you might not get that, or suggest 2 reasons why you won't get that ratio. And it's those 3 reasons. And that leads us into what is autosomal linkage and how does it mean, or how does it result in you getting a different ratio? Now, I'm actually going to quickly go over crossing over in meiosis very briefly, because this actually is from topic 4, but you need to have an awareness of it for this. So crossing over of homologous chromosomes happens in meiosis 1, the first round of division in meiosis. It's when those homologous pairs, so the chromosomes, pair up at the equator to form a bivalent, meaning 2 chromosomes next to each other. The non-sister chromatids can cross over, get tangled, break and swap that section of the chromatid. And as a result, when you come to work out what gametes a parent might be able to produce, you get new combinations of alleles in the gametes. So that's the relevance here, because you might have thought, well, these parents could make these 2 alleles, but if crossing over happens, maybe they can actually make 4 different gametes. So autosomal linkage, this is when we have 2 different genes located on the same chromosome. And as a result, they have to be inherited together. So it's going to impact the gamete predictions, because I'm using the same example here. The 2 Ps, heterozygous for both genes, the capital R, lowercase r, capital Y, lowercase y. But now we're imagining that there's autosomal linkage. So we've got gene 2, which was for shape. Gene 1 is for colour. They're still heterozygous for both genes, but we can see this chromosome has the dominant R and the dominant Y. This chromosome of the pair has the recessive R and the recessive Y. And that means after meiosis, you will have to have gametes that are capital R, capital Y, because this chromosome will be going into one gamete, this one will be going into the other. So we previously said when there was no autosomal linkage, there were 4 possible gametes that parent could make, but with autosomal linkage, there's actually only 2 possible gametes that these parents could make. So here was our original when there was no autosomal linkage, heterozygous for both genes, and here are all the possible gametes. So these 4 and these 4 for that parent. But now there is autosomal linkage, we can actually only create these 2 gametes, and that's the case with both parents because they both have this autosomal linkage. So that now means that this is the possible genotype and phenotype combinations we could get. So we have 3 yellow and round and 1 green and wrinkled, which is not our 9 to 3 to 3 to 1 ratio. So if there is autosomal linkage, that is how it's going to affect the ratio. But now let's add in though crossing over as well. So here's what we've said so far. With these 2 parents, we would expect 9 to 3 to 3 to 1 ratio, but if there's autosomal linkage, we actually had a 3 to 1 ratio. However, sometimes you're told that information and then you say, scientists actually observed the following results. And we can see we have 4 phenotypes, and here are the numbers they observed. They've got mainly yellow and round, then green and wrinkled, and then you don't have very many yellow, wrinkled or green round. And they would ask you, how is it possible they got these results? And the only way this is possible is if there is autosomal linkage and crossing over in meiosis, because that crossing over in meiosis created new combinations of alleles in the gametes, and that's why it was possible to get these 2 extra phenotypes. So let me show you a common answer for that. This is actually just modelling what we mean here. So here we had our autosomal linkage. We've got the recessive alleles for those 2 genes on this chromosome, the dominant on this chromosome. However, there was crossing over in meiosis 1. Those chromatids break off and swap. So as a result, we now have new combinations of alleles in the gametes. This was the original, so the non-recombinant, the 2 recessive alleles. This one at the end is also non-recombinant, meaning it's not being recombined and created through crossing over in meiosis. We've got capital R and capital Y. But these 2 options here, capital or dominant R and recessive Y, and this one is the dominant Y, recessive R, that is our new combinations of alleles in the gametes, and it's created these 2 new gametes. But crossing over is rare. So that means you won't get very many of these 2 gametes, and you'll get lots of this gamete, lots of this gamete. And that then leads into our answer. So explaining these results. Yellow and round, the top one, these are the 2 genotypes that you could get from the non-crossing over possible gametes. And you could also get the green and wrinkled from 2 lots of the lowercase r, lowercase y joining together. In fact, that's actually written here as well. So those are the most common, the top and the bottom are the most common because those gametes are not a result of crossing over in meiosis, and crossing over is rare. Now, the reason we have fewer green and wrinkled compared to yellow and round is there's 2 possible genotypes compared to only 1 for those phenotypes. And that's because green and wrinkled is recessive. So here we can see the heterozygous genotype still gives us yellow and round. So that's why there's the most yellow and round, then the green and wrinkled. The 2 phenotypes in the middle of the table are not as common, and that's because crossing over is rare. So we don't make very many of these 2 gametes. And that then means we don't get very many of these genotypes, which code for those phenotypes. So the key thing with a question like this is pointing out autosomal linkage occurred, if they haven't already told you, and also pointing out crossing over has occurred. But that's rare, which is why we don't get very many of these gametes and therefore those phenotypes. So sometimes the mark scheme does insist that you literally pick out which gametes were a result of crossing over, which were not, and link that to the numbers of each phenotype. So you could be asked to do a statistic on this, and it would be chi-squared, because you're looking at how many individuals fit into a category, or in this case for inheritance, how many individuals have a particular phenotype. And if we want to see, is there a difference between what we expect based on our Punnett square and what we actually observed, we can do a statistic to see if it's a significant difference between what we expected and what we observed. So here's an example. Going back to what we did at the start, cystic fibrosis caused by a recessive allele. We said we expected there to be 12.5% probability of it being a girl with cystic fibrosis. We could then actually look in the offspring and count what proportion did have cystic fibrosis and were a girl, and see what we expected. Is that significantly different to what we observed? Now, you wouldn't usually do that with humans because there's not going to be a large enough sample of the offspring from those 2 parents for it to work. But we've got an example here with corn, looking at the colour of the corn kernels. And in this example, we have got purple and yellow corn. Purple is dominant, yellow is recessive. Based on this Punnett square, we expect a 3:1 ratio of purple corn kernels to the yellow colour. So that's what we expect. We're then told that a student counted the actual number, so this is what they observed, and they got 21 purple kernels, 13 yellow. And we have to say, does that follow what we expected? So our null hypothesis would be there is no significant difference between the expected and observed frequency of the colour of corn kernels. And then we can fill in a table like this. Now, you wouldn't be expected to remember how to draw this table, but it has come up that they've given you this table with empty sections and you then have to fill it in. So you need to know that O is the observed, E is the expected. So here's what the student observed: 21 purple, 13 yellow, which means there were 34 in total. We said we expect a 3-to-1 ratio, but we need to turn that ratio into frequency. So that means we need to know what 75% of 21 is and 25% of 13. And I've calculated that here. O minus E is your observed frequency minus your expected frequency. So we've got that result here. Then we square that. So even if it was negative, because it's squared, it all becomes positive. And then we divide that by the expected frequency. The sum of this column is your chi-squared value. So we've got a chi-squared value of 3.176. You could be asked to work out the degrees of freedom, and that is the number of categories minus 1. We've got 2 categories, purple or yellow, so we have 1 degree of freedom. Now, if we were to look at this in a critical values table, it's a bit blurry, but we need to look at the P equals 0.05. That means that we'd have less than 5% probability that the difference between observed and expected is due to chance. We've got 1 degree of freedom. We've picked out the column that has 0.05. So we are comparing to this critical value, 3.841. And the critical value is like a threshold. You've got to see, does your chi-squared value meet or exceed that critical value threshold? And it has to meet or exceed it to be able to say there is a significant difference. So ours doesn't. Our chi-squared value is lower than the critical value. And there'd be a mark for stating that, which I've put here. So what that means is there is more than 5% probability that the difference is due to chance, not R, I should say is. And you do have to state difference and not just result to get the mark. So that means we can accept the null hypothesis, and there is no significant difference between what we observed and the ratio that we expected. Or in other words, the corn kernels did show a 3-to-1 ratio because it wasn't different to what we expected, and we expected 3-to-1. Next then, 3.7.2, populations. And this is where the Hardy-Weinberg principle comes in. It's normally assessed within the inheritance questions though as well. And Hardy-Weinberg is a mathematical model that can be used to predict allele frequencies within a population. So here are just some definitions to check. Turn them into flashcards if you want, because they are commonly assessed. Gene pool, that is all the alleles of all the genes within a population at one time. Population, this comes up as a 2-mark question now. All the individuals of one species in one area at one time. Second mark, that can interbreed to make fertile offspring. Allele frequency, the proportion of alleles within the gene pool. So here's the Hardy-Weinberg equation. And in fact, although the top one is the equation, you have to use the top and this one here simultaneously to get your answers. So you need to know that. Lowercase p, that is the frequency of a dominant allele in the population. Q is the frequency of the recessive allele. So a single letter by itself is representing the frequency of an allele. P is dominant, Q is recessive. And if you add all of those together, that equals 1 because that's all of the alleles for that gene in a population. P squared is the frequency of genotypes. Specifically, this one is the dominant genotype. 2pq is the heterozygous genotype frequency. Q squared is the frequency of the homozygous recessive genotype. And again, if you add all of the genotypes together, it equals 1 because those are all of the possible genotypes you can have for this particular gene. So in this question, cystic fibrosis is caused by a recessive allele. 0.02% of the UK population suffer from cystic fibrosis. What proportion of the UK are carriers? So step 1 is always identify from the 2 equations, what information have you actually been told? So we have been told that 0.02% suffer from cystic fibrosis. And cystic fibrosis is caused by a recessive allele. So to have cystic fibrosis, you have to be homozygous recessive. So 0.02% is Q squared. But we can't have it as a percentage, so I've converted it. Then we need to work out what we need to identify. And we've been told what proportion of the UK are carriers. Carriers, that would be heterozygous, so they want us to work out 2pq. And the strategy for Hardy-Weinberg is always work out which bit of the formula you have, then which bit you're trying to identify, and to do this, you then always need to work out what P and Q are. So Q, we know what Q squared is, so if we square root that, we then get what Q is. And now we've got Q, we can use this formula to work out P. Rearrange it, 1 minus 0.014, this is what P is. So now we are working out what proportion are heterozygous. So for heterozygous, it's 2pq, so we then just need to do 2 times P times Q. And that comes to 0.03. And they've just said proportion, so we can leave it as that, or you could write 3%, but do look carefully to see if they've asked for your answer as a percentage or just proportion. Next then, 3.7.3, evolution may lead to speciation. So variation, individuals within a population of a species may show a wide range of variation in the phenotype, and that is because of both genetic and environmental factors. If it's genetic variation, the primary source of genetic variation is mutations, but also genetic variation is introduced through meiosis and the random fertilization of gametes. Predation, disease, competition for the means of survival. Basically, this means there's going to be competition within populations. There's going to be disease, which means some individuals are more likely to survive, and there might be predators, and therefore if you're more likely to evade a predator, you're more likely to survive. And this then leads to natural selection. And the organisms with the phenotypes providing a selective advantage, meaning they're more likely to survive, they are also then more likely to reproduce, pass on those beneficial alleles to the next generation, and that's how we get this differential reproductive success, or in other words, the allele frequency for that gene might change in the gene pool. Now, all of this is in topic 4 as well as topic 7. And I go through that in more detail in the entire topic 4 video, because this is just a brief summary that they do in topic 7. And basically they do it so that you can be assessed on this content on paper 1 and paper 2 and paper 3. So types of selection. There are 3 different types. Stabilizing selection. So by type of selection, we mean the outcome of natural selection. Here's our original population looking at number of individuals for each trait. And the modal trait is the most common, or the middling feature. So medium color is the most common. The very pale, the very dark are not as common. And stabilizing selection does favour the average phenotype. And individuals with the extremes are less likely to survive, therefore less likely to reproduce, and therefore over many generations, you lose those extreme traits, and the middling trait that was already the most common becomes even more common. And the example that you learn in topic 4 for this is human birth weight. Directional selection also came up in topic 4. This is when one of the extremes provides selective advantage and is favoured in those environmental conditions. So maybe there's an environmental change, which now means this extreme trait provides the selective advantage. So over many generations, more individuals now have the extreme trait, the middling trait's less common, and the other extreme is almost completely lost. And the example that you learned in topic 4 was antibiotic resistance, because those with the most resistance were more likely to survive. The new one for year 13 is disruptive selection. And this is when both extremes are favoured, meaning they both provide a survival advantage. So if an individual has either of those extreme traits and allele, they're more likely to survive, reproduce and pass on that allele. And this can then lead to the process of speciation. Now, you don't have to know an exact example for this like you did for the other 2, but an example could be maybe birds with very small or very large beaks are more able to survive, because birds with very small beaks might be able to get their beaks into tiny holes in trees to eat insects, and the birds with bigger beaks are able to crack seeds and nuts and eat that. But if you've got a medium-sized beak, you're not very good at either. So you're not going to get as much food. So this then takes us into the concept of evolution, genetic drift, and speciation. Evolution is a change in allele frequency within a population over many generations. And that can also lead to speciation. Speciation is the process that results in the creation of a new species. And this is going to occur when one original population of the same species become reproductively isolated. And this isolation means you now have two separate populations of the same species, but they do not breed to make fertile offspring at this point. And that's what reproductively isolated means. It means they cannot reproduce. Now, the reason that might happen is different, and we're going to come to that. But the outcome, if you've got these two separated populations that are no longer reproducing, both populations will have random mutations happening. But because they're no longer reproducing together, those mutations stay within this population. Mutations that anyone has in this population stay there as well. And that means over time, you get this accumulation of differences in the gene pools, so that if they were able to start reproducing together again, they'd be so genetically different that they cannot interbreed to make fertile offspring, and therefore they're now classed as a different species. So that is our process of speciation. And what causes them to be reproductively isolated is either a geographical barrier, which is known as allopatric speciation, or they might be in the same location, but they still don't reproduce. And that's known as sympatric. So allopatric speciation is when they're reproductively isolated because of some geographical barrier. And that could be a new mountain range, a body of water, but they just can't reach each other. All of the other marks would be exactly the same. So you always have that concept of they are reproductively isolated. You have to state whether that is geographical, allopatric, or sympatric, which we'll get to. That means that different mutations will accumulate in both populations, and therefore they'll go through natural selection and evolution for their environment. You get an accumulation of different mutations, and therefore they're now so genetically different they cannot interbreed to make fertile offspring. So they're two new species. And this is an example of directional selection. Sympatric, this is when populations become reproductively isolated, not because of a geographical barrier, but due to something else. And it could be their behaviour. So it could be that linked to the idea of courtship, or it could be as well that they are now producing gametes at different times of year. So they're fertile at different times of the year. So even if they are mating, they're not making offspring because one of the pair in that reproduction is not making a gamete. And then you get the same marks again. So for genetic drift, this is the change in the allele frequency within a population between generations. And there will always be genetic drift from one generation to the next, but continual, substantial change or genetic drift is when it's classed as evolution. And in smaller populations, genetic drift has a bigger impact because if you have a slight change in the number of alleles, in a small population, that's a big change in terms of the frequency. Because if you just change 2 out of 10, that's 20% change. If you change 2 out of 100, that's only 2% change. So the smaller the population, the bigger the impact. And that's why smaller populations evolve more rapidly. Next then, 3.7.4, populations in ecosystems. And again, lots of key terms here. Population is a group of organisms of the same species living in the same area at the same time, and they can interbreed to make fertile offspring. Habitat is the part of an ecosystem in which a particular organism lives. Community is all the populations of different species in the same area at the same time. Ecosystem is a community and the non-living components of the environment. This bit here isn't part of the definition, but just being aware that ecosystems vary in size. Niche is an organism's role within an ecosystem, and that includes its position in the food web and its habitat. And this is going to be determined by adaptations to both the abiotic and biotic factors they're exposed to. Carrying capacity is the maximum population size an ecosystem can support. Abiotic factors, those are the non-living conditions of an ecosystem. Biotic factors is the impact of the interactions between organisms. So a bit more on these two, abiotic and biotic factors. We said that the population size and the carrying capacity are dependent on abiotic and biotic factors. Abiotic factors are the non-living factors, so that could be things like temperature, oxygen concentration, carbon dioxide concentration, light intensity, pH, soil conditions. And the key kinds of questions you get are either linking to something else in the specification to explain why any of those would affect plant survival or animal survival. And that might link to photosynthesis, might link to enzymes. And then it's knowing maybe links to how they could evolve. So that would be the idea of natural selection. If they're better adapted to that environment, they're more likely to survive, pass on those alleles to the next generation. And then eventually, over many generations, much of that population will have adaptations to those abiotic factors. Biotic factors, that is looking at things like competition and predation. Competition can be either interspecific or intraspecific. Interspecific competition is when members of different species are competing. And the way to remember this, I think, is international travel, spelt inter, T-E-R, is when you're traveling to another country. So it's a different country. So interspecific competition is a different species. Intra is when members of the same species are competing. They'll compete for the same things. It's just if it's members of the same species, they're also competing for a mate. And this is for animals and plants. It's not just animals that compete. Plants would also be competing for water availability in the soil, carbon dioxide concentration, mineral ion concentration in the soil too. Predator-prey relationship, so this is still biotic factors. Looking at the interaction between the predators and prey in a food web, you get this typical shape graph, regardless of the species, where we've got the predator shown in blue and the prey shown in red. And you always see these three features. The size of the predator population is usually going to be lower than the size of the prey population. Both populations will fluctuate in size, and there's a bit of a lag time. You'll see a change in the prey before you see the change in the predator population. And this is because the predators are eating the prey. So there has to be more prey than there are predators to sustain them. They both fluctuate because as the predators are eating lots of prey, that causes their population size to decrease. That will then cause the predator population size to decrease. But because the predators have gone down, the prey can then increase, and this happens over and over. So the last little bit then is looking at sampling, so that you can look at population sizes. And the reason we sample is it's going to be more accurate and less time consuming than trying to count every single individual. But we have to make sure that the samples we take do accurately represent the population. So that would mean you have to do random sampling to avoid bias. And that's if it is a uniform area, meaning it's equal distribution. If it's not equally distributed, you do systematic sampling, which is a line transect or belt transect, to examine that change over distance. And then also it has to be a large number of samples to ensure your sample is big enough to accurately represent the population. Now, if you say large number, that should be fine. If you're going to state a number, I would say 30 plus, just to show it really is large. Now, the different ways that you can sample then. First thing is thinking about what type of organism you're estimating the population size of. So if it's slow moving or it doesn't move, like a plant, then you'd use a quadrat. If it's a motile, meaning moving organism, like a bird, you would use mark, release, recapture. If you've got uniform distribution, you do random sampling. If it's uneven distribution, it's systematic sampling, which is your belt transect or line transect. We're going to have a look at that first, and then we'll come back to the mark, release, recapture. So random sampling. First of all, you would lie 2 tape measures at right angles to create a gridded area. Then use a random number generator to generate your coordinates. That then tells you where to place your quadrats. You go along those 2 numbers of your coordinates. Depending on what the question says, will determine how you're estimating in your quadrats. Because it might say you need to do percentage cover, in which case you're estimating inside of your quadrat what percentage of it is covered by your plant. Or it might ask you to estimate the number, and in which case you do density, which means count the individual number of plants that there are there. Frequency doesn't tend to come up, but local frequency is the idea of you would use a gridded quadrat and just count how many of the squares it's present in. So it's slightly different to percentage cover, because that you're estimating the percentage covered. The local frequency is just, is it present or not in the tiny squares? Now you have to make sure it's representative. So you have to do this a large number of times, so at least 30 times, calculate a mean. And then if it's percentage cover, it ends there. If it's density though, meaning you counted the number of plants in the quadrats, you now have your mean number of plants in a quadrat, but we want to know it for the entire field. So there'd then be a mark for describing how you'd work that out. So you'd need to know what the area of the field is and the area of a quadrat to see how many quadrats would fit into that field. And then whatever your mean is, you're multiplying that by the number of quadrats that fit into the field. Another thing that they've become really picky on is if they've said describe how to work out the mean number, you also have to actually describe how to calculate the mean and not just say calculate it. So you'd need to say add up all of your 30 repeats and divide by 30. A line transect, this is used to estimate population size when there are that uneven distribution of species. So that could be things like on a rocky shore, if you've got a river in the middle of a field, and you'd place a tape measure to act as your line transect, going through all of those different sections so that you then do get information for all of the different parts. And a belt transect means that you've got your tape measure and you're lining up quadrats next to it. An interrupted belt transect is what is most commonly used. This means you place your quadrat at intervals along your tape measure. And it might be every 5 meters, every 10 meters. However far you start to notice a difference, that's the frequency that you'd need to place them to make sure you don't miss any important information. You still have to repeat multiple times though. So here would be your method if you were asked to describe a line transect. Method, that is. So place your tape measure at a right angle to the shoreline. So that would mean it's going through all of those different sections that we want to see. Place your quadrat every 5 meters. It doesn't have to be 5, but it's at stated intervals. Collect your data, just like we said last time, so either density or percentage cover. And repeat again a large number of times, calculate a mean. But for this one, the repeats would be multiple repeats at parallel, so in parallel to your original one, along the beach or whatever area it is that you're measuring. So then we've got mark, release, recapture for if you are trying to estimate the population size of a motile organism. Here is our method. So you need to capture an initial sample. You need to mark them. So that might be putting a bit of paint on a snail shell, for example. Then you release them back into the wild. You then leave them for a period of time after they've been released so that they can randomly disperse throughout the habitat. Then you take a second sample, and the total number captured in the second sample and the number recaptured, meaning the ones that have the mark on them, are recorded. Then we can use the formula to calculate our estimate. So to work this out, we'd need to do the number of organisms initially caught in that first sample, times by the number of organisms in the second sample, divided by the ones that had the mark on them. So that would then give us, in this example, we've got 20 butterflies were initially caught. In the second sample, there were 22 butterflies, 9 of which had the mark. So we do 20 times 22 divided by 9. That comes to 48.89, but we need to have whole numbers here because it's numbers of organisms. So I'm going to round up to 49 butterflies. There's always a mark for stating this formula when you have a method question for mark, release, recapture. So if in doubt, always state that formula. There's questions linked to the ethics of this sampling as well. So you need to consider that when you're marking the initial sample, that the way you mark them is not with a toxic chemical. So if you are putting on paint, it has to be non-toxic. It mustn't increase their chance of being seen by predators and therefore eaten and killed. And it mustn't reduce their chances of reproduction. So those are the ethics. Now, a non-ethical consideration of the mark is it needs to be weather resistant, because you don't want it to be something that's going to wash off when it rains, because then they're not going to be marked anymore. There's also assumptions linked to this. The estimate might not be accurate because the population size isn't actually constant, and we are assuming it is. And for a population to be constant, that means you have no births, no deaths, no migration. And that doesn't happen in any population. So we're assuming the population size is constant. In reality, it's not. So that's one reason why our estimate will be inaccurate. And also, we're assuming that the animals, when we release them and leave them to evenly distribute, we're assuming that they are going to evenly distribute. But in reality, they don't often do that, because lots of animals will probably huddle together near a water source or a food source, or where they feel safest. Then we go on to succession, and this is the change in ecological community over time. A primary succession starts with a pioneer species, and that is going to colonize. It might be bare rock or sand. You don't have any soil yet. You don't have to know any examples, but just to give you a bit of information, pioneer species could be things like lichen. Lichen are a mixture of fungi and algae, and they can survive in really harsh abiotic factors. And through the death and decomposition of the lichen, or whatever the pioneer species is, that is going to change the environmental factors, making them less harsh, less hostile, and you get this thin layer of soil, and then larger plants are able to survive there. And that might be things like mosses or smaller plants. And they will further increase the depth of the soil and the nutrient content, because they'll have roots that will start to hold the soil together. And as they die and decompose, they're going to add more nutrients to the soil. And this pattern continues over and over. Each new species is going to improve the environment so it's less hostile. But that then does mean that bigger species, plant species, can arrive and they'll outcompete the previous one. So the changes that organisms produce in the abiotic environment can result in a less hostile environment and therefore increases the biodiversity. And the final stage of a succession is known as the climax community, and this is dominated by trees. Now, to give you the marking points, because that was more a full explanation, the actual marking points on a long answer question are very brief. There's always a mark for saying it's initially colonized by a pioneer species. Next, it's saying the pioneer species changed the environment. The environment becomes less hostile for new species. There's an increase in biodiversity, and it finishes with a climax community. That would get you full marks on a 5 marker for describing succession. So screenshot that or turn it into a flashcard to learn those key points. So here's our summary from succession. The species richness increases, the number of organisms increases. That's why the biodiversity is higher. So as succession occurs, we get larger plants. That is going to attract animals as well, and you end up with these more complex food webs. Now, you can prevent succession from progressing, and that is a technique to conserve habitats. And this is because as a succession moves through its stages, you ultimately get a climax community where you have lots of large trees. And those large trees will outcompete the smaller plants because they're going to be shaded. So if you just let succession occur, all of the area would be forest. And that's what it used to be in England, for example. It used to just be forest everywhere in the UK, not just in England. But then we cut down the trees, started to build. But you can manage forest as a way to maintain maybe one of the earlier stages of succession. to have one section of the forest thriving with grasses, another section of the forest thriving with smaller plants, and then some of the sections thriving with trees. And the idea here is you get a greater variety of habitats. That then means you've got a greater variety of food sources, and a greater range of animal species will be able to survive there, and also plant species. So you might do things like coppicing or pollarding, where you cut down some of the trees, but you don't take out the roots, because then over time the trees can still regrow, so it's not going to be harmful for the environment. And also it means you get timber for furniture, or you could use the wood for fuel as well. Topic 8 for AQA A-Level Biology, and we begin with looking at 3.8.1, alterations of the sequence of bases in a DNA can only alter the structure of proteins. So basically it's mutations, and a gene mutation is the alteration of a base in the sequence of bases for one gene. And this is probably going to occur during DNA replication, which is in S phase of interphase in the cell cycle. Gene mutations occur spontaneously, but the frequency at which they occur can be increased by exposure to mutagenic agents, and that's things like carcinogens and UV light or ionizing radiation. And because a mutation can change the DNA base sequence in a gene, that means it could result in a different sequence of amino acids in a polypeptide. And if that sequence of amino acid changes, then that can alter where the tertiary structure bonds will form, like the hydrogen ionic bonds, and that could result in it folding into a different 3D shape. And if a protein has a different 3D shape, it will have a different function and potentially make it a non-functioning protein. Alterations to genes like this can result in a mutation that could cause cancer, and cancer comes up a little bit later in this topic. So there are 6 different types of gene mutations, 2 of which you learn in topic 4, but now you learn addition. You've already covered deletion and substitution in topic 4, inversion, duplication and translocation of bases. So addition mutations is when one extra base is added to the DNA base sequence. So if we have a look at this original sequence here, we've added in an additional adenine nucleotide, and as a result we get a frame shift, meaning all of the bases downstream of that mutation shift one position, and they all shift one position to the right. And that causes a change in all of these codons, and therefore they will probably code for different amino acids, and that will result in a very different amino acid sequence and a non-functioning protein. Deletion is when one of the bases in your DNA base sequence is deleted. This also causes a frame shift, but this time all of the bases downstream of the mutation shift one position to the left. Again, you get different codons, therefore it's going to code for different amino acids and a very different amino acid sequence, and therefore a non-functioning protein. Substitution is when only one base is swapped, so it's just changed. You still have the same number of bases, and it will only affect the one codon that base is in. And because the genetic code is degenerate, meaning you have multiple codons coding for the same amino acid, even though this mutation has happened, sometimes it still results in the codon coding for the same amino acid, and therefore has no overall impact on the final protein. And if the mutation happens in the introns within a gene, which are the non-coding regions, again, it's not going to change the amino acids that it codes for. So here you can see an example. We've just changed the thymine nucleotide into an adenine nucleotide. Inversion is when a section of bases will detach from the DNA sequence, but when they rejoin, they are inverted, meaning they're back to front now. So this section of the code is back to front, and you get a completely different amino acid sequence. So we can see our original DNA here, and now we've got this section must have broken off, and when it rejoined, it's reversed itself, it's inverted. So that is going to code for 2 completely different amino acids now. Duplication is when one particular base is duplicated at least once. And because that means we're adding in bases, it's going to cause a frame shift to the right downstream of that mutation. So you end up with a different sequence of amino acids. So here's an example here. We've got the base thymine, added twice. And then lastly, translocation is a section of bases on one chromosome detaching, and it attaches onto a different chromosome. This is a substantial alteration, and therefore it can cause significant impacts on gene expression, and therefore the phenotype could be very, very different. 3.8.2 then is gene expression. And we start with stem cells. And in multicellular organisms, there are a diverse range of specialized cells, but they all originated from undifferentiated stem cells. So a stem cell is an undifferentiated cell, meaning it hasn't become specialized yet, that can continually divide, and it can then go on to differentiate to become a specialized cell, as we can see here. And there's different types of stem cells based on their ability to differentiate. So we've got totipotent, pluripotent, multipotent, and unipotent. Totipotent stem cells can divide and produce any type of body cell. So these are able to differentiate into all cells. And they occur for a very, very limited time in early mammalian embryos, so shortly after the sperm and egg fertilise. Pluripotent are still found in embryos, so we can see here, you don't need to know this term blastocyst, but the embryo then develops into this blastocyst. The cells on the outside go to form the placenta, and the ones here in this inner mass are the pluripotent stem cells. So these pluripotent stem cells can differentiate into almost any type of cell, just not the placenta, because those are the ones on the outside that have already started to differentiate, but any other cell. And these are the ones that are typically used in research or to treat human disorders. So the stem cells could be used to regrow damaged cells in humans, such as to replace burnt skin, or even beta cells to try and treat type 1 diabetes, or neurons to treat Parkinson's disease. There are issues with this though, as sometimes the treatment doesn't work, or because stem cells can continually divide, they do continually divide and they can form tumours. There is also debate as to whether it is ethically moral, because in order to use it for treatment, you would have to make a therapeutic clone of the person you are trying to treat. And the embryo is then destroyed once the pluripotent stem cells are removed as well. So we also have multipotent and we have unipotent stem cells, which are found in mature mammals, meaning after the embryo stage. And they can divide to form a limited number of different cell types. Multipotent cells are, for example, bone marrow cells that we have as adults or even as children. When we say adults, we just mean you've been born in this instance. And they can differentiate into a limited number of cells, such as all of the blood cells. Whereas unipotent cells, uni meaning one, they can only differentiate into one type of cell. And an example of that is the cardiomyocytes. They are unipotent stem cells which regenerate into new heart muscle cells in small numbers. Now, induced pluripotent stem cells, this is something that has been produced scientifically from adult body cells. Somatic cells means body cells, using appropriate protein transcription factors to basically ensure that all genes can be transcribed again. And therefore, it makes it pluripotent in property, even though you might have taken a multipotent or a unipotent stem cell. And this overcomes some of the ethical issues because you're not then creating a clone embryo, doing therapeutic cloning, and you're then not destroying embryos. You're getting permission from an adult, taking some of their body cells and reverting them back into a pluripotent state. So the way this is done then, the iPS cells are created from the adult unipotent cells. These are altered in the lab to return them to that state of pluripotency. The genes that were switched off, which basically means those genes could not be transcribed, are now able to be transcribed again. And this is done using transcriptional factors. iPS cells are very similar to embryonic pluripotent stem cells, but as we said, it doesn't have those same ethical issues, such as the destruction of an embryo and not being able to get permission. have shown a self-renewal property in that they can divide indefinitely to give limitless supplies. And for these reasons, they could be used in medical treatment instead of embryonic stem cells. So if we have a look, then we've talked about this use of transcriptional factors in induced pluripotent stem cells, but we haven't gone through what that means. So you can control transcription. You can also control translation, but we're just going to focus on how you can control transcription first, meaning you can prevent it from happening or initiate it to happen. And this is actually what happens naturally when cells differentiate and become specialized, because every cell in the body has your entire genome, so all of the genes. But because of transcriptional factors and other things, but mainly transcriptional factors, some of those genes don't get transcribed, some of them do. And that's how you get cells that produce a particular range of proteins and become specialized. So in eukaryotes, transcription of target genes can be stimulated or inhibited using transcriptional factors. And that's what we're going to have a look at. So transcription of a gene will only occur when a molecule from the cytoplasm enters the nucleus and it binds specifically to the promoter region on DNA once it's in the nucleus. And those molecules that do that are proteins, and they're called transcriptional factors. Each one can bind to a different and particular base sequence of DNA. And this links back to topic 1, knowing that because transcriptional factors are proteins, that means they have a unique tertiary structure. So it gives it that unique 3D shape. And that means it will be able to bind to a sequence of bases complementary in shape. So remember that concept of tertiary structures and complementary shapes. So once it does bind, it can then stimulate RNA polymerase so that transcription begins. And that is how it's going to be initiating transcription. So we do then get an mRNA molecule that can then go on to be used in translation. So without the binding of that transcription factor, the gene is technically inactive because transcription is not happening, and therefore that protein isn't being made. And an applied example that you need to know about is estrogen. Now, estrogen is not the transcriptional factor, but it activates the transcriptional factor. Estrogen is a lipid-soluble hormone because it's a steroid hormone, and steroids are a type of lipid. So that lipid-soluble hormone, which is estrogen, can dissolve and move by simple diffusion from outside of a cell into the cytoplasm. And when it's in the cytoplasm, there's a transcription factor that has a receptor on it, which is complementary in shape to estrogen so that they can bind. And when it does bind, it causes a change in the tertiary structure of that transcription factor. And that change in the tertiary structure changes the DNA binding site so that it is now complementary in shape to a particular sequence of DNA bases. So that activated transcriptional factor moves into the nucleus. It will then bind to the promoter region of a target gene, which has the specific DNA base sequence that it's complementary to. And when it does that, it stimulates the RNA polymerase to bind and transcription can occur. And then we get our mRNA molecule that can be used in translation. There's also another way that transcription can be controlled, and that is through epigenetics. Epigenetics, here is your definition, is the heritable change in gene function, but without changing the DNA base sequence. So 2 marks there for those 2 parts of the sentence. And these changes are caused by changes in the environment. So for example, stress and diet. And it can result in transcription being inhibited or initiated. And that is due to particular chemical tags that bind either to the DNA or to these histone proteins. So one of those chemicals is methyl groups. And methylation of DNA means methyl groups binding to DNA, which we can see here. These green circles are representing methyl groups binding to the DNA. And when those methyl groups are added to DNA, they attach to cytosine bases only. And this prevents transcription factors from being able to bind. And that is because when the methyl groups bind, it's going to attract the DNA and histone complex really tightly together. So it winds up like this. And as a result, 'cause it's so tightly coiled, you've got genes which are tightly wound up, and therefore transcriptional factors can't reach them to bind. That means RNA polymerase can't be stimulated and transcription doesn't happen. The next chemical group is the acetyl groups. And if you have acetylation of histone proteins, that means the addition of acetyl groups, and they can bind to the histone proteins. Now, an acetyl group has a partial negative charge because it's got an oxygen in it. And the DNA also has this partial negative charge because it's got a phosphate group in it. And those two negatives will repel. And that's why the DNA and histone complex is not as tightly coiled. And as a result, the DNA is accessible. Transcriptional factors can bind to the promoter region on the DNA and stimulate RNA polymerase. So that means if you have decreased acetylation, meaning the acetyl groups are being removed from the histone proteins, that makes the histones more positive, and therefore they're going to attract to the DNA, and your DNA and histones are going to be more strongly associated, and it coils back up again. And again, that means that transcription factors can't bind. So that means that if you have increased methylation and decreased acetylation, that results in this tightly coiled complex of the DNA and histone proteins, and that will inhibit transcription. Whereas if you have methyl groups removed, so decreased methylation or increased acetylation, then you get this complex where it is not as tightly coiled and the genes might be exposed so that transcriptional factors can bind, and that initiates transcription. Now, you can also have control of translation after transcription has happened and mRNA has been made. And that is what RNA interference is, or RNAi for short. In eukaryotes and some prokaryotes, translation of the mRNA produced from the target genes can also be inhibited because of the RNA interference. So the mRNA molecule has already been transcribed, but that mRNA molecule gets destroyed. It basically gets cut up into small pieces, so it can't then attach to the ribosome and be used in translation. And small double-stranded RNA molecules are involved in this, typically known as siRNA, so small interfering RNA, or miRNA, microRNA. So here's our flow diagram of how protein synthesis typically happens. We've got transcription, that results in the production of mRNA. mRNA is then translated, and that results in the production of a polypeptide chain. So that is how protein synthesis occurs. Now, we've already talked about transcription could be prevented because, or initiated because of transcriptional factors and epigenetics. But here is where our siRNA or RNA interference has a role. So siRNA can cut up the mRNA to prevent translation, and therefore small interfering RNA results in RNA interference. So RNAi, the reason this is important is it's regulating gene expression, controlling development, and also it can fight viral infections and potential therapeutic uses for this could be silencing harmful genes and disease. So that's when it's going to be naturally occurring, and it could also be used in medicine in this way. The way this works then is, in terms of siRNA, which we can see here, siRNA is produced from double-stranded RNA, and it splits into a single strand. It then is going to bind to a protein to make a complex, and one strand of that siRNA will bind to a complementary sequence of mRNA that's just been transcribed. And once it's bound to the siRNA in that protein complex, that protein is actually an enzyme that can cut up the mRNA. That is the level of detail that you need to know. I know it might sound a little bit vague, and that's because it is, but that's the extent that you need to know. It quite often comes up as application questions where they then give you a little bit more information. An alternative is miRNA, which was the microRNA, and miRNA is not fully complementary to its target mRNA molecule. So instead it binds to multiple mRNA molecules, And that often causes the blocking so that the mRNA can't attach to the ribosome, or it can also cause mRNA degradation as well. But either way, it results in translation not happening. So this then follows on to cancer. And cancer can result from mutations in genes that are responsible for controlling and regulating mitosis, as well as we're going to have a look at the role of epigenetics as well. But first of all, if these genes do get a mutation and a non-functioning protein is made, then that means mitosis is not regulated and you get uncontrolled mitosis or cell division, and that results in the creation of a tumour. Not all tumours are cancerous. They can be benign or malignant. So benign tumours, these are not cancerous. They grow, or they can grow very large, but just at much, much slower rates. And they're non-cancerous because they produce this adhesive molecule which sticks all of those cells together in a particular tissue, and they're often surrounded by a capsule. So that means that they're much easier to remove by surgery because you can remove the entire tumour, all the cells, because they're encapsulated. And also because it's in that capsule, it's localised, it's not going to break off and spread to other tissues. So it's often not life-threatening. However, that does depend on the location of the tumour because it will continue to grow and the treatment is surgery to remove it. So if it's in a location of the body where surgery would be difficult to remove it, such as in the brain, it could still be life-threatening. Malignant tumours are cancerous and they can grow large and they can grow large very rapidly. The cell nucleus becomes large and the cell can then become unspecialised again. They don't produce that sticky adhesive layer and they also don't have a capsule around them, which means that these tumours are able to metastasize. And what that means is cells from the tumour can break off and get into the bloodstream or the lymphatic system and therefore move around the body and then lodge in new tissues to make secondary tumours. The tumour, as I said, isn't encapsulated, and that also means it can grow out projections and reach blood supply. And as soon as it gets a blood supply, it's got lots of glucose and oxygen for respiration, which is why it can divide so rapidly. It can be life-threatening, and as well as trying to remove tumours, you can also have treatments such as radiotherapy and chemotherapy. But it's much harder to successfully remove the tumours because they're not encapsulated. It's very hard to tell if every single cancerous cell has been removed in surgery. And that's why it's often, if you do have surgery, accompanied with radiotherapy and chemotherapy to potentially kill any cells that might have been left behind in the surgery. So tumour development can be due to gene mutation in either a tumour suppressor gene or an oncogene, which are two genes involved in controlling mitosis. Or it could also be linked to epigenetics if there's abnormal methylation of a tumour suppressor gene or oncogene, or increased estrogen concentrations linked to the idea of transcriptional factors. So let's just go through what these two genes are. Oncogenes are the mutated version of a proto-oncogene. And a proto-oncogene creates a protein involved in initiating DNA replication and mitosis when the body does need new cells. An oncogene mutation results in that gene permanently being activated. So you are going to have the protein constantly being produced to initiate DNA replication for cell division, even if new cells aren't being made, and that's why a tumour forms. Tumour suppressor genes produce proteins or code for proteins that slow down cell division and cause cell death. So when you no longer need new cells, it make the protein, and also cell death if a copying error is detected, so that cell needs to be destroyed. Now, if a mutation results in the tumour suppressor gene, that means you won't make that protein anymore, and therefore cell division isn't going to slow down. And also potential mutated cells aren't identified and destroyed. And again, that is why it can result in a tumour forming. So if you have abnormal methylation, which links back to epigenetics, that means that this methylation could result in a gene being inactivated. And if that's the tumour suppressor gene, becoming hypermethylated, meaning lots of methyl groups are binding to that DNA. That's going to cause it to tightly coil. The transcriptional factor can't reach the gene, the tumor suppressor gene, and the tumor suppressor gene is not transcribed. So it means you don't get that protein that's needed to control the cell cycle. Or the opposite could occur in an oncogene, as they might be hypomethylated, meaning you don't have enough methyl groups binding, and as a result, the gene is permanently being transcribed. Now, the link to estrogen is after menopause in females, estrogen is no longer produced by the ovaries. While the menstrual cycle is active before menopause, the ovaries is the main place that estrogen is produced. But after the menopause, the ovaries stop making estrogen, and instead it's mainly produced by fat cells in the breast tissue. So in postmenopausal women, this local estrogen production, constantly being exposed to the breast cells, increases the risk of breast cancer. And that goes back to what we looked at in transcriptional factors, because estrogen can bind to receptor proteins on transcriptional factors to activate the transcriptional factor. And that transcriptional factor can then bind and initiate transcription. So if estrogen stimulates transcription of a proto-oncogene, it can cause that gene to be constantly transcribed, and therefore you're going to be making that protein all the time that stimulates cell division, and that can contribute to a tumour forming in the breast tissue, because that is where this estrogen is being produced. And this can result in a positive feedback loop, because if you're making more tumour cells, which are breast tissue cells that are making estrogen, your concentration of estrogen is now even higher in the breast tissue, so this process is going to happen even more. So that is why it can then happen very, very rapidly. Next, we move on to 3.8.3, using genome projects. So genome sequencing involves working out the entire base sequence of an organism's DNA. And projects have successfully sequenced the genome of many organisms, including humans. So this is just showing us the progress through that. It's a bit blurry, but many organisms have had theirs sequenced, and for humans it was finally completed, the Human Genome Project, in 2003, but it had been ongoing for many, many years. Scientists across the world all contributing to sequence our DNA. So the applications of this, first of all, in simpler organisms, for example bacteria, genome sequencing is very useful because it can be used directly to predict the proteome, which is all the proteins an organism can make in their cell. And that's possible because simple organisms like bacteria don't have non-coding DNA. They don't have any introns, they don't have other non-coding sequences, and also they don't have regulatory genes as well. So that means the relationship between the genome and the proteome is very straightforward, and it's very easy to predict all the proteins they'll potentially make. And this has been useful in identifying potential antigens that bacteria could make, and therefore making vaccines against those particular bacteria. In complex organisms, for example humans, predicting the proteome after working out our genome is more difficult, and that is because we've got large amounts of non-coding DNA, and also we have regulatory genes that affect gene expression. So that means that knowing the genome sequence doesn't always directly reveal which proteins will be produced. Now, there are constant advances in sequencing technologies, which is why the spec doesn't get you to learn any, because they probably become outdated within a year. So that's why instead you just need to have this awareness that sequencing methods have become increasingly rapid, far cheaper and cost effective, and automated. And because of that, large-scale genome projects have enabled researchers to sequence even more organisms in less time with greater accuracy. So we've gained even more information. We then move on to 3.8.4, gene technologies, and I mean the title goes on, but it's basically what we all call gene technologies. So gene technologies involves recombinant DNA. And recombinant DNA means you're transferring fragments of DNA from one organism or one species into another organism's DNA or another species' DNA. The organism that receives the DNA is called a transgenic organism or genetically modified. The reason it's possible links back to topic 4. The genetic code is universal, which means the same codons on mRNA will code for the same amino acids in all organisms. And that's why you can take the DNA from one species, put it into the DNA of a different species, and it will still code for exactly the same sequence of amino acids and make that protein. Now, as well as that, it also works because transcription and translation are universal as well. So that means genes from one species can be expressed in another. And that's because once you insert that DNA into the new species DNA, transcription will occur to make mRNA, and that mRNA will then be translated to make the functional protein in the host cell. So the ways that this has been used is producing insulin, for example, taking the human gene for insulin, putting it into bacterial DNA, and then bacteria will make the human protein for insulin. And that has been massively beneficial because bacteria can create large quantities very rapidly of insulin, which can be used to help treat diabetes. It's also useful to make transgenic crops with beneficial traits, for example, inserting genes so that they're naturally pest resistant, or even inserting gene function for research models to try and work out the role of genes in terms of helping with medical research. So how this is done then, I've split the rest of this topic into this flow diagram because from my experience teaching this, one of the challenges with the gene technologies topic is students learn all of the technology separately and don't see how it all links together and therefore struggle when it gets to the challenging application questions when it is all linked together. So here it is and how it all links. One branch is genetic screening, and this is where you might screen to see if you have a particular gene. That leads to genetic counselling. Now this is done using gel electrophoresis and it can result in personalised medicine. Genetic fingerprinting, that is how you can examine an individual's DNA based on the VNTRs, those variable number tandem repeats. And that is used in forensic science, medical diagnosis, paternity tests and more. And one of the stages in genetic fingerprinting is gel electrophoresis. Then we have over here how we actually make recombinant DNA. And the first step is if we want to put the DNA from one organism into another, we need to get that piece of DNA to start with. And that's what creating DNA fragments means, getting the gene of interest that you want to insert into a different species DNA. So here are the processes of how you can do that. Once you've then isolated that gene of interest, we then need to get lots of copies of it before we go on to try and insert it. And you can either get large numbers of copies in vitro, which means outside of the body, and that is using PCR, polymerase chain reaction, or you can do in vivo cloning, which means making large numbers of copies of that gene inside of a cell. And that's typically done inside a bacteria, and here's all the processes how. So that's a very brief overview. Let's go into it in more detail, starting with how we create these DNA fragments. So the combining of different organisms' DNA, which could enable scientists to manipulate and alter genes to improve their industrial processes, or we looked at medical treatments, always starts with you have to get your gene of interest, which means isolating the DNA fragment. And there's 3 methods: reverse transcription, restriction endonucleases, and the gene machine. So reverse transcription, this is using an enzyme. And this enzyme makes DNA copies from mRNA. The enzyme actually isn't written yet, but it is reverse transcriptase. So that enzyme is an enzyme that can make DNA copies from mRNA. And it naturally occurs in viruses. So you learn in topic 2 how HIV has the enzyme reverse transcriptase, and that can make DNA copies from mRNA. So the reverse of transcription. A cell that naturally produces the protein of interest gets selected. Then large amounts of mRNA for that protein should be in that cell and they can be isolated. You would then mix that mRNA that you've isolated with the enzyme reverse transcriptase. And what that will do is reverse transcriptase can join DNA nucleotides together, so via a condensation reaction, creating phosphodiester bonds, based on mixing together DNA nucleotides, exposing it to this mRNA, and the complementary bases will be attracted and aligned, and then reverse transcriptase can join to create that new DNA molecule. But because we'll have one mRNA sequence, and then you get your DNA sequence aligning and being created against that, you end up making a single stranded DNA molecule. And we call this cDNA, complementary DNA, so complementary copy of our mRNA. So we then do have to make it double-stranded so that we can use it. And to make it double-stranded, we then need to make sure we've still got lots of DNA nucleotides present, and then we use the enzyme DNA polymerase. Now, the advantage of this method is that complementary DNA or cDNA won't contain any introns because mRNA doesn't have introns in it. They've already been spliced out. So that means we get a DNA copy with no introns in. And then we can see in this diagram, they then go to use PCR, so you get large numbers of copies of our cloned gene. Restriction endonucleases, this is another method using enzymes, and these are enzymes that can cut DNA. They naturally occur in bacteria, and bacteria have them as a defense mechanism. So if a virus was to inject their DNA into the bacteria, they naturally have restriction endonucleases to cut up viral DNA so that they can't be infected. And there are many different types of restriction endonucleases, and this links back to the idea of enzymes having a tertiary structure, which gives them a unique 3D shape, so their active site is complementary in shape to a particular sequence of DNA bases. So these restriction endonucleases, loads of different ones, so that they each have a different active site complementary to a different sequence of bases. They can then bind at that particular sequence, and they will break the phosphodiester bond at that point. Some enzymes cut at a location in a double strand to get a blunt end. What that means is it would cut directly down, so you get two straight lines, a blunt end. But most of the time, and in terms of use, it's more useful than they would use enzymes that cut at a jagged line. And that then gives you an overhang of bases here and an overhang of bases there. And that is known as sticky ends. And sticky ends are useful because that means that they can align opposite another fragment with an overhang, and it's much easier then to join them together because the complementary bases can align. And this is going to happen at these palindromic sequences. And that means the bases, the order of the bases is the same forwards as the order of the bases is backwards. That's what we mean by palindromic. So where these enzymes can bind and cut is known as the restriction site or recognition sequence. Those are the two terms that we use to describe where the enzyme can bind. That is based on the active site being complementary to a particular sequence of bases, and it creates these sticky ends. A gene machine, this is when the DNA fragments can be created in a lab using a computerized machine. So first of all, the scientists will examine the protein of interest to identify what the amino acid sequence is for that protein. Then they can work backwards and work out what the mRNA sequence is, and therefore what the DNA sequence is. That DNA base sequence is then entered into the computer. Checks are made that you're not trying to make a protein that could potentially cause risk to human health, and other biosecurity checks, and that it's ethical to produce. Once those checks are in place, then the computer can create small sections of overlapping single strands of nucleotides, and these are called oligonucleotides. Those can then be joined to create the DNA for the entire gene. So once you've then created the entire gene, PCR is used to get large numbers of quantities and to make it double-stranded. So this process, the advantages are it's very quick, it's accurate, you can design it to be intron-free as well. And the reason we think intron-free is an advantage is prokaryotic cells don't naturally have introns. That means they don't have the cell machinery to splice introns out. So if we're going to be putting the gene into a bacteria, we need to remove the introns because they can't. So having it intron-free in the first place is a big advantage. Okay, so we've made our gene fragments. Now let's have a look at how you can clone them, meaning get large numbers of that gene of interest. First of all, with in vivo cloning. So we've already created the DNA fragment. Now we need to look at how we get that fragment into the vector and how we get that into the host cell. So the first thing though is adding a promoter and terminator region. So when we insert a gene into a host cell, so basically when we take one gene and put it into the DNA of another organism, it's not enough to include just the DNA base sequence that codes for the amino acids. For transcription to successfully occur, you also have to add a promoter region at the start of the gene. And the DNA sequence is inserted before the gene. And that acts as a binding site so that RNA polymerase and transcriptional factors have something to bind to so they can initiate transcription. The correct promoter must be used for the host organism though, so that the gene is expressed properly. Now at the very end of the gene, a DNA base sequence is added as well, and that's called the terminator region. And that will signal the end of transcription and it ensures that RNA polymerase will detach and it stops transcribing at the correct point. So they are important then because without a promoter region, the gene wouldn't get transcribed. And without a terminator region, transcription might continue so that it then continues to transcribe unintended regions. And that would be wasteful of resources and also it might disrupt the other genes. So basically these sequences, the promoter and terminator region, are essential for being able to produce functional mRNA when you do this gene technology. So how we get that DNA into a vector? But first of all, what is a vector? So a vector is something that will carry the DNA fragment that we've made into the host cell. That's what a vector is. And the most common vector that we use is plasmids, so those small loops of DNA that some bacteria have. So that's what we're going to be doing, getting our DNA into the plasmid first of all. And the way that we do that is the plasmid has to be cut open using a restriction endonuclease, but you have to use the same restriction endonuclease as the one that you use to cut out your DNA fragment. And the reason for that is that means your plasmid and your DNA fragment will have the same sticky ends. So that means they'll have complementary sequences overhanging that can align. And therefore, they are going to be able to align and join. Now, a few things just to give you extra detail that the mark scheme's more particular on now is you need to know the enzyme ligase is going to be joining the plasmid and the gene of interest together. But specifically what ligase does then is it's catalyzing the condensation reaction, forming the phosphodiester bonds between those nucleotides to join those two fragments together. So now we've got our gene of interest into the vector, the plasmid, we have to get that vector into the host cell, which is the cell that's going to make the protein, which in this case is a bacteria. And in order to do this, we need to make the host cell's cell membrane more permeable, so we're more likely to get the plasmid in. And the way that we make the cell membrane more permeable to increase the likelihood of the plasmid getting inside is using calcium ions and heat shock, which means sudden increase and sudden changes in temperature. So that's going to affect the fluidity of the cell membrane, so we should be more likely to get the plasmid in. So we've now cloned it, but what we have to do is make sure that before we grow that bacteria, we are only growing the bacteria that has definitely taken up the recombinant plasmid. And that's what we mean by identifying transformed cells. A transformed cell is a cell that has taken up the recombinant plasmid. Recombinant means recombined. It's made up of two different types of DNA. Now, not all of them will be recombined or transformed cells because, first of all, maybe that recombinant plasmid, even though we heat shocked the cell membrane, it still didn't get inside. Or maybe a plasmid did get inside, but it wasn't a recombinant one. The original plasmid that was cut open simply rejoined, so maybe that got inside, so we don't have the gene of interest. And sometimes that DNA fragment that we created actually connects back around to itself and makes its own little loop of DNA. And if that went in, then again, it wouldn't work. So we have to identify which bacteria have successfully taken up the recombinant plasmid. And that's done using something called marker genes. And marker genes are on the plasmid and used to identify which bacteria have taken up the plasmid. There's 3 marker genes that are typically used. Genes that are coding for antibiotic resistance, genes coding for the creation of proteins that fluoresce, so they go bright yellow or green, particularly under UV light, and genes coding for enzymes that can catalyze reactions causing colour changes. Now, you don't need to know the details of how that is done, but it's a common application question. So that's why I'm going through this. You aren't expected to remember this, but it comes up as application a lot. So that's why I'm going to go through this example with you. So I'm going to do it with antibiotic resistance. And we can see that here is the plasmid. And there are 2 genes for antibiotic resistance. This one here makes the bacteria resistant to the antibiotic tetracycline, and this gene here will code for a protein that makes the bacteria resistant to the antibiotic ampicillin. And here is the DNA fragment that we've just isolated. Now, what they would do is deliberately insert that DNA fragment into one of the marker genes. And in this case, they've put it in the middle of the tetracycline gene. And what that does is our DNA fragment can be transcribed and make a protein, but the tetracycline one has been split in half now, so that is not going to be able to be transcribed and translated to make the protein that makes it resistant to tetracycline. So any bacteria that takes up this plasmid will not be resistant to tetracycline anymore, but the resistance to ampicillin gene is still intact, so it will be resistant to ampicillin. So to work out which bacteria have taken up the plasmid, the recombinant plasmid, you would grow your bacteria on agar, and we can see all the colonies here that have grown, and we've labelled them. You would then use a sterile velvet block to make an imprint of this master plate and copy all of those colonies onto a second agar plate. And this time the agar plate has got agar with ampicillin antibiotic dissolved within it. And we can see that now some of the colonies have died and only some can grow. They would then take another imprint and this time grow those colonies on the agar plate that's got tetracycline dissolved within it. And now we can see only these 3 colonies can survive. And from this, we can work out which colonies contain the recombinant plasmid, because our bacteria that have the recombinant plasmid are resistant to ampicillin, because that gene's intact. So that means it has to grow on this plate, but it's not resistant to tetracycline anymore, which means tetracycline will kill it. So that means it won't be present on this plate. And that is only colony E and G, because they're on this one, they're not present on this one. So that tells us that E and G are the only colonies of bacteria that have that recombinant plasmid. So those are the ones that you should then remove and grow en masse. Next then is how you could do this, but using PCR. So PCR is our other way to get large quantities of the DNA fragment of interest. And vitro means not inside of a living cell. It's in a lab. So you can use the polymerase chain reaction to do this. It's an automated machine, so you get large quantities very rapidly. And PCR is going to be used probably multiple times per day in all genetics labs, because it's so quick and you always want large numbers of copies of everything so you can do multiple repeats, and in case everything goes wrong, you've got your extra copies. So your PCR machine involves this thermocycler machine, and you would need to add your DNA fragment that you want to be amplified. You need to add the enzyme DNA polymerase, a particular type called Taq polymerase, primers, and DNA nucleotides. So Taq polymerase, this is a version of DNA polymerase that is taken from bacteria that are extremophiles, meaning they can survive in very, very high temperatures like hot springs. And as a result, they've evolved to have a version of DNA polymerase, Taq polymerase, that has an optimum temperature far higher. So we use this so that we can increase the temperature very high in this machine, so the reaction happens even faster, but the enzyme doesn't denature. Primers, these are short sequences of DNA bases that are single-stranded, and they are needed to enable DNA polymerase to attach. So that's what we need to add into it. And then the process is, initially the temperature is increased to 95 degrees C. Now this is done so that the DNA fragment that you've added is going to have the hydrogen bonds break and it separates into two single strands. Then we decrease the temperature to 55 degrees C. And we need to do that so that hydrogen bonds can form, because our DNA primer, which is that short sequence of DNA bases complementary to our sequence on our DNA fragment of interest, those are going to be attracted and aligned, but we need the temperature to be dropped so that hydrogen bonds can form between those two. Then we will increase the temperature so that the enzyme DNA polymerase can then align all of these DNA nucleotides that are going to be attracted to their complementary bases. The DNA polymerase is then going to catalyze the condensation reaction, forming all of those phosphodiester bonds between the adjacent nucleotides. And that is at 72 degrees C. You do need to know those temperatures as well as this process. And that process will then happen on repeat. So you can end up with a huge quantity of DNA very rapidly. And this goes back to that maths formula that you can use. You would do 2 to the power of n, 2 meaning every time it doubles. N is the number of rounds of division that you allowed. So you could work out how many DNA molecules you'd have at the end. And if you didn't start with one DNA molecule, however many you started with, you would multiply by. So if we started with 100 fragments, we would then do 100 times 2 to the power of n. And if you left that going, let's say for 10 rounds, we would then do 2 to the power of 10 times 100, and that would tell you how many DNA fragments you'd have at the end. So the advantages are, it is automated, so it's more efficient, it's rapid, you can make 100 billion copies within hours, and it doesn't require living cells. That means it's quicker and less complex techniques are needed. So a few key terms as well that you need to be aware of. DNA probes, so this came up in the exams this year. They needed to define what a DNA probe is, and a lot of students struggled. A DNA probe is a short, single-stranded piece of DNA that is complementary to a specific allele or gene. Those probes are also labelled, usually with radioactive isotope or fluorescent marker, to allow detection. The process of DNA hybridization, this occurs when a DNA probe is going to bind to a complementary sequence of bases on a DNA sample. And to prepare the sample, the DNA is first extracted and heated so that it is separate strands. Then the probe is added so that it can bind and hybridize. And the point of doing this is you can then use the presence of that probe on a sequence of DNA to identify a target allele. And that's because if you've labeled it with a radioactive isotope, when you use X-ray film, it will be visualized, or if it's a fluorescent marker, it will visualize in UV light. So it's used, these DNA probes are used in medical genetics to screen individuals to see if they have particular alleles linked to inherited conditions such as cystic fibrosis or Huntington's disease, or the gene here linked to breast cancer. They are also used to identify variations in genes that affect drug response, allowing doctors to predict how a patient might react to certain medicines. And this kind of screening can help to detect individuals at increased risk of disease, even before the symptoms occur, which allows for early intervention, monitoring, or even preventative treatment. So this is what we mean by personalised medicine and genetic counselling. We're screening for the presence of that particular allele, selecting personalised medicines based on your genetics. And that's because some drugs are more or less effective depending on your genetics. And that enables it to be more effective to you, but also more cost effective as a treatment. And genetic counselling is a type of social work giving people advice and information following their screening of disease-causing alleles. So then we go on to how this process might occur using gel electrophoresis. And VNTRs are needed for this. VNTRs are variable number tandem repeats, and an organism's genome contains many regions of non-coding DNA, which are VNTRs. So they're short sequences of DNA bases repeated a variable number of times among individuals. And the number and length of the VNTRs is very different from person to person. And that is very useful in DNA analysis because VNTR patterns are inherited and highly specific. So the probability of two unrelated individuals having the same VNTRs is extremely low. So we can use VNTRs to examine relationships in terms of genetic similarities. Genetic fingerprinting is the technique used to analyse the DNA fragments containing VNTRs. And the process starts with the amplification of DNA using PCR so that you've got a large sample of DNA. The DNA is then cut into fragments using restriction enzymes, separating the DNA at specific sequences. And those fragments are then separated using gel electrophoresis, which will separate them according to their size, so the number of bases in the VNTRs. A DNA probe is then added, which binds to specific VNTRs, and the pattern of the bands forms a genetic fingerprint. And this pattern is unique to an individual, except for identical twins. So let's go through that whole process in more detail. Genetic fingerprinting. So first of all, you would collect your DNA sample. And the smallest sample of DNA can be collected, and that's because we're going to use PCR to amplify it afterwards. So any cell that contains a nucleus can be used. Then we have the digestion stage once we've collected and extracted the DNA. And the digestion stage is when we use restriction endonucleases to cut that DNA up at the recognition sequence or restriction sites. And enzymes which cut close to your target VNTRs will be added, and we get that palindromic sequence with those sticky ends. Now we need to separate out all of those cut up VNTRs. And the DNA samples are loaded into these tiny wells in agar gel. The gel is then placed in a dish which contains this buffered liquid, and an electrical voltage is applied. The DNA, because of that negative charge on the phosphate group, is going to be attracted to the positive end, and therefore the DNA is going to move towards this positive end through the gel. And the reason that it moves is, first of all, DNA is negative and it's attracted to the positive end, but it moves different distances because of how many DNA bases there are. The bigger a DNA base sequence, the heavier it is, so it's not going to move as fast or as far. If it's a shorter piece of DNA, fewer DNA bases, and that means it's lighter, so it can move through the gel much faster and it'll get further along. So that stage is gel electrophoresis, and that agar gel creates the resistance, and this is what I was saying about the smaller pieces can move faster and further. And that is how different lengths of DNA or VNTRs are separated. We then add an alkaline to make all of that DNA single-stranded. Then we can add our DNA probes to align to those single strands. So we said the DNA probes, those short single-stranded pieces of DNA, complementary in base sequences of VNTRs. Those probes will be radioactively or fluorescently labelled. So then if they hybridize, we then rinse it afterwards, and then the agar gel, this bit you often don't get assessed on, but the agar gel will eventually dry out and crack. So we do actually transfer it usually to another material that is longer lasting, and that can then be used to visualize your results. And here's one that I did over 10 years ago now, which is absolutely mad. I can't believe that was 10 years ago, but this is looking at the different VNTRs in bacteria, and we could then analyze it to see, here are our knowns, here's my unknown, and I could see my unknown had the same DNA bands as bacteria number 3. So I knew my unknown bacterial sample was the same species as bacteria number 3. So that can be used to interpret paternity tests as well. So we could look at the mother, the child, and any band that the child has that isn't from the mother must be from the father. And that's how we can use it to work out which person must be the father. And I think actually in this one, there aren't any that are a perfect match. Number 1 almost is, but there's this band here that isn't in number 1. So the uses of this genetic fingerprinting could be paternity testing. It could also be forensic science to identify if someone has their DNA at a crime scene, to place them at the crime scene. Medical diagnosis to see if you have particular alleles linked to inherited diseases to help with early diagnosis. Animal and plant breeding to make sure that you aren't about to breed 2 individuals that are very closely related. And also assessing genetic diversity to look at how much genetic diversity there is in a population. So that takes us to the end of topic 8. Hopefully you found it helpful, and if you did, don't forget to scan here for my mark scheme specific notes, which also have recall questions, examiner tips, summaries, key marking points, and essay links. On my flashcards, scan here where you can get mark scheme specific key points. But that is it for today's video. Hope you found it helpful, and I'll see you in a video very soon.